Okay, good morning everybody. Last week I was on a stroll and I thought that some things in life I'm very late at doing. I became a father when I was 41, so when you want to start a big family it's a bit late if you are 41. And now today this is my first international congress to be co-organizer of just before I retire. So for my CV it's not really a good addition, but still. Anyway, dear members of the Grasshopper Specialist Group, members and board of the Deutsche Gesellschaft für Orthoptologie, ladies and gentlemen in the audience and online, ladies and gentlemen. It took a bit of time, but finally the moment has come to host this combined meeting of the European Congress of Orthoptera, Conservation, and the 16th biennial meeting of the Deutsche Gesellschaft für Orthoptologie. On behalf of the organizing committee, Roy Kleukers, Boudewijn Audet and Rob Felix, and myself, a very warm welcome to all of you. We have an interesting program ahead, I think, and sufficient time, I hope, in between, for everyone to have a chat and meet and talk about things. Before sharing some household announcements and introducing the keynote speaker for today, I'd first like to give the floor to Eva van der Veer to officially welcome you on behalf of Naturalis. Please.
So, good morning all. So considering the snowfall of this morning, it must be April Fool's Day. So, dear grasshopper enthusiasts and conservationists, it is my pleasure and honor to welcome you here at Naturalis Biodiversity Center on the occasion of the 3rd European Congress on Orthoptera Conservation and the 16th biennial meeting of the German Orthopterological Society. So my name is Eva van der Veer and I'm the Director of Collections here at Naturalis. And to give you a brief institute to our facilities, I would like to start with a short video. Naturalis is the Dutch Knowledge Center for Taxonomy and Biodiversity Research and as such involved in various national biodiversity-related initiatives and projects. Our national commitment and central role is also expressed by the strong ties that we have with IJs, the Dutch Knowledge Center on Insects and Invertebrates. IJs is also the co-organizer and co-host of this meeting. When it comes to insects, Naturalis also has strong ties with the Dutch Entomological Society, exemplified, for instance, by the fact that we also harbor the entomological library. The fact that the entomological library is also housed here at Naturalis. We harbor one of the largest natural history collections of the world, and the insects collections, with over 17 million specimens, is the largest sub-collection here at Naturalis. The Orthoptera collection includes the collection of the former Rijksmuseum of Natuurlijke Historie, the Zoological Museum of Amsterdam, and the Willemse collections. In total, the Orthoptera collection consists of over 400,000 specimens, including primary types for some 500 taxa. its core originating from the Palearctic and Indo-Malayan region. Because of its collection and the very nature of its research, Naturalis has a strong international network and participates in some 250 projects with 180 organizations worldwide. Within Europe, for instance, Naturalis actively participates in CETAF, which is the Consortium of European Taxonomic Facilities, and Naturalis as a facility also leads DISCO, the Distributed System for Scientific Collections. This new building, which houses all of our staff and collections and includes a separate laboratory facility, opened in September 2019. In 2020, when this conference was originally scheduled, Naturalis celebrated its 200th anniversary. So most people know Naturalis as a museum, and despite COVID restriction, we were able to have almost 240,000 visitors in the past 7 months. In 2021, we also received the European Museum of the Year Award. And I know you all have a very busy schedule ahead of you, and today and tomorrow, but I hope you have a spare minute to visit our exhibitions. If you do so, please do not forget to visit our Life Sciences Hall, which is next to the entrance, where collections and research-related work is taking place under the watchful eyes of our visitors. So the program ahead of you includes a wide variety of topics concerning grasshoppers, with of course a special focus on conservation. And I would like to thank the organizing committee in advance for composing such a diverse program. International meetings like these are excellent opportunities to get to know your colleagues, to brainstorm about ideas, and to look for new ways of collaboration. And I'm sure this meeting will be no exception. So for now, I wish you a very pleasant, interesting, and successful meeting. Thank you.
Okay, thank you very much, Eva. Before introducing the keynote speaker of today, some household announcements. To start with, a very important message. The toilets are downstairs in the cloakroom, so across the hall in the cloakroom. If you're looking for information you cannot find in the program booklet, you can always look on the ECOC website for more information. This is a hybrid meeting, which means that there is also a YouTube streaming going on right now. The presentations we are going to see will be recorded, and you can look them back after the Congress. A very kindly request to all the presenters, please stick to your time, and to the chairs of each session to make sure that the presenters stick to their time. We have on the ECOC website, you can find PDFs of the posters which will be presented during this Congress. For the online audience, if you have questions, you have to use the chat in YouTube. We have a moderator here who will read out the questions to the chair of that moment. Another question I have, we realize that maybe sometimes people do not like to be photographed. If that's the case, we have stickers which indicate that you do not want to be photographed. If you do that, let me know, you get a sticker. Eva already mentioned that if you have time, visit the museum. Wearing the sticker means you can access the museum without any, it's free of charge for you. The Naturalis parking, I haven't got them yet, but I will collect some free exit tickets. So all of you who came by car, please come to me and I give you a free exit ticket. At 12:30 today, we planned a group photo. So after the last morning session of the day, we have planned a group photo which will take place on the big stairs in the museum. I'll come back to you once we finish the morning session. Then in the more open space behind us, there is several tables with books. Some of them are for free, some of them you have to pay some money for, and the name of the person you have to contact is indicated. And to that side, near the coffee table, there is a table with a lot of small carton boxes, which are mugs. Everybody can get one mug. It's a Congres mug. It's for free. Then when we have coffee and lunch, you should realize that you can take there are two stairs on the side. You can go up. The posters are behind this big screen. There's a poster wall where we will hang the posters for this Congress. But there's also a lot of chairs and tables also in that side. So if you want to have a chat with somebody, you can of course do it there. But there's a lot of other tables and chairs on the first floor there, in the back and along the sides here. And the last but not least message is that with the morning coffee today and tomorrow, we serve some pastry which is coming from the part of Holland where Roy and myself were born, from the very south. It is called, in Dutch, it's called vla. Okay, enough talk. Let's start with the serious business now. The keynote speaker of today is Thomas Vaartman. He's head of the Department of Biodiversity and Landscape Ecology at Osnabrück University in Germany, and also the chairman of the DGFO. His research interests focus on the effects of land use and climate change on biodiversity, and how species richness of ecosystems is influenced by disturbance events. Today, Thomas is our keynote speaker, and his presentation centers on Orthoptera conservation in times of global change. Thomas, please.
Luc, thank you very much for the kind introduction. I'd like to talk on our research on the effects of global change on Orthoptera in Central Europe, and I'd like to show what are the implications of our findings for Orthoptera conservation. First, let's have a look at our landscapes, how our landscapes look like. Here we see two fragmented calcareous grasslands. Here's one habitat patch, and here's the next habitat patch. And between these patches we have a matrix where the biodiversity is quite low, and such a landscape is called a fragmented landscape. And what we see is that the habitat patches differ in size. This one is quite large, this one is smaller, and they also differ in habitat quality. In this patch we have an abandoned patch. There's no longer any land use. We have encroachment of shrubs. This patch is still grazed, and we have open habitat structures. We have early successional stages. They are missing here. And here we see the main parameters that determine patch occupancy in our fragmented landscapes. And these are the area of the habitat patches. The habitat quality is the second, that is quite important. And the third one is the connectivity between the patches, the distances between these patches or other patches. They have to move from one patch to another, and in between there is the matrix. And such a matrix is everything but hostile for many species. And let's look what of these three parameters is the most important one. We conducted a study also in calcareous grasslands in central Germany, and we studied habitat specialists, leaf and plant hoppers, butterflies, and several Orthoptera species. And the parameter with the highest explanatory power, with the highest explained variance in all three groups was habitat quality, followed by the area of the habitats, and then by habitat connectivity. Habitat quality is clearly the basis for all our conservation measures, And connectivity, however, plays also quite a sufficient role, especially when we think about range shifts, when we think about dispersal. Species that want to disperse, they need a higher connectivity. On the other hand, especially in Orthoptera, there are a lot of Orthoptera species, especially flightless species, that are able to survive for longer time periods in habitat patches if the habitat quality is high, and we have consistently a high habitat quality. Connectivity is often not that important than connectivity. All these three key parameters have strongly declined in our landscapes. Habitat quality is decreasing, especially due to land use changes. We have abandonment, we have land use intensification, and there are also other stresses that affect habitat quality and cause a decline in habitat quality. The second one, habitat area, is also declining in most of our habitat patches compared to former times, and the same is true for connectivity. There are also clear losses in habitat connectivity, and with these conditions, the species have to cope with in our fragmented landscapes. And I'd like to focus a little bit more strongly on habitat quality. We have heard it's the most important parameter in determining species persistence in fragmented landscapes. And we conducted a study in calcareous grasslands in southern Germany and Bavaria, and we wanted to see what is the influence of habitat heterogeneity on species richness in these habitat patches. And we find a clear relationship between habitat heterogeneity and species richness in these grasslands. And this is a general pattern. Yeah, the higher the habitat heterogeneity in a habitat patch, the more species we often or usually have. And on the other hand, habitat heterogeneity is also a very suitable buffer against climatic extreme events. If we have different aspects, north-facing slopes, south-facing slopes, east-facing slopes, and so on, the species have the possibility to move in reaction to extreme events, and so habitat heterogeneity also buffers such extreme events. And habitat heterogeneity is one important part of habitat quality in such patches. Let's have a look how habitat heterogeneity can look like. This is habitat heterogeneity in a very small scale. We see some microtopography, we see shaded parts, we see sunny parts, and on a much larger scale, here we have cattle pastures in the mountain area in Central Europe. And here we have different aspects. We have open habitats, we have shaded parts, and such habitats are quite good in buffering extreme events, and they are usually characterized by high species richness in our Orthoptera species. And another thing that is quite important when we talk about habitat quality is land use intensity. It's a key driver of habitat quality in our patches. Here we conducted a study in southwestern Germany in the Black Forest region, and we studied different types of grasslands, and the pattern was quite clear. We studied dry grassland, massic grasslands, and wet grasslands, and species richness peaked in those grasslands with a relatively low land use intensity. And it was the same in all three grassland types. Low intensity land use is quite important for many species, and that is also one reason why we lose so many species, because we don't have these patches with low land use intensity. Now let's show how the distribution of species change in our fragmented landscapes due to global change. And one important study we conducted, that we focused on a habitat specialist and a habitat generalist. The habitat specialist is was Metrioptera brachyptera. The species occurred in the study area in calcareous grasslands and also in mire ecosystems. The habitat generalist, Röseliana röseli, is quite widespread and occurs in a lot of different types of verges or grasslands. And usually both species are short-winged. And both are short-winged, and in recent times we often observed long-winged individuals of the species, and we conducted studies in the lab and in the field. We wanted to know what are the drivers of microptery of long-winged individuals, and we observed that density stress in the first and second nymphal instar was responsible for the introduction of microptery. And now we wanted to connect this with global warming, with climate change. Why do we now more often observe such long-winged individuals? How is it linked with climate change? And due to climate change, our springs are often warmer. We have a lower nymph mortality, and this results in a higher population density. In our habitat specialists in Metrioptera brachyptera, we did not observe any density stress in the field because usually the densities of the species are much lower in the field than those of Röseliana röseli. And so we don't have any range expansions. In the habitat generalis, Röseliana röseli, naturally the densities are much higher, and due to climate change now we have density stress, and this results in a high proportion of macropters. And we also showed that these individuals are highly dispersive, and they are responsible for strong range expansion to the north, also to higher elevations. And for this species, for this habitat generalis, there are a lot of habitats available, and so expansion is possible. If we would have as much macropters in Metrioptera brachyptera, habitat expansion would hardly be possible due to the lack of suitable habitats for the species for this habitat specialist. Now let's have a look on the temperature requirements of the species and how this affects distribution changes in central Germany. We used data based on grid maps for Germany, and first we compared a historic period. That was the period before 1990, compared with 1990 and 1999. And species with a high temperature index, they suffered less from range losses in the historic period than species that are adapted to cooler climates. And when we go to the recent period, there the situation changed. The recent period was from 1990 to 1999 and 2000 to 2017. And we see that species that were adapted to cooler climates remained more or less stable concerning their distribution, but species depending on warmer climates, species with a high species temperature index, they increased, they dispersed, and they expanded their ranges. Now let's come to the farmland index. It expresses the dependence of Orthoptera species to high nature value farmland, and we have the same periods. During the historic period, most of the species declined, and especially species adapted to high nature value farmland strongly declined. And in the recent period, there was no clear relationship between the species farmland index and range shifts. there were not that severe losses in species adapted to high nature value farmland than in the previous time. And now let's have a look on dispersal ability and habitat specificity, and the range changes and shifts. And when we have a look at habitat generalists and specialists, we see that most of them suffered from habitat losses, from range losses. The only exception were habitat generalists with a high mobility. They expanded their range during the historic period, and all others declined. And there were differences between all groups that we compared. And in the recent period, we had also again clear changes. We had no longer differences between the generalists and the specialists in both mobility categories. And species with low mobility remained more or less stable during the recent period, but species with a high mobility in generalists and also in specialists, they increased their ranges, and clear differences between these two periods. In recent times, we observed strong losses in agricultural grasslands, and this is particularly due to the cultivation of bioenergy crops. We conducted a study in northwestern Germany, in the northwestern German lowland, and we observed strong losses due to the conversion of grasslands to arable fields for maize production, for bioenergy crop production. And we lost in our study area more than 1/4 of the grassland patches. On the other hand, the verges still were present. There we didn't observe such losses. And when we have a look on the grassland patches, we see that the loss occurred only in those grassland patches that were not protected. We had strong losses in grassland patches outside the nature reserves, but nearly no losses, or no losses in the nature reserves. So nature reserves were here highly effective in preserving our grasslands, but outside it was completely the opposite. Now let's see in these patches, in the remaining patches, what are the responses to climate change and to global change. And we see we had a clear decline in the farmland index from 1995 to 2012, and the same was true for the verges. And in contrast, the temperature index, we had a clear increase in both types. And the pattern we also saw before in the grid map-based study. The species that suffered most in our grasslands was Homocestus viridulus. It declined by more than 40% in the grassland patches and by more than 65% in the verges. And let's also have a look on the dispersal ability. Here a study from Western Germany, from the Eifel Mountains, and we studied again 3 grassland types: calcareous grasslands, mesic, and wet grasslands, and species with low mobility. There we didn't observe any changes between 1994 and 2015, but we observed clear changes in species with high mobility, a similar pattern to that we have found at our grid map-based data set. Now let's come back to our study from the lowlands. And here we have a clear difference to the study from the Eifel Mountains. Here we observed an increase in species with low mobility, and we explain this by the high connectivity of the patches. There are a lot of roadside verges in the study area we have tiny grasslands, and we have a high connectivity, and in landscapes with a high connectivity, Orthoptera species are partly able to follow climate warming, to track climate change. And yeah, high dispersal species also followed climate warming and expanded in our patches. And at the end, I'd like to focus on habitat restoration. We have seen we have dramatic losses of habitat patches, and habitat restoration is increasingly practiced in nowadays. And here I present a study on the restoration of heathlands in central Germany. And here, former spruce forests have been cut, the trees have been removed, and by hydroseeding, heathland seeds have been dispersed on the restoration sites, and we compared Orthoptera species richness in existing montane heathlands. Here we have the restoration sites, and here we have the controls. The control sites, there only the spruce forests have been removed. That was the only thing that had been done. And what we see is that Orthoptera are able to respond rapidly to these restoration measures if source populations are in the vicinity available. And what we also see is that we had a higher species richness, and all of these species are characteristic species of heathlands that we found there. We see a clear management deficit in these montane heathlands. Most are relatively tall stands. We have no bare ground. We have no early successional stages. They are missing in such montane heathlands. And here, the restoration sites are quite heterogeneous. We have open parts. We have parts with a higher cover of Calluna and other characteristic species. So habitat restoration can be a tool to foster Orthoptera species and other species. Let's come to the take-home messages. Some, but not all, like it hot. And habitat availability and climate will determine the distribution of Orthoptera today already and in future also. These are the two main factors that will determine the distribution of our Orthoptera species. Concerning the macroptery, the response to climate change of the species depends on the initial population density of the species and also again on habitat availability. And habitat generalists are more likely to track climate warming than habitat specialists, and this is also again due to the habitat availability for the species. The winners of global change are thermophilic species, habitat generalists, and mobile species. But we have also seen, if you have a high connectivity, also species with a low connectivity can move northwards or to high elevations. And the losers are hygrophilic species and habitat specialists. And we had severe heat waves and drought periods during the last years, and nowadays it's so that there are also other species than hygrophilics that suffer from such extreme events and that start to decline. And I'd like to show some slides that show such summer drought events. This is a mesic grassland in 2017 in August, and the same mesic grassland in 2018. And extreme events will increasingly occur in our landscapes, and they will be responsible for species losses in many patches. The key factors for conservation are based on our findings: habitat quality, and habitat quality clearly depends on disturbance and land use and the land use regime. Especially early and mid-successional stages are clearly missing in most of our landscapes. And low-intensity land use favors a lot of species. Habitat heterogeneity is quite important for many species and for high species richness, and it's also a very suitable buffer against climatic extreme events. Patch size is often related with habitat quality and habitat heterogeneity. The larger a patch, the higher the likelihood that we have a higher habitat quality somewhere in the patch and that we have a higher habitat heterogeneity. Connectivity depends on the population structure of the species. Some flighted species can persist for longer time periods in even isolated patches if they have a constant high habitat quality. But we have seen such extreme events, and therefore connectivity will be quite necessary in the long run to conserve our species so they are able to shift in response to such extreme events. And also for expansion of species, we need networks of habitats, and so connectivity is also important. And habitat restoration is one possibility to foster species, and it will become increasingly important due to the severe loss of habitats in our landscapes. Yeah, and I'd like to thank you. First of all, I'd like to thank the grasshoppers for their cooperative behavior, and I'd like to thank you for your attention. Thank you very much.
Thank you very much, Thomas. Any questions from the audience? Yes, please.
I have a question about the habitat restoration initiatives. How long did it take for the repopulation of grasshoppers to place after?
Yeah. We conducted the study 4 to 5 years after the restoration measures had been conducted. They rapidly respond, yeah.
Axel, sorry.
Thank you. As you know, we are doing similar research on the changes in Orthoptera diversity in Rhino-Palatinate, and we have similar results. And I just wondered, because of the term generalists, we use it still. We still use the term specialist for example, Statophrima grossum and Oedipoda caerulescens, who both have also massively expanded because they are, of course, very mobile. And we just wondered if they still can be termed specialists. Yeah, I think specialist is quite a subjective term which we use for species which are rare, more or less. If you go to southern Europe, for example, these will not be called specialists, of course, at least Oedipoda not. So I just wonder if we may shift more or less in the main part of the range where the species is adapted to more sites, and so that it's difficult to use a term in a long-term way.
I agree with you. We can work with such classifications. We have habitat specialists and generalists, but it's a simplification, it's clear. And another thing I also presented is the species farmland index. And there we combined the area of high nature value farmland in grid cells with the occurrence of the species across the German range. And so we have a clear gradient in this. And Oedipoda won't be a high nature, won't have a high species farmland index, for example, because you can find the species in cities, in rural areas, and so on. And there are different possibilities how to work with such classifications, and maybe such gradient analysis is the better way.
This is similar question, like Alex, generalist versus specialist. How did you measure the threshold or quantify the threshold between the generalist and specialist?
We didn't measure. Our classification was based on Reinhardt et al., a paper from 2005 published in ECOC III.
I have also the same topic, specialists and generalists. We made quite a similar study on 100 plots in whole of Austria and tried to differ habitats, specialists and generalists on the distribution about different habitat types in the whole Austrian database. And this is some, I think, a possibility to make it with some hard facts. So more than 3 habitat types for 75% of all data are generalists and something like that. But the interesting thing is we have the same proportion of specialists in the increasing part of the species as in the decreasing part, and a significantly lower amount of specialists in the stable ones. So we have very expensive specialists too, so in our case, it's not
no
so,
problem,
so
especially
clear.
if they are highly mobile, if they're capable of flight, and
There's one question by Klaus Rede online, and it's more like a technical question. People are asking questions in the room, if they can repeat their name so that people online can know who asked the question. That's the suggestion of Klaus Riede.
Okay.
Okay, Armin Landman here. Hi. You connected an alleged density stress with an alleged increase in macroptery. Wouldn't you think that in former times, when overall landscape had much more quality, agricultural landscape had more connectivity, we would have expected more density stress and thus more macroptery, especially, for instance, in Röseli?
No, I would have expected this, and there are no indications that it was like this, because in former times these macropters were quite rare, and especially in the last 10 or 15 years we had macropters quite often, and in some populations more than 45% of the population were macropters, and our results are relatively clear that we need really high densities and really low. These high densities are only possible if we have sufficient food and we have warm temperature, the combination of both. And in former times we had sufficient food and good conditions in many of the low-intensity grasslands, but there's no indication that the densities were as high as often nowadays.
Okay, thank you. There's time for one more question, if anybody has the last question.
I have a question from Howard Ree online. How 2018 drought affected future generation of Orthoptera from only 2018 generation? It may be that only the next generation from the 2018 population will be affected. Is that a clear question?
Such drought events will cause effects, clearly. And I would assume that we have losses in the next generation, clearly. But today we don't have sufficient data to say how long these effects will be visible and what will that mean for in the long run. And
Okay. Thank you very much, Thomas. It's not, when you have one hand, it's very irritating if you have one. Anyway, the next speaker is Roy Kleukers. I want to announce you properly. Wait. He currently is and has been for a long time, and hopefully he will stay for a long time, manager of ICE, Knowledge Center, Insects and Other Invertebrates. He started his career with an atlas of the Dutch Orthoptera published in 1996, and has kept his interest in Orthoptera ever since. So today, he'll present the history on Orthoptera study in the Netherlands.
Hoe werkt het, de klikker? Ja, dit heb ik niet nodig.
Here you have a pointer.
Ah, the pointer. I don't need the pointer. I
Okay.
just need this one. Yeah. Okay, thank you. Welcome. I'm going to talk about the history of Orthoptera studies in the Netherlands. We have quite a rich history, and we are very happy that we can host this Congress here, of course, in Leiden, in Naturalis. I will talk a bit about my organization, the early years of the Orthoptera studies, the Atlas projects which I coordinated, and some recent developments and future plans that we have. The goal of my organization as Knowledge Center Insects is to advance the knowledge on distribution, ecology and protection of invertebrates in the Netherlands. We are an NGO in Holland, and we have 62 study groups which study different insect and invertebrate groups. We have an office with 13 people housed here at Naturalis. And we make databases of all the different groups and publish the results. And last years we are more and more involved in protection of insects. The field work is done by volunteer recorders, and because we are covering all the invertebrates, you need a lot of different techniques to record all these species. And with that work we get an idea about the current distribution of the species. For the historic distribution, we are dependent on the publications and especially the collections. Like here in Naturalis, you have seen probably the silver tower next to the building without the windows. That's packed with collections, with especially insects and also Orthoptera, of course. This is from a recent publication where this all comes together. We collect all the data, recent and historic, put them in distribution maps and show in that way what is the status of the species in the Netherlands. This is a group of snail-killing flies. They're called Sciomyzidae. That's our latest publication. And we publish books and magazines about invertebrates in the Netherlands. Then about the grasshoppers. It starts with the first published record, as far as we know, by Goedaert in 1662, of a mole cricket, a nice picture with the nest, the eggs. Then the first critical list was published by C. Willemse, Cornelis Willemse, in 1970. He also was active until the Second World War. This brings us to the family Willemse, which is quite a remarkable story in the Netherlands. There are 3 generations of Willemses active in Orthoptera studies, covering more than a century. Cornelis was the first one, he's the grandfather. He was active in the Netherlands and Southeast Asia. Most of you probably know Ver Willemse, he's the son of Cornelis. He was active mostly in Greece. He's published nice books, they're also on the tables outside where you can take a look. Both Cornelis and Ver were very serious scientists and well-respected members of their local communities. Now we have to deal with this. You all know Luc. These activities led to 2 important publications in the Netherlands. The book of Duim and Kruseman in the 80s and Leo Beukeboom, who published The Key. And those were very important for the stimulants of the Orthoptera studies in the Netherlands. And this led to the first Atlas project in 1990, which I have coordinated for 5 years. And we had to do a lot of work. We had to digitize the collections, we had to educate the recorders, we had to make databases, and I had a full-time job on that, and we published the Atlas in 1997. It was a very nice project, but it was very time-consuming. And later, in 2008, we thought we'd do it again and see what the differences are. And that was much easier, because at that point there was waarneming.nl, the Dutch observation.org, and we could do it all online, which was very much easier than the first project. And we also had digitized the collection already, so that we didn't have to do that again. But all communication went through the forum, the databases were built up online, and the response to the recorders was also online, so that was much easier. And we published the Atlas in 2015, or 16, I think. So this is a bit idea about the time consumption, about the organization of these 2 projects. And you see that the last project was much easier. I could do it not as a full-time project, but just in the evenings I could coordinate the project. And also the yield was much better. We had 60,000 records in the first project and 320,000 in the second, so that was perfect. We saw, for example, on waarneming.nl, many people started there with birds, but it was very easy to also include the grasshoppers in their studies, especially in the summer months, when for birds it's a bit slower, and the grasshoppers and the birds make sounds, so that was an easy step to make, and that yielded a lot of records. Because we had these 2 projects from 1990 to about 2020, we were able to document the changes which were taking place in that period. Because in 1990, when I started, I thought, well, the fauna of the Netherlands is fixed. We just look at it one time and then we know it. But it was immediately clear in the first years that there were species expanding, species were declining. There was a lot going on, and we could really document that very well with the Atlas projects. See if this works. Especially after 2000, there was a huge expansion of Phaneroptera falcata in the Netherlands, and now it's almost present in the whole of the country. Even on the Wadden Islands, it's present. It's really spectacular example of expansion. It's also shown in other countries, of course, but we were really able to document it well with all these records we had. After the Atlas projects, we wanted to keep the interest alive in grasshoppers for the recorders, where many people were active with grasshoppers. And in an Atlas project, it's clear what you have to do and what the purpose is, but after that you have to keep reminding people to keep putting their records online. We've made some educational material for people who started out with grasshoppers. Nice photos of Paul van Hoof, where we could make a poster and a brochure, for example, which was really nice. We made a digital multi-entry key. We did that for a lot of groups in the Netherlands, which are quite well used, but also for grasshoppers. And the nice part of a digital multi-entry key is that you don't have to start with one character and then go like in a normal key and then have a fixed path which you have to follow. You just say, well, I saw that the wings were short, I start with that character, and then the amount of species which you are left with is smaller, and then you go to another character, and that's really a very nice way of keying out species. Image recognition was really important also for the grasshopper, for all insects. It was included in waarneming.nl. When you put your record online, you have a photo and you put it online in waarneming.nl, you immediately get a suggestion by the image recognition what species it could be. It's not always correct, but for grasshoppers it works quite well. And I think last week they added also the male, female, and nymph adult combinations that you get when you put up your image. So that was really a stimulus for many people that they got feedback on their identifications. These Atlas projects and the recent developments they kept the grasshopper community alive, and a lot of records are being sampled every year, about 50,000 records per year, so that's quite a lot. And that leads me to the last innovation is occupancy modeling. Since a few years we have this statistical technique which we can use to make trends for grasshoppers. We have a good network ecological monitoring where many people walk fixed routes in the Netherlands for birds and for Odonata and other groups, but not for grasshoppers. That's the normal way to produce trends, but this occupancy modeling gives you the way to produce trends based on random records like we sampled today. And now for 35 of the 53 Dutch species we can have these trends on a yearly basis, so that's quite nice. Of course, the more interesting species and the more rare species, that you cannot do that because you don't have enough records, but we are thinking about putting them in the network ecological monitoring, which is a very much smaller project, of course. So we can have maybe the occupancy modeling for the general species, and for the rare species we can do the normal monitoring. At first I was a bit skeptical. I thought, well, the statistics, can we really make reliable trends? But when we saw these first graphs, it was quite convincing, like the Vippiger diurnus, which is really in decline, that we know, and it shows in the graph. Chrysogram dispar is clearly expanding in the Netherlands and doing better. It's also quite convincing, I would say. We also found a few surprising results, like Tetrix undulata and Myrmedotetrix maculatus, which seem to be in a sort of a decline in the last year, so that are species which we have to look out for and see if they are not becoming red list species. Well, to end with the future, we're going to continue to support the recorders, of course, improve the image recognition, especially by putting more photos up, which gives a better reference database. Boudewijn will tell something tomorrow about the better availability of the songs through Xenocanto, and the protection of endangered species of Orthoptera in the Netherlands we will contribute to. So that's it. Thank you for your attention.
Terrible picture of myself. Any questions from the floor?
Hendrik de Vries. My question is, the multi-entry keys are deterministic, which means that when you make one error, you're wrong. While you have in image recognition, in Identify, you have a stochastic system where the system will say you have 100% chance for this species and 90%. Is there a possibility that the multi-entry keys will also be stochastic in the future?
Maybe in the future. I'm not so technical, I must say. I think it's possible to include that, but for the moment it's not implemented. So that's, in fact, it's the same as with a normal key on paper, you could say. Yeah.
There's one question from the online group. It's Klaus Riede. He says, very impressive. Are your methods and tools used in environmental impact assessments?
Not that I know of. Maybe I don't understand the question correctly, but... Ah, yes. Well, in Holland, the grasshoppers are not included in the nature policy. So there are many other groups, but not the grasshoppers. They're not protected in any way.
Okay, one last question.
I am Kiki Kati. It's a very nice presentation, thank you. I find very interesting this use of occupancy modeling for having the trends for the future, okay, for the grasshoppers. But my only consideration is that it doesn't presupposes that we are going to have the same stable effort of sampling per year, because it can be that it's false, I mean, the trends, because it's dependent on the sampling effort of volunteers.
Well, in fact, it's not, as far as I understand it, I'm not statistically much, I don't know much about statistics, but what I understood was that there is corrected within the methods for the sampling effort. So that's within the technique that you, that it doesn't, you don't have to have the same sampling effort for every year or every area. So that should not be a problem.
One last question, a very short question.
Maybe the sampling effort can be used as the variable for the trend of abundance.
I'm not sure. The trends are made by the statistical institutes here in Holland, and I'm not really involved in that. I just look at the outcome, I must say. Okay. But there are descriptions about the technique, so you can look it up in the material and methods of the papers.
I suggest any more questions, please save them for the coffee break. Thank you very much, Roy. Our next presenter is Sophie Ogan. She is a PhD student at the Department of Biogeography in Trier. And her presentation is about population status and trends of Orthoptera in Rhineland-Palatinate. Sophie, please.
So, where's the one with the pointer? Ah, okay. It's yours. Okay, welcome. Thank you very much for the introduction, and I am very happy to present to you today a new study that we did in Trier, a resurvey study on grasshoppers in southwest Germany. This is part of my PhD project, and the manuscript on this data are currently under review, so these are first insights for you. I would like, okay, this is not working. I would like to start my presentation with a little story that happened to me about 3 years ago at the beginning of my PhD fieldwork when I did my first surveys in fields. And I was walking around with a net looking mainly for wild bees, but also for spring grasshoppers. And it was around midday when an older man approached me and asked me whether I'm looking for butterflies. You might be very familiar with this situation. And I told him, no, I'm not looking for butterflies. I'm looking for grasshoppers and wild bees, and that we have this new project at Trier University where we try to understand population trends for these two species groups and what's driving them. And he was very interested because he was coming in this specific area here for around 30 years. And he told me that for recent years he had a very strong feeling that especially insects are declining in this area. And this is part of the reason why we wanted to do this project, because we wanted to transfer this feeling, what a lot of people have, into proven statements or proven data. And there's another reason, most of you might be very familiar with this graphic, the population trends for European grasshoppers based on the European red lists, and there's a very high number of deficient data, and we wanted to be part of solving or increasing or decreasing this deficient data. So what did we do? We combined 13 individual studies into one big historical data set. All these studies conducted their fieldwork between 1985 and 1991, and individually, each study over a period of a maximum of 3 years, within a year of 1 to 2 surveys per year. all had very comparable methods, so they used mainly visual and acoustical methods. And as you can see here in this graphic on the right side, the largest part of the studies are based on 2 individual studies, the one by Carsten Renker and Christoph Fröhlich. And both are also co-authors of the recent manuscript. I think this is important to know. And we added these smaller studies that you see up there to fill spatial gaps because we wanted to cover the whole area of Rhineland-Palatinate. And yeah, so we added smaller studies, but all of them are very comparable in terms of methods. And what did we do today? We resurveyed these 199 study sites. We started our fieldwork in August 2018 and ended up in October 2020. We used the same methods and tried to be as close as possible to the original studies. But we had a bit higher per year effort. So the original ones were with 1 to 2 surveys per year, so we conducted at least 2 surveys per year. The first one to cover mainly the tetrix species in the spring, and a second survey in July to August. In case we had a very early second survey, we conducted a third one. Additionally to the grasshopper species lists, we also sampled environmental data such as vegetation structure, vegetation height, but also land use type and land use intensity. And all our data are available at observation.org. And now already coming to the first results. We had a higher species per site, so an increase in alpha diversity. Of a total of 49 Orthoptera species that we recorded, 13 are significantly increasing in site occupancy. That means that they are now frequent or more present on sites than they were in the past, and we found 5 species to decrease in site occupancy. The biggest winner here on the right side, Cotopus dorsatus, the species was pretty rare in the past with around 5% presence on our study sites. And now it massively increased. So we have them now on around 65% of our study sites. So yeah, this species is like the biggest winner. And a bit contradicting, we found also losers, and the saddest story is the one for Bicolorana bicolor. The species was formerly present on around 45% of our study sites, and now it decreased to around 20%, and this might not be the end of the story, so we should really look out for this species. And I mentioned earlier on the slide that we had an increase, a significant increase in alpha diversity, and we also looked at beta diversity. And we found a contradicting pattern here. So we see in these lighter gray colors the beta diversity for the past, and in the darker colors the beta diversity for today. And you can already see that there is kind of a shift in this graphic. So 0 is here indicating complete dissimilarity, and 1 is indicating complete similarity. And we found that there is a significant shift, so species, communities in our study area getting more similar, which underlines that we should not only look at alpha diversity, but also taking other indices into account. A bit additionally to the discussion that we had earlier this day, we also looked at ecological traits, and we also focused here mainly on the specialists in the darker colors and the generalists in the lighter gray colors, and the mobility of a species between low and high mobility. And we found that specialists with low mobility, on the far left side, have all negative trends. How you can read this graphic? We calculated the relative change in occupancy. We did this by setting the presence of a species in the past to this 100% reference line here. So this one here is the reference, and then we calculated the relative change to this reference. And yeah, as I mentioned, all specialists with low mobility have negative trends, so they are declining. We found significant differences between generalists with high mobility and specialists with low mobility, and a trend between generalists with high mobility and generalists with low mobility. What we think could indicate that the mobility of a species, as Thomas Fartmann mentioned earlier today, is more important for a species to colonize new habitats and therefore also for their population trends. But what we think is also very interesting about this graphic, and Axel mentioned that earlier, is that specialists with high mobility have very positive trends, and two very good examples for this are Stetophyma grossum and Oedipoda carollescens. Both species are classified as specialists, but they massively expanded during the last years. And we know from species in general that they can be quite specialized at the edge of their distributional ranges. But when you can think of that climate change might have within these ranges and the species expanded, they might not be that specialized in our study area anymore. And speaking of climate change, we also looked at the altitude as a proxy for climate change. We did this by calculating the mean altitude that a species occupies in the past and today, and then we looked for differences between these two times. We found that all Orthoptera species are moving upwards, so they had higher mean altitudes, and we found 7 species to significantly move upwards the hill. Of these 7 species, 3 are also expanding in occupancy, and 1 is declining in occupancy. And there are 2 very interesting patterns behind this, which I would like to illustrate on this following graphic. So you see in this A plot, Omotestus viridulus, species massively declined in occupancy, but expanded in range, in altitude. And what you see is that the species loses the size with lower altitude. So the species is moving up the hill, but losing the lower sides. And we know that Omotestus viridulus needs some degrees of moisture for the eggs. So these lower sides might not be this suitable anymore as they might be in the past. So the species is driven up the hill and losing these lower sides. And we mentioned Oedipoda carollescens before, a species expanding in all areas. And this species now occupies higher altitudes, but it's not losing lower sides. So it's just expanding and seems to be, at least for now, a climate change winner. Yeah, so very interesting, 2 different patterns of altitude shift. And in the last part of my talk, I would like to give you a very brief introduction into the environmental parameters that we worked with. These are just a few slides, and I'm very happy to talk during coffee breaks more about this. So in the first slide, I would like to present you the changes that environmental parameters did between the historical surveys and today's surveys. And what we found is that the forest in our study area increased significantly, and also the tree cover, which is very likely to be connected to each other. We also found a higher maximum vegetation and an increase in medium land use type, and also of mesic grasslands. And in a bit contradicting pattern, we found significantly reduced dry and semi-dry grasslands and nutrient-poor conditions. To put this in a bigger picture, we had higher, more denser vegetation now compared to the past, and we're losing these sites with drier and more open, patchy bare grounds. But we had the highest number of species in the dry and semi-dry grasslands, which is also known for Orthoptera species. We also looked at habitat management. We mainly focused here on grazing and mowing, and we found grazing to be highly favoring a lot of species. And very positive to mention is that Bicolorana bicolor, remember, the species with the strongest decreases, is very highly favored by grazing. So sites with grazing, we don't lose the species as often as we lose the species on a site where we have no grazing. We also looked at mowing, and we found that a lot of species are very positively affected by mowing, but all these species here are also significantly increasing in site occupancy, which means that the degree of mowing might favor the expansion of the species. But we have to say that land use intensity is not very high in our study region, and especially on our study sites. And when you can think of like more intensive regions where we have like more frequent cutting, then the Orthoptera species might not be as favored anymore. And in the last part, I would like to present you the results of the protected areas, because we had an almost 50-50 distribution between protected and unprotected areas, unprotected status of our study sites. And we found 4 species to be highly favored by the role of protected areas. And the nicest thing about this for us was to see that Homocestus viridulus and Bicolorana bicolor both are very strongly decreasing in our study area, are favored by protected areas, meaning that they're decreasing not that often as they do with outside of protected areas. So protected areas might buffer their negative trends, and yeah, so this is good news for protected areas. To sum this up, what are the take-home messages? We found winners and losers in our study area, but we found more species expanding than they are declining. We found higher alpha diversity, but a decrease in beta diversity, which should be taken account. We found that the mobility of a species matters and can even cope to a certain degree for the ecological specialization. We found altitude expansions, mostly caused or driven by climate change. We found that proper habitat management can favor a lot of species, especially grazing for Bicolorana bicolor, and that protected areas can buffer especially the negative trends for decreasing or declining species. Thank you very much.
Thank you very much, Sophie. Questions?
My name is Thomas Tunagratki. It's not a question, it's more a support, because we made a very similar study the last 2 years. We resampled over 100 plots from the 90s, and we have surprisingly similar results. So the increase of species per plot is significant. Also some homogenization. It's the plots with few species increased in species richness, and very species rich plots decreased. So it's more, all plots are now quite similar. We have also other species, for instance, the most increasing were Mecostetus and Ruspolia. The most decreasing species were Homocestus hermodalis and Rufibes, for instance. So but also winners and losers. And also the winning specialists are the mobile, so it's very much supporting. But we had one thing, we could also compare densities, and we had significant decrease of density of habitat of grasshoppers per plot. So do you have any incidences to shifts in the densities of the species, not only of the distribution?
Yeah, we had the one by Christoph Fröhlich had also like rough estimations of densities, and we looked into this, and it did not show any difference to the presence-absence data that I presented here today. So, but the larger part of the data set didn't include any density in the past, sadly. So yeah, but we recorded today also this density estimations, so we hope that in the future this could be taken into account. Yeah, but thank you very much. It's very interesting that the studies are producing same results.
Axel Hochkirch, just a very short thing to add. We had one data set by Matthias Weitzel who used something like a box quadrat approach, and we tried to use the same method as he did. And actually the densities were so low on the sites that we couldn't compare the data. So we actually didn't proceed this way.
Yeah.
So I think you're right that the densities have decreased significantly.
Last question.
Thanks. Oliver Havlicek. So you mentioned that alpha diversity increased mostly and beta diversity declined at the same time. So I'm just trying to get this right. I'm not an ecologist, but and then you said that many highly mobile species were able to colonize new sites. So if we have, let's say, the same 2 or 3 highly mobile species colonizing many new sites, then the alpha diversity will increase at these sites, but beta diversity will decrease. Is that right? Because it evens.
It's all the, if these species are colonizing and we have all the same species on the plots, and we might lose like 1 or 2 species which are specialized, just an example, and now we have like, even if we have 5 species and we had formerly 3, but all of these 5 species are the same, then we have reduced beta diversity, although we have higher number of species per site, yes.
Cool. So do you know which species these are, or have you tried to find out?
For what then?
Well, so did you try to find which species caused this increase in alpha or decrease in beta at the same time? Is there any way of finding out which species might be these highly mobile?
I would just think that it's mostly the one expanded massively, which I think, like Cottypus dorsatus, Roseliana roseli, Oedipoda caerulescens. Yeah, these species.
That makes sense. Thanks.
Thank you.
Okay, thank you very much, Sophie. The next one is Jeroen van Leeuwen. He's a project leader at Natuurbalans and Limes Divergens. That's an ecological consultancy firm in the Netherlands. As an ecological researcher, he is interested in the role of soil organisms in ecosystems functioning, both as indicators for and driver of the provision of ecosystem services. And today, he's going to tell us something a bit different, but anyway, he is going to tell us more about a slightly different topic, the Western saddle bush cricket, Evippica diurnis in the Netherlands. Jeroen.
Good morning. Indeed, my background is more soil-related than the content of this topic, but I'm going to talk about the Western saddle bush cricket, which is one of the rare grasshopper species in the Netherlands, and we have run several projects on this species. I'll show you a bit of the occurrence in the Netherlands, the decline over the last decades, and what we try to do to bring that back to its former spread. What you see here is the map of the distribution of the species in the Netherlands. In gray patches are the ones before 2006. You see there's a bit wider spread than the red dots, which is the more recent distribution. And even the red dots have decreased since then. I'll tell you a bit later about. The species is limited to the dry heathlands in the Netherlands. For those who do not know the Netherlands, this is the Veluwe area, which is a high, dry, sandy area with a lot of heathlands. And there are several patches here in the higher areas of South Limburg. And this rare dot in the north, that's an extreme outlier of which I do not know the origin. All of these points are still within nature reserves, so the species does not occur outside nature reserves. If you plot it on the elevation map of the Netherlands, you can clearly see what I just highlighted. The species occur in the higher areas. So here's the Veluwe. There were former dots here on the Utrechtse Heuvelrug, which is also a glacial moraine. Nijmegen here, the area is also a glacial moraine, and there's a few dots here in the hills in the southern part of Limburg. In this spot, the species is gone since almost 2 decades at least, and now the main distribution is here and a bit more here. When we started this research, there were about 16 populations in the Netherlands, and the populations on the map here are focused on the Veluwe. This is the central area of the Netherlands, and even on the large Veluwe area, which has a lot of heathlands, you can see that even decades ago, already the distribution was very limited to small patches of heathland. So here it was all empty, and then here's a few patches, and here in the northern parts. Many of these populations have either gone or decreased in size, which is also a bit shown here. This is the more focused in on the Veluwe zone, which is the south of the Veluwe. And you see here, this is all heathland. You see, even in a single area, it's focused on very specific regions within the heathlands. It's either due to microclimate or habitat structure. I'll come back to that later. But even on a single heathland, it's rather focused in its distribution. And even in these well-populated areas, it has been decreasing in the last decades. So what does the western saddle bush cricket needs as a habitat? In the Netherlands, it lives in heathlands. So vegetation dominated by heather, Calluna vulgaris. And the major part of this heathland is older structured heathland. So larger clumps of old heather bushes and open sandy spots in between. And this enables the grasshopper to find the right microclimate, warm up rapidly, move around a bit more. So if the vegetation becomes too dense, the movability is decreasing. So it doesn't prefer that as well. And it needs the open spots between the heathland clumps or the heather clumps to deposit the eggs, mostly open sandy spots. And apart from the Calluna bushes, it also needs a couple of herb species to feed on. The majority of the diet is Calluna vulgaris. We've shown, I think Bouwman will come back to that later on this Congress. But it also eats on several other herb species, and as an adult it's omnivorous, so it also needs insects to feed on. And that's what it needs in the variety in the heathland vegetation. And the males find focalization spots in the higher areas of the vegetation structure, so the outlying bushes, the young trees, and the very old, large heather clumps. Oh, wrong button. So this is what a structured heathland in the Netherlands looks like. It's dominated by Calluna vulgaris, but you can clearly see the older clumps and the more open areas in between. And in the background you see a bit of younger trees and pine trees. And that's what they use as a vocalization point. You see here a female that has just mated with the spermal package at the abdomen. And especially the nymphs you also find in the herbaceous vegetations in between the Calluna clumps. This is on wood sage. When you look at the populations in the Netherlands, I mentioned before there were 16 populations in the past, or at least the last couple of decades, and now only 6 remain. The red crosses are populations that are gone, and also Nationalpark de Hoge Veluwe and Planke Wambuis, they were still noted as strongly declining here, but in the meantime they have been extinct as well. And you see a clear decrease in populations. This 2040 dot is of course a prediction, but it will more likely be leveling off, but still the trend is critical. The main reason for decline for the species in the Netherlands is the loss of the habitat quality, as far as we know. And there's a number of reasons, but the main reason here is the nitrogen deposition. In the Netherlands, in the years of 60, 70, there was a sulfur deposition and a strong acidification as a result of that. That's been banned, luckily, but now nitrogen deposition has taken over and there's still a strong acidification process going on. And especially the dry heathlands, which are basically scarce vegetation, they really have a strong impact on that. It's a combination of acidification and eutrophication. Acidification is leaching out all the base cations that are buffering the system, and eutrophication becomes a more dense vegetation. The increase in grasses and forests, that's the result of eutrophication. So if there's more nutrients in the soil, grasses will come up, and the Calluna is dominating the vegetation when there's less nutrients. So you get a more grassy vegetation, which is not good for the saddle bush cricket because of its thermoregulation demands and the limited protection possibilities in that area. Eutrophication also leads to a mass domination on the ground level. So if you look at a heathland nowadays, there's a large chance that the open soil is mostly dominated by mossy and lichen vegetation types. And that impacts the microclimate in the soil. It needs the open sandy areas for the eggs to really warm up and incubate it. Even in the open areas, it takes 2 winters to come out. And when there's a moss layer on top, the soil temperatures go down, the humidity goes up, and incubation is mostly blocked. And there's also a decrease in herbaceous species in the heathlands in the Netherlands. It's now more and more dominated by Calluna vulgaris by itself, and all the other herb species are gone because of the acidification mostly. All the base cations in the soil are leached out, and for plants there's not much to find anymore except for nitrogen in most cases. And that gives a loss of a food source for the nymphs and a loss of insect community as a whole on which the adult also feed on. Here's a picture of what I mentioned as a moss layer on top of the soil. What you see here is a lot of moss in between the heather clumps, and what it used to be was open sand between the heather clumps where the eggs are deposited. Now the whole deposition range is basically gone. Well, since 2004, Natuurbalance is really focusing on this species to protect and get back what is lost. And the first was a species protection plan. Later on in 2018, the protection management actions were more detailed and more applied. But it was still mainly focused on above ground vegetation structure and what needs to be done to get that back. And earlier this year, we presented a report to the province of Gelderland in this case, which has the main population area. And in this case, we also look at soil chemistry as the main underlying reason for the vegetation structure decline. And of that last report, I'll tell a bit more about what has been done and what will we do in the future. The aims of this report were the recovery and stabilization of populations of the western saddle bush crickets. And the major difference between this report and the previous report is that we focus on the habitat recovery and thereby restore the entire ecosystem, and instead of just focus on the species that we want to protect. And with that, we look at the system as a whole and protect species richness and nutrient balance and stoichiometry in the vegetation in order to protect or improve the dry heather. And there's a couple of phases in this recovery process. We start off with a selection of the suitable habitat areas. In this case, the places where the species is still occurring or where it's been gone recently. In those areas, we analyzed, together with the research institute Beware, the soil chemistry and plant chemistry. And based on that, and also on the above ground vegetation structure, we advised the application of specific management practices. It can either be burning the vegetation or raking the moss in between the heather clumps, or choppering, which is basically deep mowing, removing the litter layer. or grazing in areas where there's too much of a grass domination. After these practices have taken place, we want to restore the soil nutrient status based on the soil samples that were taken in the soil chemistry that was analyzed. An advice came out to apply, for example, a rock powder or Dolocal, which is a calcium supplement, or even fertilization in the form of phosphorus additions where needed based on the measuring of the measured soil nutrient status. And when basically the abiotic and surrounding conditions are recovered, we can bring back the herbs that were formerly growing in the vegetation there by seeding or transplantation of hay. And when the habitat is restored, we bring back the grasshopper itself. The aim is, of course, the structured header as the habitat of the western saddle bush cricket. At first, the soil nutrient status, which is the most dominant factor changing the habitat quality in this point. For this, soil samples were taken place at all the areas where the grasshopper is still living or was living, and the data from those samples was compared to a GRIP database, which is a database set up by the Research Institute Beware, in which they also have like reference sites in which they measured all the soil elements, the nutrient elements, and we can compare that to well-structured heathlands and well-structured Nardus grasslands, which have, harbor the same grass species and same herbaceous species. This is an example as for how that is working. So the gray area is the reference sites in which the data, in this case the base saturation, should be lying. There's only one site in the Netherlands which is still okay, which is the Oldenburgse Heide, the largest saddle bush cricket population. And all the other ones are have strongly decreased in base saturation. To bring that back, you can apply several soil amendments. It can either be Dolocal, as I mentioned, the calcium amendment, rock powder, which several types are used throughout in the Netherlands right now. They differ in, for example, magnesium content or potassium, and fertilization where needed, where, for example, the nitrogen-phosphorus ratio is not good enough. This is how we presented it to the terrain owners, which can either be municipalities or nature conservation organizations. And in black you see the soil samples, and for example in red the removal of the moss. In this case you see it's a small scale method that allow for habitat variation and different types of management to see what's actually working best. In some cases it will be the calcium by itself, in some cases it will be rock powder. We don't know yet, most of these methods are still experimental, but we applied it to all the populated areas. Then to bring back the herbs, you can either do it by seeding or distributing of hay. And well, the first part of the slide I already mentioned before. The effects of the management actions should be monitored, both soil chemistry and plant chemistry and vegetation structure, and only if the whole package is complete again, we can move on to bring back the bush cricket itself. And for that we started a breeding program. So since 2019 we're actually breeding this grasshopper in Nijmegen, and we collected nymphs from all the remaining populations in the Netherlands. And we did a genetic analysis to see which is the most genetically diverse populations that can be best used as a source population. And in that case, Oldenburg and Mulderskop near Nijmegen were the most diverse populations that should be used for reintroductions and also for boosting the current populations where needed. For example, the Zanding, where the population has been going down for years. This is what it looks like in the lab. So the cages in which the grasshoppers are breeding. We incubated the eggs over 2 years in an incubator, and then last year, actually, the first boost of grasshoppers came out. Hopefully this year a lot more. And yeah, basically it's now time for action in the field. So we have the captive program. We've basically assured the money from the provinces for applying these actions, and we're talking to landowners now to apply the management practices that we've advised. And hopefully in the fall of this year, the first actions are taking place, and next year we can bring back the first grasshoppers in areas that are already okay. And with this, I want to thank you for your attention, and maybe there are questions.
Thank you very much. As I have already proven to be a very bad chair because we are running out of time. I can allow one question, one short question. Anybody want a short question for Jeroen? No one? Not on the...
There's one question by Lonneke Klein-Aarts, and she wants to see the report, and she cannot find it online. Is it available somewhere?
Not yet.
Ah, not yet.
It's available for the province, but not yet for the landowners. We still need to talk to them.
Okay, that will
and after
follow
that
later.
It will be published.
Okay, thank you.
Okay, thank you very much, Jeroen. My apologies for shortening your coffee break, but I'm very strict. At 11, we continue again. Okay. It's at 11 o'clock, so we continue with the program. One question for the next speakers. We experienced some, because normally when people stand here, they tend to turn away to look at the screen, but there is also a screen in front of you, so you don't need to turn your head, you can look there. And also we would like to ask all the next speakers to collect a headset from this gentleman there before your presentation. And then after your presentation, please return it to him so that the next person can use it. Anything else I forgot? No, I think that's it. Okay. We continue with Simon Ziwecki. I'm sorry, I'm very terrible. One can think of many reasons why we should make distribution atlases. No doubt they are an important means to share knowledge and popularize the group itself. Simon will present an update on the Orthoptera distribution atlas for Poland. Please.
This one? Okay. Ah, perfect. Okay. So, hello everybody. I'm going to present the Atlas project that started in Poland 2018 with all my colleagues here, and I'm going to present the achievements and perspectives of this Atlas and how the work is going, basically. Yes, so first of all, it's Polish, like most of the atlases, national Orthoptera atlas. So Poland is kind of on the border between Central and Eastern Europe, one can say. And first I would like to give you just a short overview of what kind of Orthopteran habitats you can find in Poland, what kind of habitats we are working in, looking for Orthopterans, and the kind of collecting data from. So, like most of Poland is plains, consists of plains, which are covered like the most. I will show the most interesting, more diverse Orthopteran habitats. So, the most diverse Orthopteran habitat for Orthopterans in the plains are sandy, dry grasslands. Like you can see here, beautiful places with juniper bushes, with young pines or pine forest edges, and maybe the most characteristic species is Myrmolteticus maculatus there. If you go a little bit farther south, then you have beautiful xerothermic dry grasslands, mostly on limestone, with this colorful ones. With they are very special. They are part of this plant association Inoletum enzifolia, and I was trying to think of a characteristic species. I think I chose Ephipiger because it's widespread there, and it occurs especially in these uplands in xerothermic grasslands. But I also wanted to show one special species, Poecilimon ukrainicus, that has a kind of northwestern edge of its range in Poland, but only occurs in the eastern parts of the Polish uplands. If we go further, another interesting Orthoptera region are the Carpathians. And in the Carpathians, I think we also have higher mountains, more than 2,000 meters above sea level, but maybe the most interesting habitat there are extensively used hay meadows, like you can see here, also very beautiful habitat, and maybe the most characteristic species are Isophias there with 6 species in Poland. So, I hope you have some picture of what Orthopterans in Poland look like, but now I would like to go to the Atlas works. So it's basically the first project of this kind in Poland, because Orthopterans have been a little bit left behind compared to other groups of insects in Poland, like Odonata or Lepidopterans or Coleopterans that receive much more interest than non-Orthopterans. So we just started, one can say, but it's going well. We started with basically looking at literature from 19th century. We have from 19th century up until today. We have it quite well included in the atlas. By now, Then at the start, we started with people sending us observations in a kind of standardized form. But then fast a Facebook group was opened, which was a great success, and I think it contributed greatly to the spread of interest in orthopterans in Poland. And many entomologists that didn't know much about orthopterans started to also look for them, study them, and we got more and more observations, but also I think other people that were interested in other animal groups also started. Then in 2020, we started using ornitho.pl, so that's kind of also a database where people can contribute, where people can send their observations. And we also take observations from other open source databases like iNaturalis, for example. And yes, and of course, we ourselves also try to collect as much data as possible. But for now, these records that we have are mostly randomly collected. We did not do any organized or surveys yet that could be, for example, could be repeated. So this is something hopefully more to come, and museum collection is also on our aim now. The results we have at the moment is 308 papers in our database, 804 observers that contributed, over 52,000 records. And yes, now I would like to kind of show you just an overview of what we have, on what we stand at this moment. So the number here, you see the number of records in each square. The squares are based on a UTM grid, 10 on 10 kilometers, so quite big squares, but Poland also quite a big country, so it's not easy to cover. And here you see the number of records in each square, which may be a good proxy maybe of how well the area is covered, but I think if you look at number of species recorded in each square, you see a very similar pattern, basically the same. So maybe we can look at a number of species recorded. You see that vast areas are pretty insufficiently covered, and I was trying to kind of think like how can we say how well the area is covered. So at least 11 to 20 species should be present, like the very common species that kind of change between the regions, but should be present rather in every square. And if we calculate this, we can see that I mean there is lots to do, because only 17% of squares have 16 or more species. And I mean, this is, if you go to the north, this square that has 16 species may be well covered, or they are decently covered all over. The square with 16 species is decently covered maybe all over Poland, but if you go to the Carpathians or to the upland region, then for sure 16 species, that's not a well covered square. It can be square with 50 species. And another view on it, this is kind of a very high uncertainty level in statistics, because it's just kind of the number of places that have been visited in each square, but I thought nevertheless interesting. So in the squares that have been visited, on average 4 places have been visited in each square. So it's also still not a long way to go. Basically, yeah, but this is yes, but how can we proceed? What can we see at this moment? We can see that like before the 2000s, Orthopterans have been extremely understudied in Poland. But what we can see, maybe an increase around 2009, which may be connected to the translation of the famous Bellman Heiko book, Heuschreckenführer, into Polish. So there is some increase, which may be somehow connected to this, and then, which I find super beautiful, we see this a high increase in Orthopteran observations from the beginning of when the Atlas project started. So clearly, the Atlas contributes to the interest in Orthopterans in Poland, and yes, so we hope to continue on this level and make even more and more people interested with it, which I think the social media greatly contributes to. Yes, I mean this is what we did. This is our the results at this very moment. Some preliminary results one can say, but as it was shown before, of course, such atlases, such projects have great possibilities for looking at species changes, especially now in the time of the climate change, land use change, as we saw before, quite extreme, strong changes occurring over a short time periods. And for example, this example has been shown before from Netherlands, and here you can see maybe the resolution is a bit lower in terms of years, but you can see the extreme expansion of Phaneroptera falcata in Poland until 1990, where it only occurred in the warm upland regions, basically in the xerothermic grasslands only, and it was very rare. And then up until now, from 1991, you can find it in the Baltic Sea and quite common there. So I think that's quite impressive. And so these are important possibilities of such atlas projects. Of course, conservation is also very important. We cannot protect the species or habitats when we don't know what is happening. So we hope this project can contribute to this, and hopefully in the future even more. So future plans that we have for now, it have been only one can say records from the papers and the records randomly collected from random observations that people, entomologists, did. So no organized surveys. So we would like to start with some organized surveys, especially in the understudied regions, to get a better picture of Orthoptera distribution in Poland. Then museums collections are also on our very first list to do, because there is the next big amount of historical data that is only present in museum collections in Poland that nobody really has looked at for a long time. So this is one, and of course the kind of end idea of this project is to publish a book, an atlas of Polish orthopterans, and with information, and that should also then foster interest in orthopteras in Poland. Yes, and last but not least, also create a red list for Polish orthoptera with some conservation ideas, and maybe implement some monitoring, some stable surveys in the future to look at the endangered species, like for example the declining Psophus stridulus. Yes, you can find the website of our project in the abstract, an abstract book, and you're very welcome to visit the website. You can find all the distribution maps there and some additional information. So if you're interested, also the contact to our team. And yes, that was it. I would like to thank big thanks to the whole Atlas project team. That of course this work wouldn't have been possible with. I would like to thank all the contributors, many people, some contributing with only few records, but that's also super valuable. And last but not least, I thank you for your attention.
Thank you very much, Simon. One question only. No one? Online? Is there anything on the... I'm very sorry, no questions. Thanks.
No, perhaps I missed it at the beginning. How many species do you have in
87 species, 88 species, because one new species was discovered 3 years ago, so 88 at this moment.
Thank you very much, Simon.
Should I give it to him? Ah, yes, okay.
Our next presentation is from Giacomo Ortis, PhD student at the University of Padua. He has worked on honeybees and grasshoppers, and he's now going to shed some more light on how increasing temperatures affect the life cycle of Barbitistes visitinus. Giacomo.
Okay. Okay, thank you. So, good morning. And my presentation regards how increasing temperatures can affect the life cycle of the outbreak bush cricket Barbitistes vicetinus. Okay, Barbitistes vicetinus is an Ensiphera species described very early in 1993 as an endemic of the northeast of Italy. In particular, it can be found in the Lessinia Mountains, in the Berciunganian Hills, and it was found in a valley of Trentino region. It was considered as a rare species confined to forest areas, and because of its small distributional range, is it also considered as an early threatened in the IUCN Red List. So this species, it was considered rare until 2008, when the first outbreaks appears in the southern distribution range, that are the Euganian Hills, first located in some areas, but in the subsequent year affecting all the hillside area. So this species can shift from a solitary form with a typical green coloration to an outbreaking form with a typical aposematic coloration, and this is typical also for the genus Barbitistes. So during outbreaks, this species can cause severe damages to forest trees, although also able to affect cultivated crops such as vineyards, olive grove, and cherry orchards. I want to have now a focus on the life cycle of this species because it's very different compared to the life cycle of other species. In fact, the nymphs appear at the end of March, and the adults start to lay eggs in the ground for the entire June. So an egg can hatch in the year following the viviposition, or can remain in the soil for several years after resuming development and hatch. So to date, we demonstrate that an egg of Barbitistes vicetinus can stay in the ground for at least 3 years after hatching. So we choose to investigate how temperature can affect the diapause and the survival of this pest. So as I said, an egg can stay in the ground for several years in a state that is called initial diapause, where there's arrested development in the early embryonic stage. And instead, when the embryo starts to develop, it can reach the final diapause stage, where the embryogenesis is complete and the embryo occupies the whole egg space. So an egg will hatch in the following spring after overwintering in the final diapause stage. So our aims was to test the effect of summer temperatures on egg development, because egg development occurs often during the warm period, so during summer, and was to test also the effect of winter temperatures on survivals, that means on the hatchings in the spring. So to respond to these questions, we carry out 2 different experiments, one during the summer and one during the winter. Here in this table, I resume the methodology for the summer experiment. So we replicated this experiment during 2 different summers. And this experiment was carried out both in the field and in the lab. So for the field, we choose 21 sites along an elevation gradient to detect sites with different temperature. And in each site, we put 200 eggs of Barbitistus vicetinus coupled with a data logger inside the soil to detect temperature. And for comparative purposes, we put also the eggs in climatic chambers at constant temperature along a gradient from 19 degrees to 24 degrees. So at the end of the summer period, wow. At the end of the summer, we retrieve all the eggs from the lab. We counted the eggs that were unviable, the eggs that were in initial diapause, so that didn't develop, and the eggs that were that developed and reached the final diapause stage. And as you can see, an egg in the final diapause can be recognized for the typical green coloration, and the black dot represented the height of the embryo. So for the winter experiment, we carried out more or less the same experiment. So again, we choose sites along an elevation gradient, and now we put in each site 150 eggs in the final diapause stage. So now we put only the eggs that are able to hatch in the following spring. And again, for comparative purposes, we put the eggs at constant temperatures in climatic chambers. So at the end of the winter period, we retrieve all the eggs from the field and from the lab, and we counted the hatchings for each site. So here are the results for the summer experiment. So we replicated this experiment during 2 different years, and we obtained the same results. So we put together all this data in one single graph. So on the x-axis, we have the mean temperature of the summer period that went from 20 June to 31 of August, so the entire summer. And on the y-axis, we have the proportion of eggs that developed during the summer, so they reached at the final diapause stage. And as you can see, there is a strong effect of summer temperatures. In fact, when mean temperatures are above 21 degrees, nearly 90% of the eggs can develop and reach the final diapause. Instead, when mean temperature is under 19 degrees, only 20% of the eggs are able to develop. And we found that the incubation of the eggs at constant temperatures allowed lower eggs to develop compared to the field condition. And this is maybe due to the effect of fluctuating temperatures in natural condition. I also want to have a focus on this part because in only 1 degree of interval, so from 19.5 degrees to 20.5 degrees, there's a sharp increase in the number of eggs that can develop, so from 20% to 80% in only 1 degree of interval. So if we found an effect of the summer temperatures on development, we didn't detect any effect of winter temperatures on survival. So overwinter egg survival was high and constant among all the sites and among all the temperatures tested. And again, the incubation of the eggs at constant temperatures allowed lower eggs to hatch in the spring compared to the field conditions. So to resume, we found this different response between field and constant temperatures. We didn't detect any effect of winter temperatures on survival of this species, but we detect the strong effect of summer temperatures on diapause. So we can say that increasing temperatures can affect the life cycle of this species, shifting from a multi-year life cycle to an annual life cycle. It means that now, with increasing temperatures, a huge amount of eggs can hatch in only 1 summer after their reposition, instead of remaining in the soil for several years. So we can say that increasing temperatures due to climate change that we faced in the last decades could have contributed to the outbreaks of the species. And I want also to say that because for its small distributional range of the species, also its conservation status should be considered because it can suddenly pass to from an outbreak status to a threatened status. I want to conclude showing you that the effect of summer temperatures affect also other Ensiphera species. In fact, I reared different Ensiphera species, and here are Orthoptera schmidti and Orthoptera phallax. And basically, I collect the eggs of these Ensiphera. I put these eggs along an elevation gradient with the data loggers, and I obtained the same results as for Barbitistes. So a lot of species can shift the life cycle with the increase in temperatures. And finally, I want to say that this methodology could be useful to study the biology and ecology of Ensiphera species and also to predict their outbreak risk. Thank you for your attention.
Thank you very much, Jacob. Questions? Nobody has a question. Is there online in the chat something? Sorry.
Okay. Hi, I'm Costadina. I want to ask something, I don't know if it's stupid. You said that, from what I understand, that it affects it positively. We have outbreaks. So if the species has outbreaks and it decreases in population, how could we have a status of threatened in the future? That's my question. How can it be threatened in the future if we have outbreaks? Yes.
Okay. Okay, I forget. Well, so this species was described very early, so before was very rare. No one can see it. And now, still now, so it outbreaks from 2008 till now. So now it is in an outbreak condition. But we can say that for its small distributional range, maybe for environmental factors such as, I don't know, years of drought, it can suddenly shift its abundance. And so it can suddenly become pass to a threatened status.
Okay, the next presentation is by Paweł Radzikowski. I'm not sure why I have all the Polish people here. But he works at the Department of Systems and Economics of Crop Production, Institute of Soil Science and Plant Cultivation. Paweł does research in environmental science, and today he's going to tell us how common agricultural policy affects Orthoptera diversity in Poland. Paweł, please.
Many thanks for opportunity to be here. It's a great honor to present before you our humble agriculture research results. But before this, I guess doesn't work. Maybe this one. Okay.
This one is forward.
Sorry for that. I'd like to dedicate this presentation for recently deceased Professor Anna Liana, who was a leading Orthoptera researcher in Poland by the last 30 years. She also greatly contributed to our project and our personal knowledge about these insects. Maybe short introduction to the agriculture. Agriculture is one of the leading factors for the Orthoptera decline in Europe and the rest of the world, but this factor is not the same. Along the continent, there's a huge diversification, for example, in the size of the farms. In the Poland itself is also diverse. Most of the big farms, bigger than 20 hectares, like here in the Netherlands, are concentrated in the northern western part, but the rest of the Poland is characterized by very fragmented farm structure, most of them below 5 hectares of area, and also fields themselves are very fragmented. Farmers sometimes divide fields between their children to the point some of them are very long and narrow, some of them are only 2 meters wide. In the modern times, that resulted in the huge area of abandoned land in some regions of the Poland, which coincides with the most of our Orthoptera species observations. Agriculture in Poland, and especially the land holding, is characterized by the huge number of the small farms. Over 70% of them is smaller than 10 hectares, and the biggest ones which are above 50 hectares consist only of 3% of whole this group, but they do hold 30% of the land. So this structure is not that extensive, is still a lot of great fields. Most of the agricultural land is actually arable land, about the 70%, which is not very suitable for the Orthoptera insects, and only a little above 20% is grasslands and pastures. In the arable land, most of this, over 70% of this is cereal itself, followed by fodder crops and rapeseed. To assess the effect of the agriculture and environmental schemes, my colleague Jarek Stanenga established a project for the protection of the biodiversity in agricultural areas and also assessment of different practices. We use many indicators: farmland bird index, landscape diversity, floral diversity, and of course arthropods such as spiders, carabids, and orthopterans. We mostly make our research in the second pillar of Common Agriculture Policy, which is dedicated to environmental protection and rural development. We assessed organic farming on the arable lands, as well as the different variants of meadow management. For the control, we use actions from the first pillar with the only direct payments, with only basic good agriculture practices, both for the grasslands and the arable lands. Research area was placed in the eastern Poland, in Lubelskie region. We had the network of the monitoring on the grasslands and arable lands. The method we used were sweep net and soil traps, not really characteristic for the Orthoptera research, but that was the method for the collection of all of our arthropods. And we unfortunately found out the abundance of the Orthoptera in these samples was very, very low. So we have to combine all the samples from whole year, for whole field in the one sample. But we still achieved almost 100 samples from the arable lands and almost 200 from grasslands. The monitoring was performed between 2012 and 2016. First results, I'd like to show you classical comparison organic versus conventional crops. We found out the communities of the Orthoptera were mostly the same. Dominant species was Tetrix subulata, followed by field cricket. Also, Tetrix tenuicornis, Roseliana rosei, and Decticus versivorus were found in the huge numbers. The rest of the species was very unabundant. The rare species, for example, were Bicolorana bicolor and Horhippus mollis. What is relevant is that almost all species massively increased their numbers in organic farming. There was only several exceptions, and which resulted in the higher diversity of Orthoptera insects in organic fields. We split these variants into the 4 different variants depending on the intensity of the cultivation, based on the surveys with the farmers. We found out that only intensive conventional cereal production is performed less than organic systems, but extensive conventional is still pretty good. We also take soil samples to assess the presence of correlation between, they can be indicator from the poor quality soils, just like a Tetrix subulata. For example, he was more present in the fields with high nitrogen, so also not really balanced soils. On the other hand, there were species like Roseliana roseli and Decticus versivorus who were present only in the most fertile fields rich in the nutrients and magnesium, for example. Another part of the study was a grassland assessment. The control was conventional meadow out of the agro-environmental scheme. The actually research subject were variants with extensive farming, delay mowing date, and limited fertilization, a bird protection program with no fertilization and very late term of mowing, and also free habitat grasslands. They were different by the level of the humidity and vegetation structure. What we found out actually is that the conventional meadows were actually very diverse, and the dominant species were Horhippus albomarginatus, Horhippus bigotulus, and also Steptophema grossum, which was considered actually at the time the rare species. In the extensive variant of these meadows, we found huge domination of Gryllus campestris and Roseliana roseli, but we also found rare species such as Metroptera brachyptera, which were not present in the control. Another situation is in the bird protection package. We had a huge domination of Pseudohorhippus paraleus, to the point when we found 700 individuals in the one sample, for example. So it is like level of the locust, actually. The same situation in the Arnaterion semi-dry meadows, domination of Pseudohorhippus paraleus, but a lot of cricket as well. On the Molygnion habitat meadows, different species were dominant. There was Conocephalus fuscus, Homocestus viridulus, and other species typical for the wet habitats. On the especially wet meadows, calcium meadows, where often stagnating water, the same species were dominant as in the Molygnion, but plus Steptophema grossum. The conclusion is most of the common species of Orthoptera actually declined in the agro-environmental schemes. Some were winners like Pseudohorhippus paraleus, Gryllus campestris, and Roseliana roseli, which increased massively in the numbers, but also some less common species increased in the numbers in agro-environmental scheme meadows. And we also found rare species such as Metroptera brachyptera, Euphystera brachyptera, and many other tetagonids only in the agro-environmental scheme meadows. The interesting thing was that in the semi-dry meadows, when the mowing is very delayed, we achieved massive numbers of grasshoppers. It's especially true for the juveniles, which I could not determine. There were many, many times more than the conventional with early mowing. For the species number, there was actually no difference between species number along the experiment. For example, for exception, For the wet meadows, because they have a different species composition, not all species like to live there. And for the Shannon diversity index, only Arenatherium semi-dry meadows perform worse than others because of domination of one species, and the wet meadows because of different species composition. Summarizing, most of the Orthoptera species benefit from the common agriculture policy, especially agri-environmental schemes and organic farming, but some of them benefit more and achieve like very massive numbers. But the most important conclusion is we found rare species only in the protected meadows. For the future plan, we're planning to submit publication for the agronomy journal when we will present our research on grasshoppers in organic cereal. As well, we already submitted a proposal for the big project with remote sensing of the pastures when the on-ground monitoring of the grasshoppers will be performed on the large scale, of course, if the project will succeed. Thank you for your attention. I hope you like it, and I'm open for the questions.
Thank you very much, Pavel. Okay, we have some minutes for questions. Who has got a question?
Axel Hochkirch. I remember when I was in Poland 30 years ago, I was on a very small field, maybe potato field or something, and I was very surprised to see so many Orthoptera on a cereal field, which if you go to northern Germany, you find nothing. It's completely empty. You may find a Tetigonia at the edge or so, but not in cropland. So is this, as I've understood, it's still a case that you have many Orthoptera also on fields and not only in meadows. And
that's right, yes.
And isn't there a risk that this may change with the agricultural intensification?
Yes, it is a great risk, and because fortunately the agricultural policy by the many years is supporting small farmers to farm. Otherwise, we will be left right now only with the abundant land or very big farms. So is many direct and indirect effects of these policies for the Orthoptera insects, most of them, I think, positive.
Thomas Zunagradke. It's quite unexpected that the differences are not so high between patches with funds and without funds. And can it be possible, I think this last statement goes in this direction, that the main factor is still the small size of the patches? Because if you have a small conventional patch and a small organic field, you have more effects of border and of fields just beneath, and so that can compensate, for instance, the use of pesticides in conventional fields or so, and still have quite good numbers of Orthoptera there. So that the main key factor is really the size of the fields for the diversity in these Polish agricultural systems.
Actually, there was a significant difference between organic and conventional. Maybe I didn't mark it in the right way, but it was. The difference between extensive and conventional and organic was not significant because the extensive is only like sieving and collecting without any measures of the production. It is like really dominated. The size for the fields, which I presented before, it could be a little bit misleading because on our research, we only choose the meadows and fields no smaller than 1 hectare and no bigger than 3. So the differences between the size of the was not that like in the introduction, actually. Sorry for this.
Okay, thank you very much, Pavel. I think, yeah, please. Okay, the next presentation is by Nefeli Kotica. She recently finished the MSc, and for her master thesis, she studied the Orthoptera fauna of Crete, about which she's going to tell us a bit more in the presentation. Nefeli.
Which is, okay, forward. This one. Okay. Oh, perfect. Okay. So the pointer is here. Okay. Okay. Which one did you say was the pointer? The pointer is this one. Ah, okay. Oops. Hello. My name is Nefeli Kotica, and today I will present to you the topic, the Orthoptera fauna of Crete, new data and preliminary biogeographical analysis. So, Crete is the biggest island of Greece, situated in the South Aegean area. Oops, okay, here it is. No, that's not it. Where is it? Okay, anyways. And from literature now, sorry about that. Ah, okay, it was there. Yeah, okay. Yeah, from this island, 73 species are known according to literature. So let's start with a brief history lesson on the geological history of Crete. Once upon a time, the whole of the Aegean area was a single landmass called Aegeis. In the Miocene, about 12 to 9 million years ago, it was split in half by the mid-Aegean Trench, which permanently isolated Crete from Anatolia and the Cyclades. A bit later, around 5 million years ago, Crete was also isolated from Peloponnese and the rest of continental Greece. During the Pliocene, Crete was an isolated archipelago of 4 to 5 paleo-islands, which reunited in a single landmass around the Pleistocene. This turbulent history led to the existence of a large number of endemic species. For example, almost 1/3 of the species on the island are Cretan and are endemics to Crete and its surrounding islands. Specifically, there are 3 endemics in Crete and its surrounding islands, mostly Cyclades, Carpathos, Icaria, and Antikythera, and an additional 18 species and 1 subspecies are endemic specifically to Crete. Most unusual is the Eupholidoptera genus, which includes 11 endemic species on the island, confounded in different parts of it. So the aim of this study was the phonistic and biogeographical analysis of the Cretan Orthopteran fauna, which was based on specimens deposited in the Natural History Museum of Crete and the literature up to 2020. We mostly focused on updating the catalogue of species, on the effects of altitude in the Orthopteran fauna, and the biogeographical patterns inside the island. Let's move on to the materials and methods. So we examined the 30-year-old collection of the Natural History Museum of Crete, which led to the identification of about 20,000 specimens. These, as you can see in the map, are covering all the dominant habitat types of the island. Most of the specimens were mostly collected with pitfall traps, with the occasional use of fermenting traps and hand picking. And as a result, there are many ground-dwelling species in the collection. So in addition to the examined specimens, we collected all the published sources and some gray literature up to, until that was published until 2020. And if in any case that the altitude or the coordinates were not included in the papers, we would just calculate it using ArcGIS and Google Earth. In order to examine the effect of altitude in the fauna, we separated the island into 6 different altitudinal zones and then calculated the total number of species in each zone. We presented the results using bar charts and regression lines. In order to see the phonistic similarity and patterns between the mountain ranges, mostly in the island, we used the Yaccard index and we created a similarity tree based on the UPGMA algorithm. So, what were the results of all this? Firstly, we found 5 species that were not previously recorded on Crete, which are Pteronemobius heydeni, Parabangoblistes novaki, Stenomnemobius gracilis, Rhacocleis insularis, and Rhacocleis antikythirensis. These 2 Rhacocleis species were previously known only from the small islands of Antikythera and Cyclades that are close to Crete. We also discovered the females of 4 Cretan endemics that were previously described only by their male counterparts, which are Rhacocleis derai, Eupholidoptera mariana, Eupholidoptera gemellata, and Eupholidoptera cretica. Also, we discovered the male of the species Grillomorpha cretensis, which was described by its female. Finally, we had some new endemic species on the island. One of them is Leptophies, a species of Leptophies, for which the only information we have for it is these 2 pictures, basically, and the species Eupholidoptera Maria Therese, and a new species of Grillomorpha. Okay, these are some extra taxonomic notes. I will not stay on them a lot. So let's see the effect that altitude has on the Orthopteran fauna. As you can see here, there is an exponential decrease on the number of species as the altitude increases. This is a very common pattern in Orthoptera and other insect orders. Specifically in Crete, it has been also it was also documented in plants, but in other groups like Gnaphoside spiders and Isopoda, we can see that they basically plateau in low and mid elevations, and they decrease only on the higher altitudes. So what could lead to this exponential decline of Orthoptera species? These are only some examples, some hypotheses maybe, I don't know. The increased habitat variety in lower elevations, the extreme conditions of high altitudes, the higher sampling efforts that happen in lower elevations and especially close to urban areas, and finally the species-area relationship that we all know, which is especially in Crete, high altitudes cover a much smaller area compared to lower altitudes. Okay, so now let's see how endemism patterns are affected by altitude. You can see here that the number of endemic species is more or less constant throughout all altitudinal zones, while the rest of the species decrease exponentially. This results to the fact that in the lower elevations, where is it? Yeah. In the lower elevations, about 20% of the total number of species is endemics, while when you go to the higher altitudes, almost 60% of the species are endemic. Yeah. Okay, so you can see similar patterns like this in Coleoptera, in Gnaphoside spiders, as well as in plants, always in Crete. Yeah. Okay, now let's, sorry. Now let's see if there are any other patterns inside the island. It looks like the Orthoptera fauna of the lowlands is more or less homogeneous if you exclude some endemic species that are confined either to the west or to the east of the island. But that's not the case for the mountains. Specifically, you can see that the easternmost, the central, and the westernmost mountain ranges are very similar, are grouped together in the similarity tree. This could be a result of the Pliocene Paleo islands that existed, yes. And these islands could also explain the fact that there are some species, some endemics that are confined in the eastern part of the island, some in the western, some exist only on the tops of the mountains, etc. So if we look now at the relationships, the relations of Crete, the phonistic relations of Crete and the surrounding areas, we can see that we can see an interesting pattern. The Cretan fauna is closely related to the Cyclades, which can be seen also by the number of endemic species that these two places share, like the Gracocleis species and the Acrometopa cretensis. There is also a phonistic similarity to Anatolia, which can be seen by the large number of Eupholidopteras that both areas have. On the other hand, it looks like we don't have much of a relation to Peloponnese and the rest of Greece. One example of this is the fact that there are no Greek endemics that you can find both in Crete and the rest of continental Greece. There are only Cretan endemics, South Aegean endemics, and then general East Mediterranean endemics and larger distributions. This is very interesting since actually Crete was separated from Peloponnese much later than it did from Anatolia and the Cyclades. In conclusion, we updated the Cretan species list from 73 species to 78, 81 if you count the new species. We showed that an increase in the altitude leads to the exponential decrease of the Orthopteran species and the increase of the percentage of endemic species in the total number of species. And lastly, that the Orthoptera fauna of the lowlands is homogeneous, whereas the mountains show a clear differentiation between east and west. The Cretan Orthopteran fauna is related to the Cyclades and Anatolia, but not to continental Greece and Peloponnese. These are my references, and I would like to thank you all for your attention. And, yeah.
Thank you very much, Nefeli. What about questions? Oh, we get a tough one.
Thank you, Dragan Chubanov. Nefeli, do you have some hypothesis about explaining this strange biogeographical pattern? Why Crete has more similar fauna to southwestern Anatolia? Is it some kind of faunal transfer through the cyclads, or?
Unfortunately, I'm not really sure yet, but I know that it has been noticed in some other organisms, like the Dentarus beetles, which also follow a similar pattern, but to be honest, I don't know yet.
Okay, and one more question, if anybody has.
There are a few questions online. Maybe you can take a look later to the other questions. I select one from Josip Skeo. Let me see. He wants to collaborate with you on the Adriatic fauna. And there's one, maybe I can ask a question by Michelle Lemonnier. She asks if there is overgrazing in the higher altitudes, and could that be the explanation for the decrease in species richness?
Let me think. I don't know if I have enough data to support this at this point. There is definitely a lot of grazing, but it's more or less constant, so it could be one of the reasons, yes.
Okay, thank you.
Thank you.
Thank you very much. Okay, the next presentation is by Oliver Hawlicek. He's a scientific manager of the Molecular Genetics Lab at the Centrum for Natuurkunde in Hamburg. And his key interests are evolutionary biology, biodiversity research, and species conservation. Although he did study other organisms, everybody can make mistakes, of course, like water beetles and reptiles, he's working already for quite a few years on Orthoptera, and today he has a question for us about phylogenomics of Acridiidae. Oliver, please.
Yeah, thanks for the introduction. Yeah, I always get hate. I get hate from the herpetologists because now I work with insects, and I get hate from the entomologists because I worked with herbs and other things. So yes, maybe I'll just add a little to the introduction. Forward. Yes. You already know me. I'd like to quickly introduce Edgardo and Ricardo, who worked with me in this project. Edgardo is a bioinformatician at the Technical University of Munich, and Ricardo is an evolutionary biologist at the University of Munich. Yeah, and the background of this project was my earlier work on the barcoding of orthopterans in what they call Western Central Europe. So 3 countries, Austria, Germany, and Switzerland. The goal was just to establish a barcode library in the hope of this allowing us to identify species based on DNA barcodes, which worked in some extent, but one of the main findings, was that we could not identify all species by DNA barcoding. In fact, there were quite a lot that we could not identify. Initially, the problems are due to the DNA barcode per se, because we use the cytochrome C oxidase 1 marker, which is a mitochondrial gene, and the mitochondrial genome may not be representative of the whole genome. It may have a different evolutionary trajectory. And also, it is just a very tiny part of the whole genome. The most obvious case in which this did not work out is what I call here the Cordyceps bicuculus complex in the context of our barcoding study, which is this assembly of very notorious species, Cordyceps bicuculus, Bruneus mollis, and interesting for us, we found that we could not even distinguish them genetically from Cordyceps apricarius and even Gomphosorhipus rufus, which is a whole different genus according to current taxonomy. As we all know, we can distinguish these species using songs, for example, and morphology, but with the DNA barcoding, we found this was not possible. There may be some reasons for that. We try to speculate at least what might cause this, and one reason is that these were assumed to be very young species, evolutionary, very young species. So they had probably developed into different species only, possibly during the Pleistocene, during the glacial cycles. In some cases, as we know, hybridization is still possible. So this may cause the integration of mitochondrial genomes from one species into the other. We also know that orthopterans, especially acridids, have very large genomes and with a lot of pseudogenes. Pseudogenes are copies of genes that are non-functional but can be sequenced instead of the ortholog of the actual gene, and this may cause trouble in the barcoding. And finally, Wolbachia infections may play a role that cause various kinds of genetic chaos, I would say, that makes it more difficult for us to study these groups. Yeah, based on this, we started asking some questions, such as if we just use genomic data, more data especially, will this be more informative? If we do this, will we find different results? So more informatives, or will we also find that maybe the results we had so far based on mitochondrial data are misleading compared to the nuclear genomic data? If we find this, what will be the taxonomic consequences? So what we did in this small project is that we used transcriptome sequencing, which I will explain, with the example mostly of the Cordyceps bicuculus and related species, but also including some other acridid species, just to be able to see if the same holds true for other species too. What do we do for transcriptome sequencing? Well, obviously we start with a sample of, in this case, a grasshopper. Just a quick repetition, the transcriptome. So we have a genome, obviously, which is all the genetic or genomic information that an individual has, which consists of genes and much other material. The genes, the actual genes that code for proteins, only make up a small proportion of the whole genome. If we want to specifically target genes, the transcriptome makes sense because the genes are transcribed into messenger RNA, and this is then translated into proteins. And by extracting the messenger RNA, we get a set of the genes that are currently active and transcribed. This can be sequenced, and this will hopefully result in data useful for phylogenetic reconstruction based on hundreds or thousands of genes. Not only one gene, as in barcoding, or not only 16 genes, as in a whole mitochondrial genome, but hundreds of thousands. Ours is not the first study. This has been done by several people, for example, in the study of Burden et al. 2015 by Frida Meyer, already on Cordyceps bicuculus. And a more recent one is the study, the phylogeny of Orthoptera as a whole, by Song et al. 2020, and there's more. But so far, our group has not been included. So we did the transcriptome sequencing. Nevertheless, we started with the mitochondrial genomes, which were also included in the transcriptome because we wanted to see if we already found some information there. We also used this for a molecular clock study based on the complete mitochondrial genomes. And especially here, I think you can hopefully read this. Well, the first thing we see is that this is largely congruent with DNA barcoding in that we still cannot distinguish these species. So you see that there's Bigutulus, there's Mollis, Bigutulus again, Mollis down here, Rufus in the middle, then Bruneus down here. So this is basically a big mess in which all these species are at varying positions. Nevertheless, we used this mitochondrial phylogeny to use a molecular clock and confirmed what we already knew before, but also based on our data, that this Cortipus bigutulus complex, so here there's a mean age of 1.38 million years with some standard deviation. So anyway, it's certainly younger than 2 million years old, which partly explains that at least the mitochondrial data has just not been evolving separately for long enough to show clear distinctions. But what we were more interested in was the phylogeny based on those 540 nuclear genes that we put together here. This is just the overview of the nuclear genes and the mitogenome, and the straight lines highlight species that are in the same place, and these crossing lines highlight species that are not in the same place. So the first thing we found is that these were incongruent. We found big difference between this whole phylogeny of the nuclear, of the many genes, and the mitochondrial phylogeny. Yeah, I think this should be visible here. So one thing we find here in the coalescent tree is, or the nuclear tree, is that this may have to be confirmed by adding more specimens, but at least here with the few we have, we find that all the species of the Cortipus bigutulus complex become monophyletic. So this is Cortipus mollis forming one clade, this is Cortipus bigutulus forming one clade, Eisentrauti here. Okay, we only have one of Rubra tibialis, we have two of Bruneus forming a clade, and so on. Rufus, Gomphosorhipus rufus, is still here, is now the sister group of this complex. Yeah, and then Apricarius is finally outside. And this is different to what we found in the mitogenome, so this may show us that the mitogenome may just have evolved differently and may not be a very good representation of the whole evolutionary history of these species. So that shows us that this genomic data is more informative than only the barcodes and the mitogenomes. However, we have a very limited sampling here. We just included a few species that we could easily get hold of and made a preliminary phylogeny. This does not yet allow us to draw any good taxonomic conclusions. Some are pointed to, but we don't have a very comprehensive data set yet. So what we're currently doing is we're increasing our sampling across Gomphosorini, mostly including the type species, Gomphosorini. Not only, in fact, we want to include as many species as possible, Cortipus and other genera, and also other groups of acridiids. We want to use transcriptomes, for which we need fresh data, but we also want to use historical samples because it's not always easy to get hold of fresh samples for all species. And we will use targeted capture sequencing, which is a bit more complicated than transcriptomes, but can be applied to historical samples too, with the goal of, in the end, having a good taxonomic revision of this group sometime in the future. Also, what I would like to do personally is find a single marker of any nuclear gene that can work for barcoding, that will allow us to distinguish these species, because it should be there somehow, but we haven't found it yet. So far, I've just checked those 540 genes, but so far, only combinations of many genes allow the distinction of these species, not a single gene yet. Well, but I keep searching. Yeah, I have a lot of people to thank for, some of whom are here, and the funding bodies, and thank you for your attention.
Thanks very much, Oliver. Who has got a question?
Thank you. Dragan again. I have two questions. One just for personal use. How did you calibrate it for the molecular clock of the mitochondrials?
Yeah, it's not a very good calibration. We just extracted the calibration points from the phylogenies Pahojun song, which were in turn calibrated by fossils, but fossils which are pretty far out groups of what we have here. We don't have any good internal calibration points. So yeah, this is as close as we can get with the calibration. The other option would be rates of mitochondrial evolution.
Thank you. And another one, you said you believe you will find the gene that could separate. I doubt it a little bit. Maybe for certain groups, yes, but in general, maybe some SPNs from transcriptomes. And how do you see the future of this transcriptomics to be used in the barcoding of the organisms? Thank you.
Well, I think with the further development of technology, I think we'll be able to, at some point, not necessarily replace barcoding. We will have tools that allow us to use more genomic data for species identification. The problem is first, I mean, this is already becoming more and more affordable, cheaper and cheaper, but the analysis is not yet straightforward. So this will only be possible at the time at which a very easy analysis is possible. You just click a button and then you identify a species using the genomic data, which works for barcodes now, more or less. You just paste the sequence into the database and then it'll identify your species. But this is not yet possible with genomic data. As of now, it still takes weeks or months of analysis. And only once this is possible, we'll be able to use this instead of barcoding. And the second thing is that the crucial point is the reference library. So there's a huge reference library of millions of specimens of many, many species for DNA barcodes, but not yet for genomic data. So this would also have to be established anew, and this will take, this will, if it will ever exist, it'll be years, decades in the future, I think. But the development is there.
Okay, I think we have to go to the next, I'm sorry, we have to go to the next presentation. Thank you very much, Oliver. The next presentation and the last one before the lunch break, at least it's a name I can pronounce, Jaap Bouwman. He's an ecologist at Crown Estate at Hello, and he is also the co-author of the 2015 update on the Orthoptera grasshopper fauna of the Netherlands. And now today he's going to tell us more about the possibilities of DNA meta-barcoding offers to assess the food preferences of 2 species of grasshoppers.
Okay. Yes. Okay. Thank you for this introduction. There's only one presentation between this moment and lunch, and it's going to be about food. So everybody stay quiet. I will try to talk very fast, so we get to the lunch very fast. It looks a very difficult title about DNA barcoding, and it is a bit difficult, but the most difficult part is going to be told by Kees van Bochhoven, so I'm off the hook, more or less. The part in the beginning is mainly about grasshopper shit, so it's quite fun. The main question is, can we use DNA techniques to unravel grasshopper diets? So let's see if something like that can happen. We looked at mainly 3 species, but one of them is still, the scats are still in the freezer, so we have no data about them. The one left up is Tetrix bipunctata, and the one down is Ephipra diurnus. The other one is Gamsocleis glabra. Of these 3 species, we collected scats to see if you can use DNA barcoding to look what they've been eating. These are the research locations. It's mainly the Veluwe. Jeroen van Leeuwen also said about his presentation about Ephippidae diurnis, that the Veluwe is a main area of this species. And it's also the area where Gamsocleis glabra is present, and also Tetrix bipunctata. But from especially Tetrix bipunctata, we have only one location. So we needed another location, so we went to the eastern parts of Berlin to collect some scats there as well. This is one of the research areas. It's a big military area called the Oldenbroekse Heide. It's a very big area, it's still in use, as you can see, with big tanks, and they shoot on them with big grenades, so it's very safe field work there. You should stay on the paths and have a good look before you go into the heathlands, because there's munition everywhere there. But it is a very nice spot with a big population of Ephippidae diurnis, but also Gamsocleis glabra there. This is the only location of Tetrix bipunctata in the Netherlands, as far as we know. It's a spot of about, I think, well, 5 by 5, 10 by 10, not more. And it's the only place where we can find them. I've been there with Rob to collect the scats, and we needed about 1.5 hours before we found the first one. So, and that's on a location where we know it's there. So that shows how difficult to find new places it be for this particular species. This is a photo of the Trampa area, which is more or less alike. You see a lot of herbs there. It's close to the forest edge. A lot of open grounds, but especially a lot of herbs and flowers. So the field work, this is again the location of Tetrix bipunctata, where I did the field work together with Rob. And when you go in the field collecting scats, you need a few things. You need some gloves, because you don't want to contaminate the scats. You need some clean tubes to put them in, and then you can basically go on. This is the field work. On the right side you see a lot of our German friends who helped us with the field work in Trampe. And on the left side there's a picture you're going to see quite often, because I've already seen this picture before in the presentation of Roy. That's Rob Felix on the ground on his knees. You can see, you see it quite often. It's a normal sight in the Netherlands. So, and then it's quite easy, because you put the grasshopper in a clean tube, and then you wait, in most times, 10 minutes, 15 minutes, maybe half an hour, and then they will shit. This is Tetrix bipunctata, who, with the two droppings, which were round of shape. I always expected that all grasshopper scats would be elongated, but it's not true. But Tetrix bipunctata, they're round. So something we found out now as well. And what we also did, we did not only collect the adults, but we also tried to find out if there are differences between adults and nymphs. And that seemed a good idea, and it is in practice, but it was quite difficult to find the nymphs, especially of Ephippidae diurnis on some locations, especially when the populations are very low. They were extremely hard to find, but in the end we collected enough individuals to get enough scats to do the analysis. And then at the end of a day of field work, you've got a car trunk full of small tubes with grasshoppers in them, and at the end, hopefully, they have all done their thing, and you have enough scats to take home. And the good thing about this is that you can release the individuals after that and just take the scats home. Okay, I think now the more difficult part will come, and I'll give the word to Kees.
Well, then they arrive at the laboratory, and yeah, you have to do some analysis on land. That's where I came in. My name is Kees van Bochhoven. And in the laboratory, we started with the DNA extraction. So you end up with a vial with water, with the DNA dissolved in it. We do a PCR. We do after that we sequence the DNA and do some data analysis on them. Well, the PCR is the critical part in the process. There you can decide what markers, what part of the DNA you are looking into. And we focused on 3 markers. TRNL, which is a chloroplast marker. We used it to characterize what plants were eaten. 16S, which is a mitochondrial gene and is mostly used for characterization of what invertebrates are there. And 18S, which is a nuclear gene, and we particularly looked into the V7 region, and that's used like it's quite a universal marker. And you can, at the same analysis, look into fungi, but also algae. In fact, all eukaryotic organisms do have this gene, so you get a really broad idea of what the grasshoppers were eating. Well, interesting to know is that we could only retrieve very short fragments, especially of the mitochondrial DNA. So it was not feasible to amplify, for instance, CO1. You could retrieve some sequences, but most, well, it was not. We were not able to get a full diversity based on the CO1 gene. And that was actually only feasible using the 18S marker, which is a nuclear marker, probably because it's located inside the nucleus and thereby more protected from degradation. Well, the lab work was done in a laboratory which is dedicated to working with samples that contain a low level of DNA. And that's necessary because otherwise you could easily contaminate your samples. So yeah, you have to work in a really clean environment. So what do they eat? We found what we expected was maybe Tetrix maybe would eat some mosses or maybe algae. But what did we find? Actually, most of the DNA we could retrieve was fungi. So both basidiomycetes, ascomycetes, also several other groups as algae, but fungi were was the biggest component of the diet. It could be both. It were both the fungi, but also the fungi that are represented in lichens. Well, then the results about a vippiger. We looked into the difference in the sexes, and actually there was only a difference between a statistical difference. Well, let's maybe start with explaining a bit more the graph. On the y-axis, you can see the fraction of the diet, and that's based on the DNA. So the fraction of the DNA that's from a certain group of species. And well, the vippiger shows a more varied diet, and also plants were eaten regularly, and as well as metazoa. So that's mostly represented by invertebrates in this study. The only significant difference, if you look into the different sexes, were found in the Harskamp, where females ate more fungi than the male nymphs. Larger differences were present if we looked into the differences between the two sites and the differences between the nymphs and the adults. Interestingly, and also understandably, metazoa, so the invertebrates, were mostly eaten by the nymphs, probably because they need a more protein-rich diet to grow. And also, there were differences between the amount of fungi that were eaten versus plants. So at the Harskamp, statistically more plants were eaten compared to fungi at the Harskamp. Well, then we zoomed into the plants to see what different plants are eaten by the grasshoppers. First, again, the differences between the sexes. The only significant difference that we found was between in the herbs, the consumption of the herbs, where at the Harskamp the females ate more herbs than the males. Also, when we looked into the more of the details, interesting is here that more herbs were eaten by the nymphs, and especially at the Oldebroek, less heather was eaten, so they really switched there to the herbs instead of eating heather. Well, some of the more the methodological conclusions. Well, first, it did indeed offer some insights in the diet of the grasshoppers, but you need to take into account that there's a lot of differences between samples. So there's a high standard deviation, so you need quite some samples to get statistical significant results. And also interesting is that the DNA in the droppings is highly degraded. So yeah, you can only look into really short fragments, and that limits your taxonomical resolution. And then Jaap will tell some conclusions about the ecological part.
And I already saw Luc saying two minutes, so he's going to be a bit strong now. Ecological conclusions, I can be very quick because Kees already showed some things, but they are preliminary, and we need more data to sustain them and a lot of more research. But it seems that the nymphs prefer more mineral-rich food. This pattern is best shown at Harskamp, and in the Oldenbroek, which is a far better area for eviviger, it is less pronounced. The Oldenbroek population show a rich diet with more herbs and less heather, which we thought at the beginning they would eat a lot of heather, but they seem to prefer other herbs. And at the Harskamp population, adult males eat more woody plants and less heather. So maybe this will affect the nuptial gift. It's also something we would like to look into. For the research, we still have got a lot of scats of Gamsoclias in the freezer, which we need to analyze, but we need to get some funding from anybody who thinks, well, let's start looking at grasshopper shit. Sampling more tetrix would be nice because we have only from 2 locations, and on those locations also not a lot of scats. And we can do some more detailed analysis to see why they prefer fungi or even metazoa on one location and not on the other location. Final conclusion, DNA metabarcoding is an interesting way of looking at things. It's a new way of looking at things, and it can be promising, but we need more sampling and we need more fine-tuning to draw some final conclusion about this matter. Okay, thank you very much.
Thank you very much, both of you. I'm sorry to keep you on a short leash. Questions?
Thanks, Oliver Havlicek again. I have a question on the methods. So you said that you only retrieved very short fragments. So what size did you retrieve that you could actually analyze? My first question. And did I understand that right, that you got shorter fragment, or the fragments of CO1 were shorter than of the other mitochondrial markers too? Is that right?
Yeah, most likely because of CO1 we were not able to, well, we did retrieve only few sequences. We did use a 16S marker, which is about 60 base pair, and that was feasible, but then your taxonomic resolution is very limited. The TRNL marker, this the plant marker, is a short marker, but does allow for quite a good taxonomic resolution, which was also about 50, 60 base pair. So that's really small. Yeah.
Okay, thanks.
Anybody else? You're all hungry? Nobody on the chat? No. Okay, thanks very much. Before you run off, I have a few announcements again. To start with, I told you this morning there are some small carton boxes that side. Yeah, mugs for each of you is 1 mug. Don't forget to take one, because otherwise I'm left with a lot of them. Then I forgot to mention this morning, but I'm going to offer 2 guided tours to the collection, one this lunch break and the other one tomorrow. There are, with the mark boxes, there's a small list where you can fill in your name, but there are only 10 people I can only take 10 people at a time. So I'm sorry about if there are more people who want to join, it's simply not possible logistically. So, but if you, I just saw there's still some spaces, so if you want to join, put your name. The first tour will start at 1:15 in this lunch pause at the office entrance in that corner of the big hall, 1:15. Then don't forget to go up sometime and look at the posters which are up there behind me. And finally, we are going now, before you get lunch, yeah, it's a terrible time here in Holland. Before you get lunch, we have to make a group photo of you. So I want to ask you to go down to the big stairs. Maybe we should lead the way. Maybe you should lead the way. And after the group photo, there will be lunch served here where there was coffee also. And I see you back at 14:00 sharp, please.
Does it work here? Yes. We can continue with the program, and we will have the first presentation by Anton Kristin, who is from Slovakia. Most of you may know him because he organized the last European Congress on Orthoptera conservation, which was a great venue in Slovakia. And yes, we look forward to your presentation.
Thank you, Axel. Dear colleagues, nice afternoon. So let's go to Phaneroptera, to the species living here in Europe and in Central Europe. They both are within the 2,400 species in the world, what is nearly 9% of the world Orthoptera species fauna. Genus, it is 44 species, only 4 we have in Europe, and 2 we will study. Then description, they are very good flyers, polyphagous, polytopic, and some are good colonizers. Both of studied species are late season species. It seems to be that, at least in my study area in Slovakia, the Nana is later than Falcata. From Orthoptera species file, we can see the map of Falcata, limited in the south on 23 degrees and 60 in the north. And Nana we have quite lower or smaller area and range, but there are some introductions in United States and some question introduction to Australia, but maybe that is not correct. To the questions, the main question what we today mentioned several times, this so-called expansive species, it is really knowledge increase, species expansion, or sometimes can be read also about the recolonization of historic range. There is question, how big is the percentage of syntopy in overlapped range of these 2 species? There we hypothesize that Falcata, as known till now, is going higher than Nana. It is similar like with Sparsa and Spinoza. Is the Falcata more in forest zone distributed? Nana, riparian forest fringes and urban habitats. So to the goals. The first, I would like to review the distributions. Then differences in latitudes and altitudes in this overlapped range. Then to analyze a little bit habitat and woody species. And then the last, or not completely last, are the rivers in this case, also the corridors of Phaneroptera spring, like in Rusfolia or some similar species. And then also such ornament of what is the Orthoptera species richness in these Phaneroptera sites. When we check the situation with Falcata in Europe, this from IUCN Red List, the first data coming from end of 19th century from these countries, from Slovakia 1867. Then we have data from Poland, all data from Czech Republic, then we have from here Benelux, some from United Kingdom, Anglia, then we have new data and new data and new data from the northern parts of the range, what seem to be really expansion. Next one, Nana. Also we have here some updated range from IUCN Red List for Africa and Asia. And now we can see the first in Austria, 20. Then we have Hungary, then we have in Slovakia, 1962. Czech in Prague, isolated, probably local introduction. Then probably local introduction in the cities or rural and urban areas in Germany, as shown in the last paper of Hokier and collective. Overlapping range, we can see now the overlapped range in Europe, Falcata and Nana. And we can see now the updated limit of distribution in Slovakia. So, to the methods and material. In these years, 96 to 21, we studied 2,327 sites. What we can see not only by colorful circles, but also by empty circles in this map. And approximately the year 2010 was given as a threshold for these comparisons. We had the 47.4 data before 2010 and nearly 53% after. Then each side was swept around 1,000 sweeps of bush and herbal vegetation, sometimes also by acoustic identification, by visual collection. Then we described the 7 habitat types in the sites and 9 wood species or wood categories. And we studied, following in the last 5 years, riparian versus non-riparian sites, mainly the river banks along the 12 rivers, and 125 sites in each habitat in these 5 years. So, to the distribution of both these species in Slovakia in whole period. You can see that falcata we found in more than 30% of sites, nana nearly in 10% of sites, and mixed population we found only in 2.7% of sites. When we compare the periods, 96 to 2010, and then after 2010, so this period, this threshold 2010, then we found 46% of data before 2010 and 52 after 2010. So it seems to be that there are no big differences in the northern border of distribution of this species, but we found in the last yellow period distributions in higher altitudes of Carpathians and in the periphery. But the trends is not easy to estimate. To Nana. Now the trend seems to be in the data collection maybe also, but also increase in the sites in yellow period in the last 10 years. You can see the really nice contribution in around the whole Pannonian area of Slovakia, you see, and this is also coming northernly, more northernly. The color, this purple, is in both periods, and only 2 sites we have what was only up to 2010, and we did not found this species more. When we compare the main habitat groups in these sites, what I mentioned, this 95 sites of Alcata and 245 sites in Nana, then we can see that Alcata prefer or was found mainly in the broadleaf, within the broadleaf forest, coniferous forest and mixed forest, it means in the forest zone, and the Nana mostly in the riparian fringes, riverbanks within the rivers, main rivers of Slovak Carpathian range. Then Nana was also significantly more in urban and suburban areas. You can see also the main habitats of Falcata here, also the altitudinal distribution on riparian vegetation. Now it is correlating also with the main woody species. The Falcata is, Nana and Falcata are very similar in oak, within the oak forest, in the lowland sites. Falcata is more distributed in the beech forest zone than just opposite, the Nana in the willow and poplar, and other woody species or other scrubland and so on prefer more or is located more in other habitats, other woody species than is mentioned here. So now to the small experiment during the last 5 years. Along these 12 rivers, what we can see in this picture here, 4 rivers in the western Slovakia, 4 rivers in the central Slovakia, and 4 rivers in eastern Slovakia. In the case of Nana, the red dots show that Nana is going quite very far along the also mixed populations along the rivers, also here in the eastern Slovakia, also in the western Slovakia. Here not so high because here are the much higher mountains and the basins in the central Slovakia, but here we can see the clear break everywhere where are not going far the Nana, only Falcata. Of course, we did also 125 sites in the surrounding habitats, not in the riverbanks, but in surrounding habitats, and you can see the Nana is coming only in this low Pannonian area, these yellow and red triangles, and the Falcata is nearly everywhere. The gray triangles are no Phaneroptera data from the sites. Here is the comparison in the graphic. Falcata is mostly in control habitats, Nana more in riparian habitats. Both species occur in the same habitat very similarly in riparian forests, and no Phaneroptera. These are these data. One explanatory variable for distribution of the species can be altitude. Sure, Falcata is coming higher. Thus can we see on the gray graphic. Also the mean, as the Nana, what is coming only up to 530 meters above sea level, and Falcata up to this altitude. Nearly 95% is going in Nana under 350 meters, when Falcata to 750 meters. Now something for these habitats. So the highest position where we found Falcata is Low Tatra National Park, but we think this is only local occurrence because this is well-flying species. That no breeding site we found there, no names. This is position of the site within the area, or within the range of Falcata in Slovakia. Now we have Nana in Slovak Karst, completely in the south of Slovakia. In the warmest position, Nana is going also higher than usual in Carpathians. Now, small comparison, how is the assemblage around Falcata, Nana, and the mix. Interestingly, we found that in mixed, in sites with the mixed population, is the species number the highest. But it is not easy to interpret. We should take this result with caution. For example, the differential species, also for your introduction, Camptoxifa or Polysarcus or Tetigonia cantana and Pseudoboris manadia are characteristic or differential for Falcata localities. For Nana, we have xerothermophilos like Krausi, Pelucens, Ephipiger, Euchortipus dercleus. And now to the conclusions. So we found only 2.7% of syntopy in overlapped range of both species. Spreading rate. The term spreading rate we have to consider with caution because maybe this is the spreading of our knowledge. But when we compare it in Nana and Falcata, we see that Falcata has the similar ratio as is ratio of the sampling effort, so the 50-50 nearly. But in Nana, we have 74% of all data after 2010. Yeah, of course, we confirmed that Falcata is going higher, also latitudinal, also altitudinal than Nana. It seems to be global pattern. Falcata is more in forest zone, beech and conifers, and Nana seems to be more in riparian forest fringes and, of course, in urban habitats with poplar, willow, parks vegetation. And regard to the rivers as corridors, Nana occurs more northernly than Falcata along rivers than in surrounding habitats. So thank you for your attention.
Thank you. Are there any questions?
very interesting to see that we have a very similar pattern in other countries as well. In Germany, we have also a spread of Nana, but we mainly find it in urban areas, actually, while falcata is more in the countryside.
Thank you, Szymon Czyżewski here. Very interesting. I wanted to ask, what do you think, is it the spread of Nana in the river riparian habitats rather due to its spreading in these habitats, or rather that these are like more suitable habitats for Nana than for falcata?
This is not yet decided because, you know, we have no enough data about riverbanks, habitats, riparian habitats from the typical Nana area. We have enough data only from 5 last years, and we have comparable data from the riverbanks and the surrounding habitats. But when we will consider the habitats in the typical core range of Nana, I am not sure if the riverbanks will be the main preferred habitat. Maybe the urban parks or some.
Yeah, I wonder whether they could also be distributed by garden centers or so, because they are always found in gardens in Germany. Okay, any other questions? If this is not the case, thank you very much.
Thank you also.
So we come to the second presentation, also from Slovakia, by Sonja Nudličková. Is this correctly? Okay. She's working on a very exciting project on one of the most threatened species in Europe, Esophia babienkoi. And yeah, the floor is yours.
So thank you for your attention. My name is Sonja Nudličková, and now I would like to tell you something about distribution and ecology of endemic bush cricket Esophia babienkoi, and I would like to show you preliminary results. So, study species. It is Esophia babienkoi flightless species with low dispersal abilities. It is mostly crepuscular to nocturnal species, and it is listed as critically endangered. As you can see, it is the endemic species. We know only one site from the Slovak Kars National Park. It is the site in the Slovakia in the Central Europe, and the geographic range is very small. However, we still need to improve the current knowledge. There is a limited information about distribution, species ecology and biology, and information about population connectivity. So, our study aims are to investigate the current distribution. We conducted the first systematic survey of all historical, recent, and expected sites. And then we would like to determine microhabitat requirements based on geobotanical data. And finally, we would like to provide the first information about genetic variation. Regarding material and methods, here you can see the investigation of the distribution. In the season 2021, altogether we conducted 21 transects. We walked through more than 300 kilometers to provide detailed information about the distribution and to cover almost the whole study area, almost all plains in Slovak Kars National Park. Here you can see examples during our investigation. We included recent, historical, but also potential sites to check the presence or occurrence of the target species. Regarding determination of microhabitat requirements, we use 2 approaches. First, we have done vegetation survey on confirmed GPS locations of the target species. And secondly, each GPS location of the bush cricket we intersected with normalized difference vegetation index. It is an index derived from remote sense land cover data, and this index provides us the information about vegetation density. For instance, red color indicate bare soil, orange color sparse vegetation, yellow, green, blue colors dense vegetation. And finally, we extracted DNA from his femur, we made PCR reactions, and finally we obtained sequences from mitochondrial DNA genes. And here you can see the results. First, this is the situation or status until 2020. There are all historical data to this year, and you can see this by indicating yellow dots. But now we expanded many historical sites, and we found some novel sites, especially on the on the western part of the study sites. There is completely new. And regarding habitat requirement, we found that the species occurs on the sites with rich vegetation. For instance, we found more than 200 species of vascular plants. And here we compare the number of mono and dicotyledonal plant species. Monocotyledonal species are, for instance, grasses. Dicotyledonal species are broad-leaf flowering plants, and we find out that there are more broad-leaf flowering plants. Similar pattern we obtain it when we look at the cover regarding these two plant groups. And here you can see pictures with the target species sitting on the Vincetoxicum hirundinaria, and our results suggest that these broadleaf plants provide shelter, microclimate, protection from predators. And in most cases, there were the higher plant species, and we also our results suggest that the vegetation architecture is very important for these relatively heavy insects, as you can see. Both the ecological indicator values indicates that the species occurs mainly in medium light demanding species in semi-humid habitats. You can see these two pictures regarding light conditions and moisture. And here we comparing NDVI indexes, we didn't find any significant difference between sites, and our results indicates that the species prefers ecotonal habits on the border of sparse vegetation, shrubs, and forest edges. You can see as this yellow color, yes, here is a forest, here are very hot places, for example stones, and here are the ecotonal habitats. Here you can see examples of habitats with the confirmed presence of Isophia babenkoi. For instance, broadleaf semi-dry grasslands, species-rich mesic meadows, subxerophilo fringe communities, or fringe communities in the contact with xerophil grasslands. And here our first genetic data indicate that we found 15 haplotypes. The greatest difference is between western part, green dots, and eastern part of the study area, red dots. And here is the dominant haplotype indicated by brown dots. But however, here you can see also another haplotypes suggesting some level of isolation too, yeah, on the specific sites. And here, this dashed line indicated that we need more data, more sampling, because here on these planes we have a low sample size. And with this research we will continue. Main conclusions, we provide detailed information about species distribution. Extent of the occurrence, we doubled this value, but however the species still falls into critically treated category. These are preliminary results, and the research will continue. Regarding determination of microhabitat requirements, we confirmed strong link with dicot herbs, and we find out that the species occurs mainly in ecotonal habitats. And here we found a relatively high genetic variation. With our data suggests at least 3 different lineages, and we suggest that these fragmented populations with limited gene exchange. And here I would like to show you future research. We would like to identify more potential sites using NDVI model and vegetation data. We would like to study population sites using mark recapture methods and species mobility. Then we would like to study reproduction biology in ex situ conditions. And finally, we would like to do street analysis to create a conservation plan. So this is my Greece. Thank you for the project donor and Slovak Kars National Park and my team. And finally, thank you for your attention.
Thank you very much also for keeping the time so well. We have plenty of time for questions.
Yes?
Dragan.
My question is, you still identify the species as a critically endangered, no matter the plenty of new data. Have you identified new threats, or you stick to the already proposed ones, to the IUCN, and which ones are these threats?
What I have to identify, new trends?
Threats.
Yes, I think these are preliminary results, and the research will still continue, but maybe it would be different. I don't know till now. I'm not sure. Don't know?
Maybe I can add something to this. Sure, there are identified already some threats for species, because this is abandonment of the pastures in the Slovak karst. It is the succession of the shrubs and the woods there a lot. And then, of course, there is also another extreme, the intensification of moving, and this, I don't know how to tell this kind of the moving and living there, the grass. Mulčovanie. How we can say English? Mulching. Thank you.
Yeah.
Thank you. This is what is not very suitable strategy for all of Orthopteras, I think, or most of Orthopteras, not generalists. Yeah. So I don't know if I answer you.
Treats, I don't understand correctly. Yes, I think there are many serious treats. For instance, car driving is a big problem through pastures and grasslands. This is the first thing. Maybe too intensive pastures and succession, I think. Forest is very dangerous, especially for the red dots that I shown. There is very small very small sites of the species where it occurs, and it is very dangerous because the forests come to the rocks and it's over. Yeah.
Sonia, I'm a bit confused about the meaning of the dominance of dicotyls over monocotyls. I think I don't know any area where monocotyls are dominating dicotyls. This is just a normal pattern. We have about 6 to 1 on a global scale. And if you count species numbers on a separate area, you will always have a dominance of dicotyls. You could also say, okay, less grasses around, but this doesn't come out from your data. So what is the meaning of this graph where you show that dicotyls are always dominating the vegetation around the findings?
Yes. I think these are only just preliminary results from the first season. Of course, this is only positive controls. Till now, we determined what plant species are on the places where the species occurs, positive. But however, the next season, now, we plan to do negative controls. We would like to compare also other places where the species not occur. So we get maybe the good chance to analyze this picture that we would like to hear. Yes, this is just initial data, just to show where the species is, which vegetation there is. It's very simply.
There's one online question from Ho-An Ree. It's about the threats that were mentioned. Do parasite flies also include threats of the species? Do you know?
Once more, please. Do you?
Parasite flies, whether these are threats to the species.
Deutsch?
The flies which are parasiting. Fliegen? Fliegen. No.
No.
I generally, well,
No.
think it's a natural process, but we never know what's really going on in the world of parasites. Okay, I think we have to go on. Thank you very much again. The next presentation is by Lisbeth Sechner, who is now leading the project for the conservation of the crow-playing grasshopper in southern France, so another exciting species.
Good afternoon to everybody. So I will present you our live project on the grasshopper in the crow plain. I work for the Conservatoire d'Espaces Naturels in southern France, in Provence-Alpes-Côte d'Azur. And I just have to look how it works. It's the other one. Der Grüne. Dankeschön. I thought somebody will do it for me. I'm sorry. Our project is located in the Crow Plain, not far away from Marseille, in the former Durance River Delta. And I think most of you know perhaps this area, but because it's really known for its bird species and for its biodiversity. Parts of the area are designated as a nature reserve, and it's also Natura 2000 area, according to the two directives. The natural habitat is mainly dry grass, pseudostepic grassland. It is called Kosul in the local language, and it was grazed for centuries, and it's still grazed with quite a lot of sheep. So we have around 30 to 40 sheep flock, mainly in the spring. You have the typical Mediterranean climate. It's quite hot, and we have also strong winds. It is called Mistral, so the summer is quite dry and hot, and in winter it can be also quite cold. As in France or in Europe, dry grassland is really endangered. So habitat loss is one of the main reasons for this area to have quite a lot of endangered species. You see on the left side the loss of this Kosul habitat during the last centuries. But it is still going on, so the pink parcels you see here, we lost about 1,000 hectares between 2008 and 2020. And it's due to construction, to urbanization. We have also pollution. We have quite a lot of industrial zones around the area, and also intensive agriculture, so there is also a problem perhaps with pesticides. And we don't know exactly, but there is also a change of grazing practices ongoing, perhaps linked with climate change. Another problem for the grasshopper is the predation, and there are several bird species in big numbers which have increased during the last years. So one species is the cattle egret, who came in the crow plain mainly in the 1990s, and it is linked with sheep flocks. And we have also other species like different corvid species who are mainly observed with the sheep flock. And as the grasshopper is quite early in the season, it will be eaten by the bird species. Another species of bird could be a problem or have been a problem perhaps or probably. It's the lesser kestrel. It's the same. The species arrived in the crow because there were several projects to install nest boxes, and it's really in the period where the grasshopper started to decline. So this crow plain grasshopper is endemic in the area. It is protected in France because due to the strong decline. And in 2014-15, IUCN with Axel Hochkirch and Laurent Tatin of the Conservatoire started to work out a conservation strategy. And at the moment, we know only 3 isolated populations. You have a small population in the center of the Cros, Baudemont. The blue parcels here around show the area where the grasshopper was known, but it has disappeared. And then we have 2 populations in the periphery of the Cros plain. One in Calissan, it's a military site, and the 3rd population is on the test center site of BMW. The 3 sites are isolated. The species is not mobile, so the capacity to recolonize the area is very limited. The species is classified as critical endangered on the red lists, and this gave us the possibility to ask for life funds 2020. So the LIFE project is based on the conservation strategy and on all the work which has been realized during the last 5 or 6 years. So we try to continue in the same, with the same actions, almost with the same actions. We will work on habitat management, on the reduction of the threats like predation by the birds. We will do breeding of the grasshopper, and like all LIFE projects, we will have also quite a lot of action on communication, education, awareness raising, and exchange with experts. The Conservatoire is the coordinating beneficiary. We work with 3 associated beneficiaries, the Chamber of Agriculture and 2 zoological gardens. We started last September, and the project will last for 4 years. We have almost a budget of almost 2 million euros with 60% of co-financing from Europe. And here you have all the partners and all the people, the team of the project. So there are quite a lot of people who work within this project and who are involved. Some details on our actions and the expected results. We are starting now, so I don't have really results at the moment. We started with studies to improve our knowledge on the links between grazing, vegetation, and habitat of the grasshopper. So we will also use remote sensing to understand what happens the last 30, 40 years in the Cros. Was there a difference concerning the grazing practices? We will also work out grazing management plans for the reintroduction sites and for the existing subpopulations. We will also work on the habitat restoration to reopen areas, and we also work with the shepherds, and we will improve the accommodation for shepherds because it's important to have still this kind of management of the sheep flocks. For this, we have quite a lot of material to communicate with the sheep breeders and shepherds. Concerning the birds, we do a little bit the same thing. We try to improve our knowledge on lesser castrate, but also on corvids, mainly on species which are breeding in the sheep barns. One action which was already realized during the last years is mobile fencing. We did this in Baudemont, and it was combined with the closing, or the nest boxes of the lesser kestrel were closed, and we observed that the population was increasing in Baudemont, because if you don't have the grazing, vegetation will be higher and denser. It's mainly Linda Bröder who worked in the framework of her PhD, who showed that this denser vegetation is quite favorable. So we will go on with the same actions, but on a larger scale. The most important part of the LIFE project is the breeding of the grasshopper, with the idea to start a reintroduction program. At the moment, we work on a combination of in situ and ex situ breeding, because we don't manage the whole cycle in the crow. The period of the egg pods, the conditions are very particular, so we don't manage them outside of the crow. So the LIFE project allows us to work with 3 ex situ breeding stations. It's Cathy Gibault, who is the coordinator of this action. She was already involved also during the conservation strategy. So we want really to increase our breeding capacities, and if everything will go well, we would like to start reintroduction 2024, and with the idea to increase the range of the grasshopper. This will be, first we will also do analysis, and we will work on a strategy for this reintroduction according to the guidelines. Concerning the population monitoring, we have different methods. We will work, for example, with detection dogs. It's also something which was already tested in the crow with Laurent Datin and Rita Santos with her dogs, so we will do it again this year on sites where we didn't find the grasshopper for the last 15 or almost 20 years. And one method which was optimized by Linda Bröder is the capture-recapture, so we will continue with this method, and this will mean that you have to go 16 times on one site with 3 persons, so it's a quite heavy protocol, but it allows us to estimate the population size. The grasshopper is not really easily to detect, so it's the only method to know the population size. Here you have the results of this capture-recapture with Kalisan, where my colleagues started 2013, and you see there are fluctuations. Last year, unfortunately, it was decreasing once again, and Podemo, where we had our actions with the mobile fencing and the translocation of the lesser kestrel. First we got good results, but unfortunately last year it's the same, the population size decreased once again. So we will do this capture-recapture this year at the site of BMW and Kalisan once again, to know whether it's really linked to a decrease of the population, or whether it's we have fluctuations. And fortunately also I will have 2 students from Axel Hochkirch, so we will have enough people to work with us. Some examples of our communication supports. Life is Life, with many possibilities to communicate. We have also the website. There is already a film on the grasshopper, which you can find in the internet. And one final subject, which is very important for me, is the international cooperation. We started with Axel Hochkirch last December with the first conservation symposium, where we invited experts from the different countries, and we discussed on all the Prionotropis species, with the idea to know better the status of the different species, and to look also whether there are common issues. It will be grazing, and this could be one point to work together on the management of the species. I'm sorry I was too long. So thanks a lot, and if you have any question, perhaps one.
It was not too long, it was fine, it was great. Thank you very much for this presentation. Are there questions from the audience? Yes, Roy.
I was wondering, in the Netherlands there's a lot of talk about the food quality being of influence on the grasshoppers and other insects. Is that something that you look into for this species?
Unfortunately not. We didn't look, but now we started we intend to do perhaps a genetic analysis to know what the grasshopper is really eating. Because I think it was also a problem, or it could be a problem of our breeding. They gave not really the plants you find in the cropland to the grasshopper, and we had some difficulties in the breeding, in the ex situ breeding. And I think we should give really plants from the cropland. And unfortunately, we don't have good. We have a list of plants, but we have to improve this knowledge. Yes, thanks a lot.
Okay, thank you very much again. So we move on. Just a reminder for those who ask questions, please shortly mention your name because the people on the internet will not see you. And the second is that people who are, so the speakers could actually also see their presentation here, so they do not need to turn around. Okay, we will have the next presentation by Gergely Szévényi from Hungary. He is, well, one of the leading orthopterists in Hungary, I could say. Yes, you are. And he's very humble. But he will tell us something about another exciting species, on Paracaloptenos calaptenoides. It's really not easy for the tongue. The floor is yours.
Hello. Thank you, Axel. And first of all, let me say a few words about the co-author of this presentation, about Barnabás Nagy, who was really a leading orthopterist in Hungary. And I would like to this one? Okay. Okay. So I would like to dedicate this presentation in his memory. He passed away about 1.5 years ago, and before we started to prepare this material together. Thank you. And after, let's see that nice species with nice long name. Balkan pincer grasshopper was described from a locality of the territory in Serbia by Brunner from Wattenwil, and after that was described two further subspecies, one Brunneri from Bursa from western Anatolia, and another by Fer Willemse from southern Greece. So the species distribution, as you can see on that map, covers most of the Balkan Peninsula, western Anatolia, and in the northwest reaches the Pannonian region as well. Here, at the northwestern range border, the species became very rare and localized. In Austria, it was discovered nearly after a century in recent years, and now, according to the recent publications, it is known two populations there. In Slovakia, in South Slovakia, it is known only in the last 2 decades, and recently it is known from 11 locations. In Hungary, where the species is known for a longer time, it is listed in the Hungarian Red Data Book as actually endangered species. It is protected since decades, and now is strictly protected in Hungary. The species is listed in the red list of Carpathian grasshoppers as endangered, and for Hungary and Slovakia as critically endangered. Our aims was to collect all available data on the species distribution for Hungary and get as much new data as possible. The study period covered a very long time, thanks to Barnabas. By the fieldwork, we documented the locations and some other data on the site. Let's see the distribution of the species. All distribution data shows that the species inhabit the hilly and mountain areas of Hungary, from the middle Transdanubian mountains to the northeastern mountain ranges of Hungary. The western part of the occurrence, unfortunately, gone. According to our best knowledge, the species regionally extincted from Bakony Mountains, from Vértes Hills, and also from Buda Hills, where it is very nice that the specimen, the only specimen known from Buda Hills is before the description of the species, the only one data. So recently, Paracaloptenus caloptonoides occurs in the eastern part of former Hungarian distribution. In Visegrad Mountains, we know 3 recent populations. In Bergeny Mountains, a bit north of this first place, nearly 20 populations. In Cserhát Hills, 7 very tiny, small populations. In Matra Mountains, also nearly 10 surviving populations, similarly to Bükk Mountains and Aktalek Mountains. So, as we can see, if we compare the formerly known populations with the recent knowledge, we can see that the population trend is okay, or stable or increasing. Yes, but it is unfortunately most probably not true. If we see the extent of occurrence, the former 1100 square kilometers just halved, the AOO remained nearly the same. So when we consider that we detected nearly 30 local extinctions, we could consider the trends to be rather decreasing. And according to the recent knowledge, the species could be categorized under the IUCN Red List criteria as endangered. We can see a bit in detail the picture. In the recent western distribution edge in Visegrad Mountains, the species was known at 7 locations, and to check the recent distribution, we made thorough targeted research in this area. We checked more than 160 seemingly suitable habitat patches to check the presence of the species, and unfortunately we couldn't find it, not in the former known localities, but we finally found 3 recent populations, but only one of them is not a really small one. That southern population covers nearly 20 hectares, while the other 2 small populations cover less than 1,000 square meters. So they are really small ones. In this picture, you can see individual trees, and this is one habitat. A bit north from this place, in Bergheim Mountains, it was known the species from 2 localities, and after a similar survey, we found a much better picture. We found 17 new populations. Most of them were not so big, but at least between 1,000 square meters and 1 hectare. If we see the habitat requirements of the species in Hungary, we could find that the species inhabits grassland patches between 200 and 900 meters above sea level, with a dominant elevation between 400 and 600 meters. The exposition of the habitats inhabited by the species was a bit surprising for us, because we expected that it will be prefer the south face slopes, and we found that prefers rather southwestern, southeastern slopes, and not the real southern ones. As it is known, Paracaloptenus caloptanaeus lives in Hungary also in relatively short grass, open grasslands with small plots of bare grounds and sometimes with stones, as you can see in that picture about an actual habitat. So we concluded that the primary habitats of the species in Hungary are the edaphically treeless, so primary, generally small patches of dry, sometimes rocky grasslands, in generally a forested, hilly and mountain landscape. However, we found the species also in sometimes in secondary habitats, human-made or human-influenced habitat like mountain hayfields, pastures, or even forest clear cuts. These ones were or are somehow connected with the primary habitats, since the species is wingless or short wings with very low ability of movement. The population size is generally very small, very low. We found most of population with rather tens or sometimes hundreds of specimens. So most of populations has a relatively high local extinction risk. The main endangering factors, as we heard about in the previous presentation, mostly are in connection with the habitat quality. The forestry activity is maybe one of most important ones. Forest planting and cutting forestry activity may disturb and even totally eliminate the suitable habitats. And also spontaneous reforestation and succession may eliminate the habitats. In these 2 pictures, you can see a place in Matra Mountains. It was in the 60s, and now you can say that formerly hand-cultivated, partly grazed and moved grasslands just disappeared and replaced by bush and forest. Another problem is the game. The overpopulated game populations, especially white boar and mouflon, may totally destroy the vegetation. You can see two otherwise seemingly very suitable habitats from Visegrad Mountains, but in one of them nearly without any plants, and in the other picture with very few of one. Other problem is the intensification or the abandonment of habitat use. In this picture you can see machinery moved grassland, a former habitat of the species. And I think that I have not explained the human activities, consequences. So the future, what are the future prospects? The main task is, in the remaining population, the appropriate habitat management to protect that kind of short open grassland from the succession and invasive plants, to reduce the game pressure, keep away possibly the forestry activity from that patches. And since that habitat plots are of very low or have no economical value, but often are categorized as forest by the forestry, it is sometimes a problem. Since most inhabited patches are very small, it's very important to maintain that places because of the very low ability of recolonization by the species. So finally, these small populations require special attention and sometimes active conservation measures, but since they are really small patches, it is not often a mission impossible. And finally, I'd like to thank the founding of Bükk and Danube Ipoly National Park Directorates and the help of colleagues in the fieldwork. And finally, thank you for the attention.
Thank you very much. Maybe one short question. Yeah.
Thomas Zunagradki. The population you mentioned in Austria, these two sites are in fact connected together. It's one population. And it was possible to increase the area inhabitable by this species by cutting down a pine forest and turning it into a sheep pasture. So I think this, do you think it is possible to, because you know, you mentioned it, forests are somehow also quite strictly protected, but in this case, for this special species, which is also of European Union, it was possible to just cut down the forest. So this
fortunately, most of population occur inside the protected areas. So we have some chance, but it's sometimes not too easy to convince the forestry. They like to
they love trees.
To be, yes, even pine trees. So just be tree on that plot.
Yeah. So. Thank you very much. Very interesting presentation.
So we come to the next threatened species, which will be presented by Filippo Bozzetti, who is working on several threatened species in Italy, but particularly currently on Soneriana marmorata.
Thank you. for your introduction, and good afternoon, everybody. I will talk about Zeunariana marmorata. This species, it is listed in the red list of Orthoptera by the IUCN as endangered, and was thought to be extinct since its discoveries and since few collectings in the early century. Then population was rediscovered by Roy in the 90s, and this instigated further researchers by other orthopterologists in Italy. And after a national plan by the IUCN for Italy and for this species, we have been able to find other subpopulations where this species it still occurs and is abundant. In 2016, we had a result of researchers with about 6 to 8 active subpopulations in Italy, and mostly on the coast of Adriatic Sea in northeast Italy, and one population in Slovenia, in the inland of Slovenia. Then we started more deep researches thanks to financial support by Mohammed bin Zayed Conservation Fund, and we had the possibility to investigate each population or subpopulation at the bioacoustic level and at the population density level. Here you have two pictures. If you compare the picture on top of the slide, you see heterogeneous locality with more than one habitat, but this is not a positively heterogeneous habitat as Thomas this morning stated, because in this locality, Zeunariana marmorata lives only in the high herbs on the right of the picture, and can walk in the shrubs and in the tree habitat, crossing this low grass field that is crossed by cars and by mechanics for land movement. So all the individuals that goes through this part of the locality will be possibly killed. Instead, if you look at the picture on the bottom of the slide, you see a very homogeneous habitat where the species is very abundant and is safe. It is a very coastal habitat and very large that hosts a very dense subpopulation. So now we can have the first assumption that even if we are worried about the restriction and the fragmentation of habitat, we can assume, or at least hope, that even small suitable habitat can host a population of this species. And so we have checked the number of individuals for each populations. Here you can see also the morphological features of the species. that is from the Handbook of Italian Orthoptera that was recently published, and the population trends of the world-known localities it shows a very different pattern, and we can only say that maybe because we are thinking about 3 years of results, there are 2 populations that are increasing. Then, after spreading the voice between colleagues and other entomologists, some found another population, very new, very large and very dense in number of individuals, that is located in the Italian inland, in a very large river that goes to the Po River, that is the largest Italian river, in a park, not a national park, but a protected area, the Mincio Park. And so we get the second result, that is that we can confirm that this species is more present than we thought before, and that protecting and managing natural or semi-natural areas, it can be positive for species richness and species protection. So this is the new locality from Parco del Mincio, and you can see a very large meadows of Molinia, this tall grass that host a very dense population. In the last year, other localities along the Mincio River have been investigated, and we have found more populations not so far from this new one, but not connected, at least for what we know now. We have analyzed the song of almost all the populations that we surveyed, and we found that the song is quite similar. There are no appreciable differences. And then, despite that, some get in touch with offices and get all the necessary bureaucracy during COVID emergency, it has been a challenge. We had some interest from newspapers and the Natural Park Organization of Italy and other institutions. We had interest in sharing our results and our projects because it was found very interesting that this species, especially in the last locality, was found not looking for this species, but looking for butterflies. And so we are planning for our future and the future of this species. We will continue the acoustic comparison on Zeuneriana marmorata for the known populations. We are planning the translocation of individuals from the Caneo locality to Schiavetti, and will monitor other populations. And the translocation was made already last year. So this year, next summer, we will check if it was successful or not. We will have 2 students from Turia University in the field this year to carry out all the field activity. As you can see in the picture, there are patches, there are suitable habitats and localities in the region where these populations are present, but all the populations are isolated and are surrounded by hostile habitat. And as you know, this is the first step to extinction for species, the habitat distribution, fragmentation, and disappearing of small populations. Then we will enhance public awareness with information panels and information workshops. We will build fences not to avoid that insects, that the grasshoppers, the crickets move, but to avoid that people and the cars goes inside the locality where the insect live. And we will organize a meeting between Italian and Slovenian researchers to share our experience. Thank you very much for your attention.
Thank you, Filippo. Are there questions from the audience? Yeah.
Dragan Chubanov. Have you checked the delta for possible occurrence of the species? I think it's quite a vast territory. Or do you think there are habitats for these species there?
Yes, I think Ania checked the delta of the Po River, but without success. But I must say that there are many apparently suitable habitats and localities that they don't host these species. We don't know why. Maybe this is related to the some microclimate conditions that affect the presence of the species.
Actually, I think she found one population south of Venice.
Yes,
during her last year.
Maybe
Yeah,
one is.
there's still.
But it's far from Delta.
But many of these populations are extremely small.
Yeah.
So this is why you can probably find still some. Yeah. And well, I think the bioacoustics really helped to find it.
Yes, most are surveyed by ear, because it is very difficult to catch, to collect specimens.
Okay, so I think we have to move on. Thank you very much.
So the last presenter will be Luc Willemse. I don't have to introduce him, but he will talk about recent Orthoptera discoveries from Greece.
Yes, Greek Orthoptera. Greece is a country rich in species, and still the funny thing is new species are being discovered, which seems maybe a bit odd sometimes. I'm going to show you a number of slides with a lot of photographs taken in the field. This is the list of the people who I have to thank for all these very, very beautiful and nice images. I thought instead of finishing with this slide, I'll say first of all thank you very much to all these people for the nice images, and a lot of them are found in the field guide for the Greek Orthoptera. I'm shortly going to tell you something about some of the factors behind the high number of species found in Greece. Then I'm going to tell you a little bit about why, for some species or some instances, it may seem difficult to discover species at all. Then I have some latest additions to the Greek fauna for you, and I will end up with the latest species, or not one of the latest new additions, which is a Leptophius species. If you look at Greece, then obviously the northern part, the very, very northern part of Greece, still contains some elements of Central European fauna, which is exemplified by some of the species shown in this slide, like Mycostatus parapleurus, Schizochaon, Dispar, Bibetissus oxcari, and Tetrix turci, which are all species which are found more to the north in the European flatlands, lowlands especially, not necessarily in the mountains. And again, there are also some species in the northern part of Greece which also are more boreal and more Central European in their distribution pattern. But in Greece, you only find them at higher altitudes, like Podisma pedestris, or Acipta fusca, Psophis stridulus, or Stenobotrus stigmaticus. Then I show you a picture of, which already was mentioned earlier by Nefeli, about the fact that Greece, in some parts of Greece, there's a lot of geological history which has caused speciation in some of the parts of Greece, and Crete is maybe the top example of this, and maybe Euphylidoptera is the top genus where this is an example of. This slide shows 2, 4, 6, 8 species of Euphylidoptera found in Crete, Antikythera and Gavdos, but all in all, together with a more common species, this island with the 2 neighboring islands now, after the paper which is going to be published hopefully this year, will have 14 species of Euphylidoptera in total. And that's mainly because of the recent paleogeological history of Crete. Of course, Greece is a country with a lot of different habitats, which also are part of the reason why Greece also harbors so many species. It's quite obvious. It has nice rivers, streams. It has mountains all over the place. It has caves. A lot of caves, although my experience is they're very difficult to find. It has got maki, it has got frigana. It's a lot of different habitats which altogether harbor a lot of different species also. Then, of course, the altitude is an aspect. Some of the species found in Greece are limited to above the tree lines, like the Spanassiana and Oropodisma. Some of the species found in Greece you find everywhere, from the lowlands up to the high mountains, like Pseudochrotipus and Chrytippus. It was Bornholmi, but I should say Maritimus these days. And then there are some lowland species which you only find in the lowlands, like Tridactylus and Ochelidia. The other big factor behind the large numbers of species in Greece is, of course, also the fact that a high number of the endemics in Greece are found in species which have very poor, they have no dispersal capacity really, because they are short-winged. And this slide shows you that if you look at the the total number of endemics of Crete, this is just a number 149. It may be, I'm not sure, maybe it's slightly more, slightly less, but it's, let's say, 150 species are endemic in Greece. Most of them, by far, 140 are short-winged, and only a very few are long-winged, which is another reason behind, I think, the large number of species found in Greece. Then another, well, more or less in the same direction is the fact that if you look at four genera with the most of the species in Greece, they are all short-winged, and this slide shows you some of the where the species of these 4 genera occur. For Psilimon, a large number occurs on the mainland, about a third on islands, and some species found on islands and the mainland. For Dolichopoda, it's of course found in caves. It's a cave cricket, and it's found also in islands and in mainland, almost in the same number of species. Euphylidoptera, because of Crete, is mainly, most of the species endemics are found in islands. And for racoclase, it's about the same number on, not the same, but let's say more species found on islands than in the mainland. This graph shows you through the time from 1813 up to now, how many species are known from Greece, and the line is still going up. It's not bending yet. So how can we explain that although we have so many species, they're still being discovered, more species are being discovered? I think one of the factors is that the habitats where the species which are being described, especially the ones which are being described in the most recent years, where they are found, that's the habitat. Of course, cave crickets a cave is a habitat where it's not easy to go in and not easy to find them. So that's obvious that if you go into a cave where nobody has ever collected a cave cricket, chances are there that you find a new species. But also racoclase, which is a completely different kind of habitat. With this species, Racoclase crypta, is found on a scree slopes, even last year we found a new species of Racoclase again on a scree slope on Mount Parnassus. Mount Parnassus is in fact one of the best visited mountains of Greece, but still we found a new species of Racoclase last year, not this one but another one. Another surprise for me also was in 2015 or 14, the mail I got from Florin Hoeksman that he found some strange Homocestus along the coast. And this, as far as we know now, but I still have to look at it a bit more closely, but it's very close to Homocestus raimondi. But the fact that we had never found it before is that it's only there it's adult in May up to maybe mid-June, and then it's gone. So if you go to Greece in July and you go and visit the beaches, the lovely beaches of Greece in July, you will never see the species. So it's too early for it. So you're too late for it. So the seasonality of some of the species may also be a reason why sometimes they go undiscovered. Then again, another example from Crete, but you can show the same image for Greece. Greece, although a lot is being published lately, there's still a lot of white spots in Greece. This is a picture of Crete with all the localities where we have found Euphylidoptera species. The blue ones are the ones which have been published so far, and the green dots are the ones which are going to be shown in the new publications, which is coming up soon, I hope. And they are the result of hand catching, but also of a lot of pitfall traps carried out by the museum in Heraklion over the period of 40 years. But still, you can see there are some areas in Crete which have not been covered yet, or at least where they haven't found Euphylidoptera yet. Then another reason why we still keep finding species is the fact that some of these species have very small ranges. One of them is, two examples here. Brinotropis is a species which has a very small distribution area in the northwest part of Greece. And then, of course, there's Hortipus lacustris, which also has a very small distribution area linked to wetlands in northwestern Greece. And then I think this is the last example of why it's difficult to find species. There are, especially, for instance, in the Pamphagids, the densities of the population is so low that even if you walk somewhere, you have to be very, very lucky to and if species is there, you still have to be lucky to find it. And this is the last another factor why it's sometimes quite difficult to find species, and still is possible to discover new ones in Greece. Some species are very cryptic. This is an example, of course, of an ant cricket, which obviously you can only find if you start turning rocks and look in ant nests, which is not everybody's favorite pastime. Natula averni, it's maybe easy to hear, but not easy to see. And then this is Stinommobius gracilis. I was very happy to see an image of a male. This is a female, but it's quite a species which is quite elusive, at least in my experience. So there's all species in Greece, and still, because of all kinds of reasons, some of them are still being found in recent years. In the left-hand side are 8 species which have been described after the publication of the field guide in 2018, where there are except for the last Poecilimon, of course, was a known species, but the other ones are newly described species from caves. Then on the right-hand side, we have a mention of 2 new Orthoptera species which are going to be described this year. We have awaiting in publication Platycladinae genus from the border mountain with Albania, which is this is a new species at least. Pseudonotus fibri hasn't been mentioned yet from Greece. We have a new Rhacoclase from the Parnassus, and we have a new Leptophius from Crete. And then, which is a little bit more tricky, but of course we also, in these days, we also have iNaturalist and observations. And on these websites, you also sometimes find images of grasshoppers from Greece. One of them, which is pretty obvious, is Sphingomyces obscuratus, which was photographed I think in the Peloponnesos or something, I don't know, but anyway, from Greece, quite distinctive. And then there are some other images from the western coast of Greece, which may be Platyplegius. And from Crete, I've seen a picture which could be Sphaerchus palmatorum. So there is still species are being discovered even up to now. And I'm just finishing off my presentation with this Leptophius. This left-hand image was already shown also by Nefeli this morning. We found it 2 years ago when we were looking for this Orthoptera genus in Western Crete. Jos and myself, we were looking for Orthoptera, and we found some small nymphs. So we brought them home, and we raised them to adults, and they are very typical, very strange and new species. This is some more of the details of the species. It's contrary to other species of Leptophius, the last tergite in the male is expanded backwards, a bit like Andrenimon, but not in such a degree, but a little bit like that. And the cerci is a little bit spirally turned downwards. We found it on 3 spots in western Crete. Then, of course, we started describing the species, and then we had to think about a name. So I thought it was nice to just look up all the names of the endemic Greek species and see what was the etymology of the name for these endemic Greek species. It turns out that 61 of them were named after a person, 59 after some geographic entity, which could be an island or a mountain or a village or a cave. And then some 30 of them were named after a character, which could be a shape or the color or the behavior or the winglings, something like that. So, so now we had to come up with a name for this one. And of course, if we could call it Cretica or Cretensis, which is, in all fairness, which makes completely sense. We could call it Spiralis. But then we thought, no, we're going to name it after a person. And the very reason why I give this presentation is not because it has a very high scientific content, but because we named it after Axel. It's I did send the second proof to Zootaxa Tuesday, so it should be published anytime soon. Maybe today, maybe tomorrow, I don't know. Anyway, it's because of everything you've done for ECOC, for Orthoptera, for conservation. We thought it's high time you deserve a name. And not only a species, I mean, a nice species also. And also, I show you we have made a certificate for him to make life even more difficult. So I'm going to hand you over a certificate. Is it in one of these presents, maybe? No. It's is a certificate of this species.
Oh, thank you very much.
I don't know what to say, but I would like to thank you for calling it Axeli and not Hochkirche. Because nobody can really pronounce the name Hochkirche. But Axeli is much, much better. Thank you very much.
You're welcome. No questions.
There are. Are there questions? Still are there questions, yeah. Yeah, there's one.
Sorry for making you walk such a distance.
Oh no, that's good. It's good for my health.
I was wondering about the relation between the mainland species and inland species, because evolving on the islands means isolation, means limited competition, but on the other hand, situation is changing, climate is changing, there's a common transport and habitat loss. What do you think? What are the relations between those two groups of the species? And can the mainland species threaten local population somehow?
I'm not very sure I understand the question, sorry.
Make it shorter.
I ask for the relation between a mainland Orthoptera species and inland Orthoptera species.
Inland or island?
Island, oh, sorry. Sorry, mispronounced. What are the relations between the species? Are the mainland species competitive towards this island species?
I don't think they are competitive. I think it's a matter of geological history of Greece. That's the islands, when they were connected to the mainland, the process of becoming an island meant that the species living there became isolated, and speciation started, and they evolved to new species. They are, of course, they are related, but I don't think they are. I don't know. I don't know exactly what answer you expect. Sorry.
Sorry for taking so much time. I ask if the introduction of the mainland species doing the transport by the ships and the planes can threaten local species on the islands.
Okay. Well, that, to be honest, I don't know, because I have no good example for grasshoppers, at least, of introductions of mainland species from Greece to islands. I don't know. I don't know. Sorry, I have no examples. Yeah.
It's interesting question, but I think at least in Germany we see that on areas where Meconema meridionale is becoming expensive, that Meconema thalassinum appears to be disappearing. So it could happen, I think. Yes, it's possible. Yeah, so I give you now time for the coffee break, and we will meet back at, sorry, to check at 4 o'clock. So I've shortened the coffee break a little.
so we will continue to keep in time. And I have to introduce the next speaker, who is me. So I don't need to take too long. Okay, sorry. Can go back. Yeah, so the title of my talk is very simple: the rediscovery of two species which we thought could be extinct on Gran Canaria. And actually, I could do the talk in probably 1 or 2 minutes, but I will take longer. So when we did the red list for Europe, we had a couple of species which were threatened. 1/4 of the species are threatened, and we see that 4.6% are in the critically endangered category. And we saw some of them during the present talks. And we didn't decide to call one of the species extinct. And the reason was that we were never sure whether it's really extinct and whether it may have survived somewhere, like Ephipiga camille, for example, which might be the one which is really extinct. But anyway, so Derichorus minutus is one of the species which was classified as critically endangered, possibly extinct. And this possibly extinct flag was based upon the fact that it was described from a single male in 1954, and this male was collected at Maspalomas Beach in 1949. I don't know who of you have been to Gran Canaria before. Maspalomas Beach looks like this. It's a very small village at a very nice sandy area, a lot of interesting habitats. At least this is how it looked like in 1960, and this is how it looks like today. So maybe that's the reason why we didn't find it. We had a couple of searches, and we always looked in the vicinity of Maspalomas, and then we thought maybe it would be useful to go to other coastal areas. And we already had these plans when, in 2015, Heriberto Lopez, my colleague from La Laguna, found this photo on Flickr, which was taken in 2014 by David Marquina Reyes. And this photo was not taken in Maspalomas, but on the very opposite of the island, in the north of the island, also close to the coast in Sardina del Norte at the roadside. So I sent a student, Anja, some of you know already, to Gran Canaria to search for this species, but she couldn't find it at this roadside. So she also looked in other coastal areas where we thought the species might still occur. And she found another population here in the southeast of the island, near Arenaga. And this was a good basis for then starting more intensive research. So in 2017, I sent 2 other students, Anna Michaela and Jakob André, to Gran Canaria to have more detailed studies on the population size and distribution and habitat preferences of the species. And you see that the extent of this population is actually quite large. It ranges from Arenaga in the south to Vargas in the north. And the good news is that a large part of the population is in protected area. So it's probably not critically endangered anymore. But at least the southern part is in the area where wind farms should be built, and even Arenaga was, there were some development plans in the area, but now the local authorities also know that the species occurs there. The host plant of Derichorus minutus are Chenopodiaceae. All Derichor species appear to be specialized on Chenopodiaceae. And these are the 2 species, Zoaeda vermiculata and Basea tomentosa, which the species feeds on. And you can see a specimen here. Well, it's quite well camouflaged, but I think most of you see it probably here. This is a female, and this is a nymph. Not all nymphs are of this color. They sometimes are brown. Sometimes they can be very green, like rubber gums. And you always find them on these bushes. Well, Anne and Jakob also confirmed this record in the north of the island. And that was interesting because the purple dot here is a locality where David found it in 2014. But they found it south of these banana plantations. So this was interesting. So we thought, okay, 1 year later, I sent other students there, Claude, Johanna, and Mareike, who did some research on this northern population. And you can see that they
found that actually north of this banana plantation there is a large population, but there are only very scattered individuals south of the plantation. And this shows you already that the banana plantation is probably, or was probably, a problem. Yeah, this was probably a large connected population here, but the banana plantation has probably destroyed a lot of habitat. And they told me that in the south of the populations, it didn't look very well. There were some construction work going on there. So we contacted the local authorities, and they told us they're actually doing some habitat restoration measures there to restore some coastal vegetation. And we said, oh, that's perfect. Then probably there's a chance that this species will increase there again and maybe colonize this area. So this is the current situation for the Rhecoris minutus, and the question is now, what would the red list status be for a species which was formerly critically endangered, possibly extinct? We have now two populations, and if we look at the IUCN red list criteria, there's one criterion B1, which is based on range size and so on, and you see that we have an EOO, an extent of occurrence of 121 square kilometers, which is still small. We have two locations. A location is defined in the IUCN red list as an area which can be wiped out by a single threatening event. And we say, okay, this could be easily wiped out if the banana plantations continue. And the question is whether there is a continuing decline or not. So this is the third subcriterion which needs to fit until a species will be in a red list category endangered, if there was a continuing decline. If we say this was a past event, the decline, it's now increasing, then it would only be near threatened under this criteria. The same is true for the AOO, the area of occupancy, because it's also quite small. And finally, we have this third criterion, D2, which could also fit a species which occurs in a very restricted area, which can move to the critically endangered category very fast if, well, a threatening event occurs, a realistic threatening event. And the threatening event here could be an expansion of the banana plantation or even the urbanization in either Sardina or Arinaga, or maybe in tsunami or whatever. So vulnerable would be would be one choice. The second species I want to talk about is Evocodurus capreari. This is even more mystic. It was described by Bolivar in 1936 from a male which was collected in the Barranco de Agaete in 1930. And Heriberto Lopez had his study sites for Aminda and other species in this barranco, and he never found it. He said it's probably extinct. We searched for it, and all these students I showed you were always asked to go to the barranco and search for Evocodurus too. We have a good photograph, so it's easy to identify because it has a very strange pronotum, very restricted pronotum, so it's easy to identify and cannot be confused with other species. In 2018, I had 3 students which I just asked only to search for Evocodurus, nothing else. So that was not for their thesis, it was an internship, so they cannot, for a thesis it's very dangerous to have a objective like this. So Lukas Knob, Lisa Mahler, and Jan Kolmsee were there in September, October 2018. And then when they sent me a photo one night, shortly before they left, and yes, it was during the night they found a singing individual. And actually this is a female, as you see. So they heard a strange song, which they didn't know, so they approached this song, and after a while they found this female and made a photo. This was exactly at the type locality, more or less. So, but this was very difficult to reach because there were very steep slopes. They were there during night, and so they were really afraid to going further upslope. Because of this discovery, there was a press release in Spain and also in Germany, and the press picked it up quite quickly. So a lot of people heard of it also through the social media and so on. And a few days later, we got this photo of a nymph, which was found in July already at a different locality, actually. And so we went to this area as well a few years later. 1 year later, a smashed individual was found here in the city of Telde. That was on the road. Okay, we don't know where this comes from, whether it has been flown in or whether it has been transported there. So it's completely mystic. So in 2019, I sent 3 students again. One of them was Lisa, who also rediscovered this former record, to get more data on this species. But this was for their thesis, so it was a bit difficult because after 2 weeks, they told me they only found 1 specimen. And again, in the near La Aldea de San Nicolas, so in the second locality which was known. So I told them, okay, maybe you concentrate better on Oedipoda camariensis or something which you can easily find. Then I found in 2020 a photo on iNaturalist, and this was a record which was already from April 2006. So Alex Patera has collected these nymphs and raised them in his home, and yeah, so he had adults after a while. And I don't know if the sound will start now. I hope it will start. If not, because I have a song file here, but I don't hear it. So maybe it doesn't work, sorry. Anyway, so we had now the song of this species. I can maybe shortly sing it. It's a little bit like starting with the very high-pitched, and then yeah, and this is continuous song. And it's so high-pitched that only oh, sorry. I can't hear it. Yeah. During the night, yes. And the interesting thing about this is, well, we had now this song, and we now had a better way to search for it, yeah, because we had a bioacoustic method to search for it. But it cannot be heard by me, for example, and only 2 of the people who discovered it could hear it. So the man couldn't hear it. Then we had actually 2 records of nymphs, which were also sent to Heriberto, and you can see they are again from different localities. And you can see they are sitting always in rocks, in small crevices and so on of rocks. So this may be the reason why they are so difficult to find. Yeah, so it's probably, and it's very steep slopes. Yeah. So in last year, I had again 3 students who searched for it using the song, and they actually found 25 individuals in these 3 populations here. So they found another new population here, but again. Well, even though they now heard more individuals, they told me that they appear to be only very few. So apparently these populations are all very small, and it's active during night, so they found them singing between 10 and 12 o'clock in the night, and all records are from very steep rocky slopes. So one wonders, well, what can threaten a rocky slope? Yeah, except for mining, maybe, but I mean, there's so much slopes and Gran Canaria, so I thought maybe it's not threatened. But interesting, if we look at the extent of the recent fires, it looks like all the records are from outside the burned areas. Yeah, so I don't know if fires can really be a problem for a species living in rocky slopes, but it very much fits to the data we have also for California aluaudi, which occurs more in forests, of course. So what would be a red list status for this species now? First of all, the EOO is also very small. It's probably severely fragmented. Severely fragmented means that it has very small isolated populations, which fits very well here. And I assume there is a continuing decline because this complete area appears not to be colonized. But this is, of course, on a weak basis. So it could be endangered or also near threatened, depending on whether one would classify it as declining or not. And the same happens if we use AOO. So both species are not possibly extinct anymore, at least. So thank you very much. So maybe one question. I don't have the mic, so my double role is not filled very well. Any questions? If not, you can go to Gran Canaria and search for it.
In methodological question, the AOO and AOO estimation, did you use the Delaunay configuration and the Delaunay
triangulation and alpha counting?
We use the GeoCat tool. Do you know this? A GeoCat does an automatic calculation if you upload your localities of the species. Then it automatically uses a minimum convex polygon. Yeah, for the EOO. No. No, I think not. No, but, well, the EOO is for measuring risk spreading, so it's just the outline of the habitats, while AOO is based on a 2 by 2 kilometers grid. Yeah. Okay, so I have now 2 mics, so I think we continue with the next presentation, which is by Armin. Sorry, I need to check for the title. Armin Landmann is working on the alpine fauna. Many of you may know him from recent talks from an area which has the highest biodiversity of Orthoptera compared to Germany at least. Yeah, we very much look forward to your presentation.
Yeah, dear colleagues, my report today deals with wild rivers in the Alps and with elusive grasshoppers inhabiting the banks of such rivers. So let me just start off with a few data about the dimension and problems of the Pan-Alpine river network and of the traditionally high diversity of califera on these riverbanks. According to a WF study from 2014, this alpine arch spans about 1,200 kilometers and has river units, more than 10,000 river units, which total have a length of 75,000 kilometers. So such braided rivers, traditionally for thousands of years, of course, were key ecosystems all over the Alps. And you can still find such dreamlike landscapes, of course, south of the Alps, at the edge of the Alps, the Tagliamento River, or also at the northern border, the Lech River in the Tyrolean Lech Valley. But such dreams, I have to say, have long gave way to the harsh reality of modern times. You could say, south of the Alpine chain, north of the Alpine chain, here, Alto Adige, River Adige, or here in the so-called center of the Alps, near Innsbruck. What is a special tragedy is that especially the larger rivers on the valley floors, which would have space to shape such dynamic habitats, are the most impacted river courses. This is shown here again. Over only about 5% of the 11,000 kilometers of the larger rivers within a catchment area of more than 500 square kilometers have been assessed to have high ecological value. And this is, of course, especially from a biodiversity point of view, a real tragedy, because these large valley rivers, with their high diversity and in soils, in microhabitats, in topography, traditionally have been the home, the refuges for a surprisingly large number of grasshoppers, especially of califera. And I just show one short slide of grasshoppers which still are somewhere typical or have been typical for such river stretches. But as you can see here, most of these species shown on this slide are not exclusively bound on river edges, but also inhabit even artificial habitats like gravel pits or so on. And at least on an international scale, as you can see here with the status on the European 28 states red list, all these species are still assessed as of no or least concern. As soon as we switch to the highly specialized species, which are more or less exclusively bound to bare grounds on alpine rivers, the situation is totally different. As you can see here in the inlets here and the injured, and I just focus now on 4 species which belong to this group. This is first Epachromis tergestinus pontus, secondly Proedemella tuberculata, 2 larger very mobile species, and 2 small species, wingless, flightless species, the Alpine groundhopper, Tetrix turkey, and Cortipus bullus. Even a short glimpse on the maps, distribution maps of the 2 central countries of the Alps show or give a good, or may I say a bad feeling about the crisis. This is the Switzerland, of course, and you see, please note that Epachromius and tuberculata are extinct in the Switzerland, and the other smaller species are critically endangered in the national red list, and the populations are declining. And the situation is not much better in Austria and in the bordering countries to the north and to the south, Bavaria, Germany, and Italy, especially South Tyrol. You can see that Epachromius is extinct in Germany, in Italy, in Austria since decades. That Pruridimella tuberculata can be found in a very small area in the northern limestone Alps of Tyrol and the adjoining Bavaria, and that also the 2 other species are decreasing and at least endangered. So if we summarize these findings and use the red list status of the 7 countries of the Alps and the information which is available about the population trends, we end up with this rather frustrating table. Please note that Epachromius can only be found in the southwestern parts of the Franz Alps, in the Durance systems, but its natural populations but is critically endangered there. I assessed the AOO, the actual area of occurrence of Pruridimella, which in my opinion is less than 50 square kilometers. And please also note that Cortipus bullus is not only extinct in Slovenia, but is also critically endangered in most other parts. It might be that the situation of Tetrix turkey is slightly better because, of course, this is a very cryptic species which might have been overlooked sometimes. But overall, if you focus on the bottom row, it's really a critical situation with decreasing populations of all species, so that conservation measures, of course, are urgently needed. But if we go to conservation, we have to need more information about the threats, and of course, overall and ultimately, the long-lasting degradation of alpine rivers due to hundreds of years of regulation measures. The hydropower plants, which are built everywhere on the Alps, are the basic problems, habitat destruction, you could say. More locally, of course, there are other problems, other impacts, especially gravel mining, and in the last decades, maybe the ongoing and the increasing recreational use of riverbanks. Just to highlight once more the situation in the Alps, I just give this map of the mentioned WF study without any need of a comment, I think, which shows the hydropower plants and other barriers on the larger rivers of the Alps. I just would like to mention that even at the date when this study was published in 2014, by no means this map has been complete. And the special problem is that many of these river runoff plants are operated with hydropeaking, which is shown here for the Swiss and Austrian Alps in these 2 maps. Hydropeaking means that an artificial flood is rushes down the riverbed, maybe even daily, downstream the plant. And of course, you can imagine that these unnatural dynamic is disastrous, especially for the egg development, for the larvae, but also for more or less immobile insects or also fish, and especially, of course, our river grasshoppers as well. A good example is shown here. This is the Upper Inn river in the Swiss-Austrian border, where all these 4 species were present with good populations until the 1960s. And then at the Swiss part of the Inn river, there was a hydropower plant built with hydropeaking. And since then, albeit we had really intensive searching for grasshoppers at this river stretches for the last 25 years, and albeit the fact that there are many gravel fields which look quite appropriate for the species. There was no species ever found there in the meantime. In the meantime, there is a new river plant project in construction at the Swiss border, which is aimed to reduce the hydropeaking and which is supplemented by some river restoration programs like here. Looks very beautiful. But this doesn't solve another more general problem, that in the meantime, many of these river stretches, which formerly have been inhabited by these specialists, are so isolated at the moment that it's, in my opinion, very unlikely that a natural resettlement will happen. And another problem is that the few river stretches where still vital populations are preserved, there is another severe problem. This is the ongoing and very, very quick succession by plants, especially by willows, due to the lowering of the water table, due to different measures along the river. And Michael Reich, this is a very beautiful example at the Bavarian Upper Isar River. This is maybe a 1.5 kilometer long stretch. And you can see the rapid succession which goes on here in only 2 decades due to less peak floods and less bed load and less inundation areas in the consequence of some river measures. And Pruridimella tuberculata, which was the main focus of Michael Reich's studies, this was the reaction. You can see that in the late 90s, the red areas, many of these riverbanks had good populations, and nowadays it looks like this. And the same problem, this rapid succession, is also a problem in the second stronghold of this beautiful grasshopper in the Natura 2000 area in the Lech Valley, where about 25 kilometers along the 60-kilometer long stretch are still inhabited by Pruridimella tuberculata, Tetrix turci, and Cotipus pullus, albeit not continuously. But we observe also there a rapid succession here due to the lowering of water table and of the building of hydrological barrages along the river and gravel traps in the side valleys. And if this goes on and on and on, in the end, if any grasshopper is present at these formerly pair grounds, it's maybe Cotipus prunius, but no other species. Most of these negative measures have ended at the end of the 20th century. However, the consequences are still ongoing and influencing the river ecosystems and the grasshoppers. And this is despite the fact that the severe programs were going on here in the last 20 years by 2 live Lech river programs, and there was really they tried hard to regain a natural dynamic in the river course. A very good example is here shown on, along this stretch in the midsection of the Lech, which is about 3.5 kilometers long. And in the first live Lech program, which ran from 2001 to 2007, so already more than 15 years ago, one has reduced these short grounds here on both sides, and there was a large parage which was removed, and as a consequence, as you can see here, the riverbed widened, and we had a lot of new gravel banks, especially in the midstream area. However, we made intensive monitorings during the last 5 years or so, and we couldn't see any effect on the 3 species which are still present at this stretch, but mostly they are declining, they continue to decline, and there was no new site occupied along this river stretch. So I think and I fear that beautiful pictures alone will be not enough to save our specialized river species. It is self-understanding, self-evident that, of course, there must be a strict conservation of the last really natural wild river stretches, and it's an admirable effort, a very expensive effort, by the way, to renaturate such stretches. But I think that the old saying, they never come back, at least needs the addition, they never come back alone. I think it will be necessary to supplement these conservation measures with directed supplementation or even reintroduction of some species because of the high isolation. There's at least one promising example from the Alps in the Tyrone Valley in the Valais, where Epicromius went extinct in 1989. And then in 2004, a few nymphs have been released from this southwestern France population. And even nowadays, the species is there and has reoccupied a few new sites along the Rhone. So if you are a positive man or woman, you can say there's always reason for hope. No, I'm a pessimist. No, I'm a realist. I think I'm a realist. I fear that the dark clouds over the Alpine rivers will not fade away in the next few decades. On the contrary, I really fear that the clouds will become darker in the near future for reasons I'm glad to explain if there is a need for. Thank you very much.
Thank you, Armin. I'm an optimist.
Nice for you.
Otherwise, I couldn't work there. Need to be optimistic and do something against it. Yeah. Are there questions? Yeah, Filippo.
Thank you. Filippo Buzzetti. Do you know how many nymphs of Epicromius telgestinus has been released?
37.
37, okay. I didn't notice the number.
Yeah, at least as far as I know, it might be that afterwards there were a few other released, but this one is the published information.
There's, of course, a positive relationship between the number of individuals you release and the success of the introduction. And there is a good study which has shown, I think, that the threshold is between 100 and 200 somewhere. So if you have more than 100, you have a good chance to be successful. Of course, the others sometimes are also successful, but yeah, of course, if you just release a few individuals, then it's difficult.
Yeah, Armin, one question. You're a realist. The question I have, what can we, the audience, the researchers, the biologists, the conservationists, you think still do to maybe get a piece of blue sky in those dark clouds?
Don't buy and drive an e-car. Don't buy and drive an electric car. This would be a nice thing to do. I mean, the most reason for my pessimism is climate change, but not in the way you would think, but the other way around. It are the actions against the climate warming, which I fear, especially when it comes to river, because it's now clearly visible that hundreds of projects are planned or are even in construction of new hydropower plants under the, a bit, if you ask me, short-sighted and stupid slogan of the sustainability, environmentally friendliness, and CO2 neutrality of electric or hydropower energy. And we have a real strong conflict of nature conservation and climate activities going on, and these conflicts will harden, I think.
Okay, thank you. Well, damming is indeed a big problem, and not only in Europe. Yeah, large parts of the Himalaya are meanwhile dammed, and I'm much more concerned about this because biodiversity is a different thing. Yeah, maybe that's also not the solution. Okay, anyway, we come to the next presentation. Yeah, thank you, Armin, again. Next presentation will be by Jens Schirmer, who has been working a lot on urbanization effects on Orthoptera, and I think his presentation today is also on the question of how urbanization affects morphology and behavior of grasshoppers.
Thank you very much. Welcome.
So this is not me, as you can see. This is Florian Rech. Florian studied in his bachelor thesis the effects of urbanization on traits of Quadrupus bigutulus, behavioral traits, and the morphology. Unfortunately, he cannot attend the meeting, so I'm here to present his results. Is it this one? Yes. So 4 years ago, in this very nice Smolenice castle, I had a presentation about urbanization and grasshoppers. So urbanization, as you all know, is a major factor for global change. It's increasing worldwide. So we have much areas which are increasingly urbanized. And 4 years ago, I could show that increasing urbanization has a strong negative effect on Orthoptera diversity. So this was a study done in Berlin. So a decrease in species richness and also functional diversity. And in the recent years, several other studies showed very similar patterns. And also a recent global meta-analysis showed predominantly negative effects of urbanization on arthropod diversity. So these urbanization biodiversity relationships are relatively well studied and understood. But what is about species traits or the behavior of specific species? And there are very famous examples that species or animals can change their traits or adapt to the to urban habitats. So all of you know, I guess that, and this is the most famous example, birds can change their traits in terms of communication behavior. So it could have been shown that some species, like here the great tit, can adjust their songs and sing at higher frequencies to avoid signal degradation in noisy urban habitats. The same was also shown for grasshoppers 10 years ago by Lampe et al. So again, an adaptation in the communication behavior, also for insects. But potentially many other traits are affected due to urbanization. So for example, in terms of morphology, so some studies showed that individuals from urban sites increased their size. Here an example for a grasshopper where the wings were longer in individuals coming from an urban site compared to individuals from a rural site. And also other behavior traits might be affected, for example, the exploratory behavior, so measure of boldness more or less. Here a study from Schitt et al. From Germany for carabid beetles. This is Carabus nemoralis, a very nice one. And they could show that individuals from urban sites showed a much higher exploratory behavior than individuals from rural sites. So Florian was therefore interested if urbanization can also affect behavioral and morphological traits of Quadrupus bigutulus, so as you know, a very common species in Central Europe. And in particular, he was interested if urbanization affects the size of individuals coming from different sites regarding the urbanization level, and also in terms of the fluctuating asymmetry. Fluctuating asymmetry means small deviations from perfect symmetry in two body parts. So if we have a high fluctuating asymmetry, so a high deviation in these two body parts, this means that the individual was exposed to environmental stress during the development. So this is the basic assumption. Further, he tested the boldness. So if individuals differ in their boldness behavior, and finally also in their walking activity behavior. And we were also interested if there were differences between females and males. And therefore, Florian sampled Quadrupus bigutulus in meadows around Karlsruhe. This is a larger city in southwestern Germany. And he sampled meadows with different urbanization level. So this was measured based on the percentage of impervious surface in the surrounding. And we had then 5 meadows with low urbanization level, so less than 10% impervious surface in the surrounding, 5 meadows with medium, and 5 meadows with high urbanization level. And then he collected more or less 10 individuals per site and this yielded then in 92 females and 57 males. And then he brought the individuals into the lab, and they were caged in mesocosms, reared at standardized conditions in climate chambers, and they had 24 hours time for acclimatization before and between the experiments. So I will explain the experiments in the next slides. And after the experiments, he did some morphological measurements. So he measured the length of the femur and of the forewings, and based on this, he was also able then to quantify the fluctuating asymmetry, so the difference between the lengths in the two body parts. So now the experiment, the first one regarding boldness, this was a very simple idea from Florian. So the basic assumption was that organisms show some specific behavior when they are in a risky situation. And some grasshoppers seems to defend themselves by playing dead or show tonic immobility. And he had the idea to simulate the risky situation by grabbing simply the grasshoppers at the hind legs. And then he measured simply the time the individual played dead. I'm not sure if this is really a good idea, but we will see later on. So I said, let's give it a try. Okay, maybe it works. He repeated this 3 times per individual, and then we took the mean. So this was really a very simple experiment. The second one was much more complicated. So this deals with the walking behavior or the walking activity. So we conducted, or he conducted, an open field test to observe the walking activity in an unknown area for the individual. And we used video recording, so video tracking of the individuals. So he constructed an artificial arena, as you can see on the right, and we had a camera on top. And then individuals were recorded for 10 minutes, and we had this EcoVision software, which is a video tracking software, which can automatically track the individual walking behavior. And based on this software, we then calculated, or again he calculated 3 different parameters. So the first one is simply the distance the individual traveled within these 10 minutes. So this is then expressed in centimeters. The second one is the mobility, and this means the percentage of the time the individual was moving, but not the whole individual is moving. This also includes only the movement of some body parts. So for example, if they move the antenna, or if they just turn around or move a leg, this was also then tracked and called mobility. And finally, the time the whole individual moves, so really the time the individual moves in a directed manner. And this was then expressed in seconds. So these were the 3 parameters indicating the walking activity of the individuals. Okay, and now the results. I have only 3 slides for the results. First of all, the size and the fluctuating asymmetry. On the left, you can see the size. Of course, we have difference between the females and the males. But as you can see, there was no effect of urbanization. So we tested in the models always urbanization, sex, and the interaction to disentangle if the effect of urbanization differs between females and males. So urbanization, in our case, did not affect the size of Cotepus bigotulus. But as you can see on the right, we found a strong effect on the fluctuating asymmetry for both the asymmetry of the femur and also for the forewings. So especially the individuals from the highly urbanized areas, here always indicated in red, showed a much higher fluctuating asymmetry than the individuals from the low urbanized meadows here in green. For the wing asymmetry, also at least as a trend, urbanization and sex, the interaction, at least as a trend, was significant. And here you can see that this wing asymmetry was especially pronounced in the males. So we had a higher fluctuating symmetry in the males than in the females. Then the boldness, also unsure if this really was a sound experiment, but nevertheless the results showed a clear pattern. Indeed, we found that individuals from the highly urbanized meadows showed a much shorter time playing dead. So the tonic immobility was lower in these individuals from the highly urbanized sites, indicating that these individuals are bolder than the individuals from the lower or medium urbanized sites. So at least something seems to go on there. And then finally, the walking activity. So on the left, the travel distance in centimeters. And here you can see no effect of urbanization, also no difference between females and males. Also the interaction was not significant. So the total distance seems to be not affected by urbanization. But in contrast, as you can see in the middle and on the right, there was a very strong significant effect of urbanization on the mobility, so the percentage the individual was active with the antenna or the other body parts. So here we had an increase of almost 30%. So the individuals were more or less 30% more active when they were from the highly urbanized meadows compared to the meadows from the lowest urbanization. And similar, the time moving, so in seconds here, also increased by more or less 24%. So again, strong increase in the movement time of the individuals. So, to conclude, Florian could show that urbanization had no effect on the size in our study of Cotybus bigutulus, but he found very strong effects on the fluctuating symmetry in both body parts. So this could indicate that individuals are exposed to environmental stress during the development. He could also show that urbanization favored the occurrence of bolder individuals, indicated by this simple experiment. And he could also show that urbanization has a strong effect on the walking activity. So individuals from highly urbanized meadows were much more mobile than from meadows with low urbanization. And this means that urbanization can not only affect biodiversity, but of course can affect morphological and behavioral traits of grasshoppers or insects maybe in general. And the fact that we found these differences in the traits between individuals from the different urbanized meadows, this could really indicate that Cotybus bigutulus show a high phenotypic plasticity, so it's very flexible, and this could indicate the success of the species or can explain why the species is also able to live even within highly urbanized areas. And that's it. Thank you.
Thank you, Jens. Very interesting presentation. There are there questions? There's one in the internet? Or?
Yes. There's one online question from Klaus Riede. Wouldn't it be more adequate to explain increased boldness by terms familiar to neuroethologists, such as habituation or arousal?
Maybe I didn't get the question where we were. Can you translate it? Yeah, into more easier words for me? I don't. Or maybe just repeat it, but I'm not sure what it exactly meant. Sorry.
Yeah, I also have to think about what exactly. Klaus, can you please write us exactly what you mean? Habituation. I mean, they were all raised in similar conditions.
Yeah, so I mean, they were all sampled at the same time. They were reared under standardized conditions. So I mean, the differences we observed should really come from the different habitats they were sampled from. Yeah.
Yeah.
I mean, the main problem in all these urbanization studies is how to define urbanization and the gradient. I mean, you have a rather small sample size and high standard deviations, and you made these observations not in the field, but in the laboratory. This would, of course, mean that all animals have some genetic basis for their behavior. So did you have a look on the history of your sites? Because many meadows which are now maybe really in the middle of an built-up area, maybe only a few years ago, could have been at the edge of the city or of the urbanized area, and this should, of course, be taken into account. Did you have a look on the history of your, I think, only 2 or 3 meadows from the so-called urbanized area, and also of the history of the rural or more or less median sites?
No, I have no idea about the age of the sites. Regarding your first remark, urbanization is in most studies calculated by using impervious surface, and this makes really sense because all other environmental stress caused by urbanization is associated with this amount of impervious surface. So I know very much urbanization studies, and if we have a high amount of impervious surface, this means we have high noise pollution, we have high light pollution, we have high atmospheric pollution. So I think this can really be a very effective way to express urbanization. Sure, this was a bachelor thesis, so the sample size is rather low, but I think 150 individuals is also quite okay. So from my point of view, I mean, 500 would be better, 1,000 even better, but I think it's more or less a solid basis.
Okay, I think we have to go on. Thank you very much again.
Presentation of this session, so by Sebastian König, who will talk on biotic harmonization of Orthoptera assemblages in the Hasberge, so in Bayern. And the title is already quite similar to Sophie's, so apparently the results are quite similar. So I'm very much looking forward.
Thank you. So, good afternoon also from my side. For those who don't know me, I'm Sebastian. I'm from the vicinity of Würzburg. This is a really nice region in north of Bavaria with nice species-rich calcareous grasslands, and I guess this is where I got fascinated by Orthoptera when I was a young child. A little bit to my background the last couple of years. I did a lot of field trips, also to mountain ecosystems like the Rila Mountains or the Pirine Mountains in Bulgaria, or even the Drakensberg mountain range in South Africa and Lesotho. But I spent most of the time in the European Alps, like mostly in the National Park Berchtesgaden in Germany. And also did empirical assessments there. I'd like to present you the results, or the first results, of 2 case studies, which I have to admit, both of the topics have been covered today, so this is why I don't have to talk very much about the method part, and maybe just show you a lot of cool photos of Orthoptera. It's even late in the evening now, yeah. The first one would be on feeding interactions along a microclimatic gradient. And therefore, we sampled, did botanical assessments of the vascular plant species and mosses on 40 study sites along a gradient of temperature close to the ground throughout Bavaria. And what we did on these sites is, besides the plant assessments, we also sampled or surveyed the Orthoptera communities. And on the other hand, we collected field observations of feeding interactions. only between Orthoptera and plants, not between Orthoptera and other insects. We did this for 5 years now, so gathered a lot of data, but as you might know, this field observations of feeding interactions can be really exhaustive and you need a lot of patience for that, so we decided to also use another method, which was also presented today, this collection of fecal samples. We did it almost in the same way. And then later on, laboratory workflow with DNA metabarcoding to estimate the diets of the Orthoptera species. Yeah, what we found there was an increase in species richness and abundance of Orthoptera with temperature, but also with plant species richness. The proportion of Gomphocerinae, or the grasshoppers, within Orthopteran assemblages was highest when the grass cover was highest, and it tended to decrease with temperature. Whereas the plant species richness peaked at intermediate temperatures. When we take a look at the diets, which I show here, derived from the metabarcoding data set, we can see, as expected, a phylogenetic clustering within the plant species that were eaten by different lineages of Orthoptera, with high proportions of algae and mosses, for example, in tetrachids, and high proportions of grasses in grasshoppers, with a few exceptions, of course. The picture changes when we go into Eutypodine or Tetigonids or Melanopline, where they feed on a much broader range of resource plants. Yeah, during these field surveys, we also observed some interesting sightings of Orthoptera populations, like this Cortypus populations in the Berchtesgaden Alps, which remind me a lot of Eisentraut's bowwing grasshopper. I have many pictures of them with me, so for those interested, we can maybe later on or tomorrow take a look at them. I guess they're some sort of hybrids or something. This is what an example network on site level would look like. You have the plant species, the plant phylogeny on top, and as rows the Orthoptera species which occurred on this study site. And what directly catches the eye is that there's a high proportion of grasses in the diets of the Gomphocerinae, which are the lower rows here. It differs for the Tetigonid species, which are in the upper part. So the first question we were asking us is if the specialization, if we can measure them in any way, is changing along this temperature gradient. And we try to do this by using this phylogenetic structure of specialization index, or on the same way the phylogenetic specialization index based on dietary phylogenetic relationships. And this is measured as the mean pairwise distances in the diets compared to a null model, which is based on the botanical assessments on the study sites. And what we see there is that on assemblage level, we find the highest specialization values in intermediate temperatures. And we looked at this also at species level a little bit for those species which occur on a larger range along this elevational gradient. And there was not so much change within species, so we think this might be explained by a turnover in species often, this pattern. Now leaving this spatial temperature gradient and going on to a temporal temperature gradient, this is a study very similar to the one Sophie presented before. We did a reassessment of around 200 study sites in north of Bavaria. And the results match very good with that what you presented. We also had wet mesic, ruderal, and dry grassland sites there. They were first sampled in 1988 and then reassessed in 2004. And now we did the reassessment in 2019, always using the same methodology. And all those people involved in the study were also sampling. Yeah, what we see is that temperatures were increasing over the study period, and the composition, so incidence-based of the Orthoptera assemblages, the compositional differences between these grassland types decrease over time, which is also an indication for this beta diversity decline. Looking at the species themselves, we could not detect some of the species which were formerly there on the study sites anymore, like the Cotopus vagans, the heath grasshopper, or the rattle grasshopper, for example. But those were formerly also rare on the study sites. Other for some of the formerly widespread species like Pseudocotopus montanus or very pronounced Omocestus viridulus, also the common green grasshopper, they lost a lot of their incidences in the study region, on the study sites, also on the wet meadows. On the other hand, we found strong increases for quite a number of species. One species was new for the study region, it was European tree cricket, Eucanthus, and most pronounced, the steppe grasshopper, Cotopus dosatus, also increased in this region of Germany, paralleled by Cotopus bigotulus and Cotopus mollis, as well as Stetophyma grossum or Oedipoda cerulescens. Yeah, looking at the traits, we had also an increase in alpha diversity, so the number of species at almost all of the study sites and in all habitat types, but we found that the differences between the habitat types decreased, and this is maybe due to the loss of specialists, both adapted to dry or to either dry or wet habitat types, and a parallel increase, a strong increase in general species, which also occur on the warmer microclimates, as indicated by the increasing macroclimatic preference. This is the measure from Thomas Wartmann's group, the Community Temperature Index. Good. As I also I'm quite hungry now, I just show you some impressions of my last field trips with some interesting species sightings. We also heard about Paracaloptinus today. This is, these are photos from Bulgaria. And here also from high altitude grasslands like Pseudodonotus viperi. Oh, here, this is also a nice photo of the speckled buzzing grasshopper, a pair. And let's hope that this Gomphoceribus found a matching female in the end, because he tried hard. Not successful. Thank you for your attention.
Thank you very much, Sebastian. Very nice end of this session. Are there questions from the audience? I think it's very interesting that your results match so well with ours, and we had some discussions during the coffee break also with Thomas, and who had this study in Austria, and I know that Thomas Wartmann also made similar studies in Münsterland and so on, and they all appear to go in the same direction, yeah, which means that they actually are quite a good basis also for future red list assessments, probably. There's a question by Szymon.
Yeah, Szymon Czyżewski. Super interesting. I thought that the decline, I think the highest decline was in Stenobotrus stigmaticus, right? Am I right?
Yeah, but it was only 2 occurrences before.
Ah, okay. But because I mean, in Poland, I think it's less in one region. I mean, it's not me doing the field work, but a colleague of us from the Atlas too. He's kind of doing similar thing, and he found like an extreme decline. And I think when you look, you can kind of put the hypothesis that it is really declining, and I was wondering if you have any hypothesis, but if it's 2 records, then maybe it's difficult to tell anything.
But it's interesting that also some of these dry specialist species are decreasing, and I think this can also be the result of microclimatic conditions which change in the habitats, like more plant growth or whatever, which changes microclimatic conditions.
Yeah, thank you.
Okay. Are there any other questions? If not, I end the session, but I hand over to Luc.
Thank you very much.
Yeah, so this is the end of the sessions for today. We have scheduled for those who have indicated there is, again, this evening you can join us at Luigi, the same restaurant we were yesterday evening in the center of town, as from 7 o'clock in the evening. We have dinner and there will be a pub quiz. So for those of you who are not a member of the Deutsche Gesellschaft für Orthoptologie, the rest of the day you're off until 7 o'clock. Those who are a member of the DGFO, please stay here because we will have a meeting of the German Orthoptological Society. Okay? So see you for those of us who come to the restaurant, see you later. Otherwise, we see you tomorrow at 9 o'clock again in this room. Okay. No, it's
Okay. Es kam gerade noch der Wunsch auf, eben eine ganz kurze Pause zu machen. In 5 Minuten würden wir starten mit der Mitgliederversammlung. Ja, wir warten gerade noch, wie viel im Chat dabei sind, wie viele Mitglieder. Da guckt Luc gerade noch, dass das ja, mit dem Chat funktioniert. Gut, wunderbar. Ja, es sind über 20 Mitglieder online dabei und hier im Raum sind wir ja auch über 15 auf jeden Fall. Und es müssen 5% der Mitglieder müssen bei einer Mitgliederversammlung immer anwesend sein, damit wir beschlussfähig sind. Und ja, die Mitgliederversammlung ist ja, wie immer eigentlich in der Articulata angekündigt worden. Das muss 6 Wochen im Minimum vor der Tagung erfolgen. Die Articulata ist, glaube ich, Ende Januar verschickt worden, also das müsste terminlich passen. Und ja, kommen wir vielleicht erstmal zur Tagesordnung heute, wie sie auch in der Articulata angekündigt worden ist. Da hätten wir zunächst einmal den Bericht des Vorstandes und ja, ich würde kurz beginnen mit einem kleinen Teil, worüber ich berichten kann, was sich in den zurückliegenden Jahren in der DGFO getan hat. Das ist eine relativ lange Zeitdauer, der wir uns jetzt nicht gesehen haben, Corona-bedingt. Und auch hier die Tagung, das ist ja zweimal verschoben worden wegen Corona. Ja, gut, so ist es halt. Und zunächst einmal vielleicht zum Thema Forschungsförderung. Die Forschungsförderung, die ist ja durch Peter Detzel ins Leben gerufen worden. Und wir haben insgesamt bislang jetzt ca. 15 Projekte gefördert und seit 2018 war es so, dass wir ja auch wieder eine ganze Reihe von Projekten gefördert haben. Und ja, das sind erstmal die ersten 6, die wir da gefördert haben. Und dann gibt es noch 2 weitere, die hier auftauchen. Also 8 Projekte haben wir in diesem Zeitraum gefördert. Und das werden wir nachher auch sehen beim Kassenbericht. Das sind ganz erkleckliche Finanzsummen, die da auch geflossen sind. Das Maximum, was gefördert werden kann bei einem Antrag, sind 2.500 €. Ja, wenn es viele Anträge gab in einem Jahr, dann haben wir das auch ein bisschen reduziert, dass also nicht voll die 2.500 € ausgeschöpft werden konnten. Aber bislang war es, glaube ich, so, dass alle Anträge auch bezuschusst worden sind. Also es ist keine abgelehnt worden. Die waren aus unserer Sicht von der Qualität her alle gut und ansprechend. Und ja, und nichtsdestotrotz ist es so, das hatte Peter Detzel ja auch angeregt, die DGFO ist jetzt nicht gerade reich, aber das werden wir auch gleich sehen beim Kassenbericht. Wir sind finanziell eigentlich ganz gut aufgestellt. Wenn ja, jemand Interesse hat, explizit für die Forschungsförderung auch zu spenden, sehr gerne, nur zu. Also das ja, ist sehr zu begrüßen, aber wir sind eigentlich gerade finanziell ganz gut aufgestellt. Dann eine ganz massive Änderung in der DGFO, das ist die Schriftführung der Articulata. Georg Weber hat das ja, mehr als 15 Jahre gemacht. Er war die prägende Person eigentlich der DGFO in der Zeit, muss man sagen. Er wollte eigentlich auch kommen, aber sein Flug ist kurzfristig gecancelt worden, sodass er nicht kommen konnte. Und er hat im letzten Jahr die Geschäfte an Jens Schirmel übergeben, der bislang Schriftführer war in der DGFO. Und Jens hat jetzt eben schon das letzte Articulata-Heft gemacht. Und ja, in diesem Rahmen möchte ich mich nochmal ganz, ganz herzlich im Namen der DGFO bei Georg bedanken für diesen Einsatz. Er war wirklich die prägende Person in der DGFO in dieser Zeit. Er hat von der DGFO oder im Namen des Vorstandes ein größeres Präsent vor mehreren Wochen bekommen. Und ja, eigentlich wollten wir ihm hier auch noch was überreichen. Das ist jetzt auch auf dem Postweg an ihn gegangen. Also leider haben wir hier nichts, weil er auch nicht da ist. Aber ja, wir haben zumindest versucht, uns gebührend bei ihm zu bedanken. Und ja, da nochmal ganz herzlichen Dank. Er hat wirklich eine Epoche geprägt, das muss man so sagen. Aber zu diesem Thema Articulata und Schriftführung, da wird Jens, glaube ich, gleich dann noch ein wenig was dazu sagen. Ein weiteres Thema ist die rote Liste der Heuschrecken Deutschlands. Da sind wir dran, sie zu bearbeiten. Es gibt eine Endabstimmung mit allen Landeskoordinatoren in Deutschland. Also der Entwurf steht. Jetzt geht es darum, die Texte zu verfassen. Also sind über 20 Heuschreckenkundler, die mitgearbeitet haben an diesem Entwurf. Also für jedes Bundesland wenigstens ein Experte, darüber hinaus noch 2, 3 weitere Personen. Und wir haben es gemacht und diese rote Liste wird, ja, zumindest die Endfassung sollte irgendwann in diesem Jahr dann stehen, die textliche Endfassung. Wann sie erscheinen wird, kann ich noch nicht sagen. Das läuft in Kooperation mit dem Bundesamt für Naturschutz. Ja, da weiß ich nicht, wie der Druck vonstatten gehen wird. Aber zumindest gibt es diese Liste und es hat, wie wir heute eigentlich auch schon gesehen haben, wir haben massive Veränderungen der Heuschreckenfauna in Mitteleuropa und auch die rote Liste sieht ganz anders aus, als die letzte aussah. Bestimmte Arten, die früher auf der roten Liste standen, sind runtergekommen und eine ganze Reihe von anderen Arten sind jetzt raufgekommen. Insbesondere Arten, die ja, auf extensiv genutzte Lebensräume angewiesen sind und eine geringe Mobilität aufweisen. Da haben wir also eine ganze Reihe von Arten, die heute nicht oder die ja, jetzt auf die rote Liste draufgekommen sind. Gut, das wäre vielleicht erstmal von mir bis hierhin der erste Teil. Da ist die Frage, gibt es aus dem Auditorium Fragen oder aus dem Chat zu diesen Punkten? Aus dem Chat erstmal nicht. Gut, ich würde nachher nochmal den Kassenbericht auch vorstellen. Dem hat Edgar Bayerl, konnte leider nicht kommen und hat mir die Informationen übermittelt. Die werde ich gleich nochmal vorstellen. Aber erstmal möchte ich übergeben an Josef, dass Josef aus seiner Arbeit berichtet.
Ja, danke Thomas. So viel gibt es das denn vom stellvertretenden Vorsitz auch nicht zu berichten. Vielleicht nochmal ergänzend zur roten Liste beim Bundesamt für Naturschutz. Das macht ja das Rote Liste-Zentrum da. Und ich bin mir da relativ sicher, weil ich die auch noch vor einigen Monaten besucht habe in meiner Tätigkeit jetzt im Bundesumweltministerium, dass das zügig gehen wird. Also wenn die da vorliegt, wird der Ablauf sehr schnell sein. Am Ende müssen wir da nämlich auch den Haken drunter tun. Deswegen ist es gut, dass ich nicht mitgearbeitet habe, wenn ich sie dann begutachte nochmal am Ende. Genau. Ansonsten bei mir ist ja die ganze Sammlung der Articulata-Hefte. Wir haben viel Platz, es sind viele Bücher da. Es kommen auch immer noch Fragen, also nicht Fragen, sondern Wünsche, dass auch neue Mitglieder wirklich die gesamten Hefte auch haptisch haben wollen. Also wirklich, ne, kriegen sie dann auch gegen Spende weiter. Ich habe das jetzt nicht überprüft, was da an Spenden eingegangen ist, aber da dürften einige Mittel da sein. Das läuft auch weiter. Und irgendwann müssen wir uns allerdings dann mal überlegen, ob wir dann doch das Altpapier mal auffüllen, weil von einigen Exemplaren gibt es große Mengen noch, von anderen eben nicht. Also das ist das eine. Das andere sind kleinere Anfragen, die dann zu bearbeiten sind. Natürlich auch die gemeinsamen Beschlüsse im Vorstand. Das läuft, das muss ich auch mal sagen, dann Richtung Thomas, weil Eigenlob stinkt ja. Also Thomas macht das richtig gut, der hält den Verein wirklich zusammen. Da kann man als stellvertretender Vorsitzender sich beruhigt zum Mittagsschlaf legen und weiß eben, dass es bei Thomas auch in guten Händen ist. Es läuft alles sehr einvernehmlich, ne, sehr gut, mit wenig Aufwand, muss man auch mal sagen, in der Gesellschaft. Aber das, was wir uns vornehmen ja auch, was unsere Agenda ist, ich glaube, da brauchen wir uns nicht mit zu verstecken. Das vielleicht von meiner Seite.
Noch Fragen an Josef aus dem Chat, da irgendwas? Gut, dann würde ich übergeben an Jens. Ja, ich sag Bescheid.
In welcher Funktion rede ich jetzt als erstes?
Das kannst du dir überlegen.
Dann bin ich jetzt Schriftführer.
Ja, genau.
Na, das ist dann aber nicht das Richtige. Egal. Dann fange ich an als Schriftleiter. Genau. Das habe ich ja jetzt, wie gerade schon von Thomas angekündigt, letztes Jahr übernommen. Ich möchte mich auch nochmal ganz herzlich bei Georg bedanken, mit dem ich natürlich ein paar Mal telefoniert habe, mich auch auf ein Bier getroffen habe letztes Jahr in Franken. Genau, der mich dann natürlich sehr unterstützt hat. Ich möchte jetzt ganz kurz die letzten beiden Hefte vielleicht nochmal hervorheben. Das heißt, Band 35 hat ja noch der Georg gemacht, Band 36 habe dann ich übernommen. Wir haben derzeit eine Auflage von 570 Stück. Wir haben die letzten beiden Bände, wie man sieht, die waren extrem dick. Das hat Georg, glaube ich, auch geschrieben. Das waren die dicksten Bände, zumindest Band 35 mit 200 Seiten, die es jemals gab. Ich habe das jetzt mal für die letzten Jahre aufgelistet. Das heißt, wir haben den Cut nach Band 27. Davor hatten wir mal 2 Hefte pro Jahr, seitdem 1 Heft pro Jahr. Und wir hatten mal so eine leichte Durststrecke bei Band 28, 29, 30, wo wir nur so 120, 150 Seiten, 140 hatten. Ich glaube, wir haben uns aber wieder erholt. Also wir sind jetzt eigentlich auf einem ziemlich guten Niveau wieder. Da wäre fast schon die Frage, ob man wieder auf 2 Bände gehen möchte. Können wir vielleicht am Ende nochmal drüber sprechen. Also das heißt, der Eingang an Manuskripten ist, glaube ich, in den letzten Jahren ganz positiv zu bewerten. Genau. Nur noch mal ganz kurz, weil es mir auch wichtig ist, der Anteil deutsch- und englischsprachig, das gab ja schon ein paar Mal Diskussionen darüber, einfach mal im Verlauf dargestellt. Also eben englische Publikationen nehmen eigentlich immer schon eine wichtige Stellung ein. Und genau, der Verlauf ist jetzt hier mal so ein bisschen zu sehen. Ich denke, wir haben immer so einen Anteil von 20% grob, was ich denke auch sehr ordentlich ist. Und das kann von mir aus eben gerne so bleiben. Und gerade diese Bände 28, 29, 30, wo wir relativ wenige Eingänge hatten an Manuskripten, da war eben besonders wichtig, dass wir dann die internationalen Artikel auch hatten, damit das Heft überhaupt voll wird. Dann gibt es Änderungen im wissenschaftlichen Beirat. Seit letztem Jahr ist Martin Husemann mit dabei und auch Axel Hochkirch dankenswerterweise. Ausgeschieden ist Michael Wallaschek in diesem Jahr. Das heißt, auch hier haben wir eine kleine Erneuerung, was mich auch sehr freut. Genau, da sind wir, glaube ich, nach wie vor sehr gut aufgestellt und haben auch beide schon begutachtet für die letzte Articulata, waren ganz eifrig. Das hat gut funktioniert. Genau, das haben wir wahrscheinlich alle gesehen. Jetzt habe ich es übernommen, auch leichte Veränderungen am Layout gemacht, ein bisschen Autorenrichtlinien verändert. Genau, es war jetzt, glaube ich, 20 Jahre lang das gleiche Design. Deswegen haben wir gedacht, dass man vielleicht ein bisschen mal was ändern kann. Genau, das nur noch mal ganz kurz zum Merken. Und jetzt habe ich ein paar Dinge, die mir so jetzt in den letzten Wochen, Monaten bei der Arbeit in den Kopf gekommen sind, was ich einfach vielleicht mal zur Diskussion stellen möchte oder was ich mich selber einfach gefragt habe. Ich weiß nicht, ob es das schon mal gab, sowas wie einen Redaktionsschluss. Also ich habe jetzt, und auch Georg hat es mir erzählt, die Erfahrung gemacht, dass natürlich fast alle Manuskripte kurz vor knapp erscheinen oder bei uns reingehen. Das heißt, irgendwie ab Oktober kriegt man dann noch auf die Schnelle ganz viele Manuskripte, die unbedingt noch in das letzte Heft rein sollen. Das ist natürlich schön. Ich fände es perspektivisch natürlich aber auch sehr schön, wenn die Articulata wieder in dem gleichen Jahr erscheint. Das ist ja in den letzten Malen, hat das nicht so gut funktioniert. Dann erschien sie erst im Januar, ich glaube einmal vielleicht sogar erst im Februar. Das heißt, ich würde das schon versuchen, dass die wirklich auch zu Weihnachten auf dem Tisch liegt. Und da ist es natürlich schwierig, wenn ich bis November oder so warte mit den Manuskripteingängen. Entweder man macht mehr Werbung, dass die Leute wirklich frühzeitig einschicken, oder man könnte eben an sowas wie einen Redaktionsschluss denken. Ich habe da aber auch keine Erfahrung. Ich weiß nicht, ob es eine gute Idee ist, ob es schon mal gab. Vielleicht hat jemand anders Erfahrung dazu, würde ich gerne einfach mal zur Diskussion stellen. Dann, genau, rein von der Seitenanzahl könnte man theoretisch auch wieder auf 2 Bände pro Jahr gehen. Das würde ich selber ungern machen, weil es einfach viel mehr Arbeit für mich ist und auch mehr Kosten natürlich mit Versand und so. Genau, also von mir aus lassen wir das sehr gerne bei einem Band pro Jahr. Ich weiß nicht, wie da sonst die Meinung ist, ob jemand oder ob die Mehrheit lieber wieder 2 Hefte hätte pro Jahr, dann könnte man sich das ja noch überlegen. Dann kriege ich relativ häufig Anfragen nach der Verfügbarkeit von PDFs. Wir haben die PDFs ja auf der Homepage hochgeladen, allerdings nicht so wahnsinnig aktuell. Ich glaube, was habe ich geschrieben? Genau, bis 2015 kriegt man die PDFs, alle danach nicht. Das hatten wir auch, glaube ich, schon ein paar Mal angesprochen. Innerhalb der letzten 2 Tage?
Ja, also gut, aber zumindest ist das, wenn es noch nicht so sein sollte, ist das auf jeden Fall klar das erklärte Ziel. Ja. Das werden wir auch schnell.
Also das erklärte Ziel ist, dass bis auf der letzte Band alle PDFs da. Okay.
Auch auf einer Mitgliederversammlung mal genauso beschlossen.
Okay.
Dass wir gesagt haben,
Ach so. Ja, dann genau. Wenn die Vorgabe ist, oder laut Thomas Fahrtmann ist die Vorgabe, dass quasi bis auf die letzten beiden Bände alles als PDF verfügbar sein soll. Das ist jetzt ist jetzt nicht ganz aktuell gewesen, dann sollten wir das einfach machen. Die Frage ist auch, ob ich die PDFs trotzdem rausgeben darf auf Anfrage, auch ganze Hefte. Das ist jetzt zum Beispiel auch eine häufige Anfrage, ob man nicht jetzt Josef nach dem haptischen Heft fragen muss oder ob man nicht das PDF direkt haben. Habe ich persönlich nichts dagegen. Ich weiß nicht, wie da die Vorgaben sind. Also ich würde jederzeit die PDFs rausgeben, auch ganze Hefte habe ich persönlich kein Problem mit.
Also wir können das aus meiner Sicht auch gerne so machen, dass alle Hefte online stehen. Ne? Das ist einfach eine Frage, was wollen die Mitglieder? Ne?
Genau, also das könnte man natürlich auch überlegen, dass man direkt quasi ein Open Access Journal hat. Das hatten wir auch schon mal diskutiert. Ich glaube, damals wurde die Kritik geäußert oder die Gefahr, dass dann vielleicht Leute natürlich irgendwie austreten und dann das Heft nicht mehr zugeschickt bekommen, der Beitrag fehlt und die Leute gucken sich nur das PDF an.
Ja, vielleicht sollen wir sonst einfach so ein kleines Votum mal abfragen. Also gibt es irgendjemand, der dagegen wäre? Im Chat ist da was? Okay. Ja. Ja.
Ich meine, es soll diesen Fragen sicherlich gerechtfertigte unterschiedliche Ansichten. Ich glaube zum Beispiel, ein DOI ist absolut wichtig und richtig. Wenn man 2 Bände einführt, dann hat man natürlich grundsätzlich wahrscheinlich mehr Manuskripteinläufe, weil der Autor einigermaßen sicher ist, dass sein Beitrag einigermaßen rasch publiziert wird. Und dann könnte man eventuell eher auf eine Online-Version gehen. Man würde dann natürlich auch sinnvollerweise einen Redaktionsschluss einführen für Heft 1, für Heft 2. Ich weiß, es ist natürlich mehr Arbeit, aber es würde mit sicherlich also die Publikationsmotivation, könnte ich mir vorstellen, manche Autoren verbessern. Wenn ich sage, okay, ich reiche bis zum Redaktionsschluss meinetwegen Ende März für das erste Heft ein und weiß dann vielleicht, oder das kommt zumindest im zweiten Heft noch heuer raus. Nicht? Also da, was übrigens diese Strategie betrifft, dass man alles sofort online stellt, da würde ich vorsichtig sein, weil auch von der Altersstruktur der Mitglieder möglicherweise das nicht unbedingt bei allen extrem freundlich aufgenommen wird, wenn man das Heft nicht mehr haptisch sozusagen genießen kann. Ne?
Also, ja, wir hätten beides. Ne? Wir hätten eben die Druckversion, wir hätten die PDF-Version. Vielleicht, wenn wir erst nochmal wieder bei dem ersten Thema bleiben, alle PDFs direkt online stellen oder nicht, gibt es dazu noch Wortmeldungen?
Ist das erste Mal, dass ich hier dabei bin. Vielleicht ist es auch gut mal zu fragen, wie the World Orthoptera Society es macht. Die haben jetzt alles online und da kann man fragen, verlieren die Mitglieder? Dann weiß man auch, ob da eine Gefahr ist, weil ich denke, das ist wichtig, online is the future. So, ich denke, dass wenn man morgen etwas finden will und man kann das online finden, dass das gut für den Autor ist. Es ist gut für dieser Verein, denke ich. Und ich wäre da, Open Access, I think, is the future.
Noch weitere Wortmeldungen dazu? Können wir das so auffassen als ein Votum dafür, dass wir die PDFs online stellen? Okay, gut.
Genau, das finde ich super. Eben, die DOI wurde jetzt auch schon gerade erwähnt. Das wäre was, wenn wir es jetzt rasch online stellen, dann macht es natürlich Sinn, die direkt auch mit anzugeben. Das finde ich, das ist eben auch die Zukunft, dass man direkt den Artikel mit der DOI findet. Ich habe mich kurz ein bisschen schlau gemacht, mit ein bisschen Kosten verbunden, aber ich glaube, das hält sich in Grenzen. Das sind einmalig 300, 400 Euro, um diese Hauptnummer für das Journal zu bekommen, und dann hat man ein Paket, je nachdem wie viele Nummern man vergibt pro Ausgabe, aber das hält sich dann in Grenzen. Das heißt, man hat einmalige Kosten von ein paar hundert Euro und dann jedes Jahr, ich weiß es jetzt nicht, 80, 90, 100 Euro oder so. Also genau, ich persönlich fände das total super, viel einfacher aufzufinden. Wenn wir es jetzt direkt online stellen wollen, dann denke ich, können wir das direkt integrieren, oder? Okay. Ich glaube, mehr habe ich nicht. Nee, genau. Das sind die Kosten, aber das wollte ich gar nicht zeigen. 300 Euro und genau 80 Euro noch dazu. Ein Angebot. Genau, das war es eigentlich von mir. Ja, ich würde schon gern erstmal bei einem Heft bleiben, muss ich ganz ehrlich sagen. Und so viele Manuskripteingänge sind es dann auch nicht unbedingt. Das heißt, man hat immer die Ungewissheit, ob es wirklich reicht für ein Band oder nicht. Von dem her würde ich vielleicht die Entwicklung noch ein bisschen abwarten, wie es sich vielleicht in den nächsten 1, 2 Jahren ergibt. Wenn wir wirklich merken, es geht weiter bergauf, dann kann man das machen. Aber ich würde es, glaube ich, jetzt zumindest das nächste Jahr mal mindestens mit einem Band weitermachen.
Genau, ganz pragmatisch. Und dann hätte ich noch eine andere Frage. Gab es eigentlich noch mal irgendwelche Anfragen wegen Beiheften? Das letzte war ja, schlag mich tot, ich weiß es gar nicht, also das ist schon einige Jahre her, ne? Ja, genau.
Mich hat keiner erreicht. Bei Georg weiß ich jetzt natürlich nicht, aber mich hat bis jetzt noch keiner kontaktiert. Ja.
Möglich ist es. Auf der anderen Seite dieses Thema Monographien, oder auch jetzt, früher waren es ja ganz häufig Dissertationen, die da erschienen sind. Heute sind es alles kumulative Dissertationen. Ja.
Also wir können die Möglichkeit nach wie vor offen lassen, aber genau, ich glaube, da kommt nicht. Ja.
Also DOI machen wir und ein Heft pro Jahr weiter. Ja, auch keine Gegenmeinung, gerade im Chat. Gut, wunderbar, sehr schön. Dann können wir gerne zu deinem zweiten Punkt kommen.
Ja, jetzt bin ich Schriftführer. Noch. Genau, also die Mitgliederverwaltung. Wir haben derzeit 527 Mitglieder. Ganz aktuell, habe ich im Zug gestern noch gemacht. Und man sieht unten mal so ein bisschen den Verlauf, also Austritte und Eintritte in den letzten Jahren, seit 2014, seitdem ich das mache. Und genau, man sieht, das ist schon einfach stabil, ne? Also mal mehr, mal weniger. Insgesamt haben wir jetzt 11, also ein Plus von 11 Mitgliedern, also sehr stabil, genau. Das ist bei Austritten mit drin. Also genau, von dem her, glaube ich, gibt es einfach wenig Veränderung. Einfach mal, wie die Mitglieder sich auf die Länder verteilen. Gut, wir sind die Deutsche Gesellschaft für Orthopathologie, das spiegelt sich hier wieder, aber es sind trotzdem tatsächlich aus sehr vielen Ländern Mitglieder vorhanden. Genau, also schon einigermaßen international. Ich habe mir mal, das ist allerdings jetzt schon vom letzten Jahr, geguckt, wo denn in Deutschland die ganzen Mitglieder herkommen. Das ist ein bisschen vereinfacht, also bei der Postleitzahl die letzte Stelle weg oder die letzten beiden. Aber das fand ich irgendwie cool, weil also einfach Deutschland komplett mehr oder weniger gleichmäßig verteilt ist. Das fand ich irgendwie nett. Genau, also überall gibt es Heuschrecken-Nerds. Ja, stimmt, aber das kann vielleicht mit der Postleitzahl Genau, man sieht auch so ein paar Hotspots, ne? Also ein paar Städte, genau. Trier ist auch ganz gut vertreten, Münster, Osnabrück, genau. Genau, dann, wie teilt sich das auf in Institutionen, Männlein und Weiblein? Das ist natürlich ein bisschen, ich will nicht sagen schockierend, aber es ist auch derzeit gerade ziemlich genauso wahrscheinlich hier in der Sitzung verteilt. Also wir haben 76% männlich und nur 15% weiblich. Genau, das ist schon krass. Ja, siehst du. Ich habe es genau nicht nach Ländern aufgeschlüsselt. Das macht wahrscheinlich auch nur bei 2 Ländern Sinn. Aber genau, wir sind ja manche von uns optimistisch. Seit 2014 ist immerhin der Anteil bei 26% von den Damen. Also ein deutlicher Anstieg. Darf ich? Ja. Ist nicht ungewöhnlich, ist trotzdem nicht gut. Genau. Aber, genau. Ja. Ja, genau. Ich glaube, darf ich verraten, wie es weitergeht mit meinem Posten?
Kannst du gerne machen, ja.
Genau, weil also wir haben Nachwuchs dann gefunden, Nachfolger für mich. Das ist der Raimund Klatt, der ab sofort hoffentlich dann die Aufgabe übernehmen wird. Weil genau, Doppelfunktion brauche ich dann jetzt auch nicht noch weiter. Genau, also vielen Dank an Raimund, dass er das übernimmt. Ich werde ihn instruieren. Das genau, ist jetzt auch nicht die schlimmste Arbeit, also das kriegt er wohl hin. Genau, genau, das war's.
Ja, noch Fragen an Jens? Alles soweit geklärt. Gut, dann kommen wir zum Kassenbericht. Ja, Edgar konnte leider nicht kommen und ich werde versuchen, seinen Kassenbericht ein wenig vorzustellen. Bin aber, muss ich gestehen, nicht wirklich voll drin im Thema. Ja, genau, also prinzipiell sieht es bei uns ganz gut aus. Die Einnahmen überwiegen ganz klar die Ausgaben und wenn man weitergeht, dann sieht die Bilanz aktuell so aus. Wir haben etwa ein Guthaben von 16.977 € und da müsste man aber jetzt noch ca. 7.000 € abziehen. Das sind die Druckkosten von der letzten Articulata, aber wir sind trotzdem eigentlich ganz gut aufgestellt, ne? Also das sieht eigentlich so gut aus, haben keine Finanzsorgen und haben auch ein bisschen Puffer für die Forschungsförderung. Die Kassenprüfung haben Dominik Poniatowski und Daniel Elias übernommen. Und ja, Dominik konnte nicht kommen und Daniel Elias hat letzte Woche Corona bekommen und konnte deshalb auch nicht kommen. Und die beiden haben ja, die Kasse geprüft und haben dann auch eine Mail geschickt. Und ich würde Klaus-Jürgen bitten, vielleicht das Ergebnis einmal kurz vorzustellen. Und ja, bitte, Klaus-Jürgen.
Hier ist sozusagen Dominik und Daniel meine Stimme. Lieber Thomas, lieber Edgar, liebe Mitglieder, Daniel Elias und ich haben die Kasse der DGFO in den letzten Tagen für den Zeitraum 2018 bis 2021 geprüft. Edgar hat sie sehr gewissenhaft und ordentlich geführt. Die stichprobenartige Durchsicht der Unterlagen ergab keine Unstimmigkeiten. Genau. Die beiden wollen schriftlich den Antrag stellen, den Vorstand zu entlasten. Das bitte ich jetzt aber euch und würde das mündlich hier sozusagen tun. Das heißt, aus den Ergebnissen heraus den Vorstand entlasten wollen und euch um diese Entlastung bitten. Ich denke, ich bin jetzt nicht ganz sicher, wie wir das online mitmachen, aber wir können, glaube ich, hier per Handzeichen abstimmen. Und ich bitte darum, dass diejenigen die Hand heben, die für eine Entlastung des Vorstands sind. Vielleicht können wir online einfach im Chat abfragen, ob jemand dagegen ist. Kann man das? Ja? Okay. Ich glaube, ich weiß nicht genau, wie ihr protokolliert, aber wir können ja auch die weitere Frage stellen, ob jemand sich irgendwie enthalten will. Hier haben wir 2 Enthaltungen. Entschuldigung, Jens habe ich leider gar nicht gesehen. Ja, 3 und Genau, Gegenstimmen hatten wir ja gerade abgefragt. Gut, dann sehe ich den Vorstand als entlastet an und Thomas hat mich gebeten, einen Schritt weiter zu gehen, nämlich die Vorstandswahlen auch zu moderieren. Das wäre jetzt euer nächster Punkt auf der Tagesordnung. Okay. Da ist zunächst jetzt die Frage zu stellen, macht ihr das en gros oder einzeln? Gegenstimmen dazu? Dann wählen wir den Vorstand komplett. Allerdings müssen wir jetzt die Vorstandszusammensetzung nochmal darstellen. Ich glaube, der Vorsitzende und der sozusagen der Ersatzmann, ihr beide bleibt. Und Jens hat aber jetzt den Posten gewechselt, hat den von Georg übernommen. Und der Raimund Klatt, ist der eigentlich anwesend?
Nein, er ist nicht anwesend, der konnte heute nicht.
Okay, aber er hat seine Zustimmung gegeben.
Schatzmeister wieder zu fungieren als Kassenwart. Ja.
Okay, dann kennt ihr alle die Zusammensetzung des alten, in Anführungszeichen, neuen Vorstands mit dem entsprechenden Wechsel. Dann würde ich tatsächlich die Frage stellen, wer ist für die Wahl dieses Vorstands? Der hebe bitte die Hand. Zählt das eigentlich einer durch oder muss ich das machen? Ich sehe aber nichts. Nochmal die Hände heben, bitte. 1, 2, 3, 4, 5, 6, 7, 8, 10, 11, 12, 13, 16 habe ich jetzt gezählt. Enthaltungen bitte. 4, Entschuldigung, 4. Wer ist dagegen? Ohne Gegenstimmen? Müssen wir das jetzt im Chat oder im Online auch noch machen?
Ja, zumindest, ob es vielleicht Gegenstimmen gibt.
Ja, zumindest die Gegenstimmen können wir abfragen.
Ja.
Gibt es da Reaktionen? Kannst du, nein? Okay, gut. Dann gratuliere ich dem alten, in Anführungszeichen teilweise überwiegend neuen Vorstand ganz herzlich zur Wiederwahl. Und übergebe direkt wieder. Danke.
Ja. Genau, wir nehmen die Wahl an. Ja, Klaus-Jürgen, vielen Dank. Genau, der nächste Punkt, der auf der Tagesordnung steht, das ist die Satzungsänderung. Die hatten wir schon vor einiger Zeit mal vor, diese Satzungsänderung umzusetzen. Und zwar steht bislang in Artikel 7 der Satzung der DGFO zum Thema Mitgliederversammlung. Die Mitgliederversammlung findet mindestens einmal im Jahr statt. Und das ist ja nicht der Fall, weil unsere Tagung auch nur alle 2 Jahre erfolgt. Da gibt es eine Anmerkung? Eine Gegenstimme, okay. Okay, gut. Ja, genau. Und genau, es ist nicht der Fall, dass wir jedes Jahr tagen und deshalb haben wir auch nicht jedes Jahr eine Mitgliederversammlung. Wir schleppen das schon seit Jahren mit uns rum. Also rein rechtlich ist das nicht ganz sauber, aber weil wir so ein kleiner Verein sind, interessiert oder hat das bislang nicht wirklich interessiert. Aber es wäre trotzdem schön, das Ganze auf solide Füße zu stellen. Wir treffen uns alle 2 Jahre und daher ist der Vorschlag des Vorstandes, diesen Artikel zu ändern. Also die Mitgliederversammlung findet nicht mindestens einmal im Jahr statt, sondern die Mitgliederversammlung findet alle 2 Jahre statt. Und dieser Entwurf dieser Satzungsänderung, der ist auch schon vorgestellt worden in der Ankündigung der Tagung, in der Ankündigung der Mitgliederversammlung in der Articulata. Das ist auch wieder wichtig, damit es rechtlich alles in Ordnung ist. Und ja, da wäre die Frage eben an die Mitglieder, wird da die Mehrheit zustimmen, diesen Weg zu beschreiten? Gibt es Gegenmeinungen, Enthaltungen? Prinzipiell alle dafür. Ja, ein klares Votum dafür. Gut. Okay, dann ja, können wir weitergehen und zwar kommen wir dann zum Punkt Verschiedenes und zwar ist das die nächste DGFO-Tagung und die wird aller Voraussicht nach an der Uni in Gießen stattfinden. Das ist relativ zentral in Deutschland gelegen. Für viele sollte es besser erreichbar sein, als es jetzt diesmal hier in Leiden der Fall war. Und die Organisation wird die Hessische Gesellschaft für Ornithologie und Naturschutz übernehmen. Und das sind insbesondere der Stefan Stübing, Inga Hundertmark und der Leo Meyer, die das in die Hand nehmen wollen. Und das Datum steht auch schon. Die Tagung soll vom 22. bis 24. März 2024 stattfinden. Und wir achten ja immer darauf, dass es keine Überschneidungen gibt mit der Tagung der Gesellschaft deutschsprachiger Odontologen, wo Klaus Jürgen der erste Vorsitzende ist. Und die Tagung findet immer eher statt. Die findet, glaube ich, immer am zweiten Wochenende im März statt. Ja. Okay. Wunderbar, das hört sich gut an. Und dies wäre auch sogar schon das 4. Wochenende im März. Also es ist sofort, der März startet sofort mit einem Wochenende. Ja, also das ist der Punkt. Und ich glaube, damit bin ich eigentlich auch schon durch mit den Punkten, die hier auf der Tagesordnung standen. Ist die Frage, gibt es noch weitere Dinge, die besprochen werden sollten, die relevant sind? Sieht nicht so aus, ja. Im Chat tut sich auch nichts. Gut, der ist weiter tot, halbtot. Ja, gut, dann bedanke ich mich bei allen Mitgliedern und wünsche uns weiterhin noch eine angenehme Tagung. Vielen Dank.